Super. And you? This is
Good morning, everyone. Thank you for being here. It's a great pleasure to open this plenary session, the first one of today. I apologize that my voice is a little bit lower. I think I spoke too much yesterday. But to announce our speaker of today, you really don't need to shout. I think his reputation and his name is already well known to most of you, if not all of the food microbiologists. We spoke about Luca Coccolin from University of Turin, a member of the executive board of ICFMH, member and a high actually contact person of EIT in University of Turin, active probably in food microbiology throughout all the continents, active in so many different areas of food microbiology, including pathogens, including spoilage, fermentation, you name it. His publication speaks for itself, right? So without any further ado, I would like to call Luca Coccolin, colleague and friend, to take the floor.
So thank you very much, Andrea, for this really inspiring presentation. Thanks a lot for the words, and good morning, everybody. Actually, it's quite amazing to see how many people are attending. I know that Athens and the nightlife of Athens is offering a lot of opportunities, a lot of possibilities, and Greeks usually they go in bed late at night, so you had the possibility to join. Greeks, for sure, you went in bed like me yesterday, pretty late in the night, but that's fine. But let me first say Μεγαλοευχαριστώ το Γιώργο, το Πάνος και την Έφη για την πρόσκληση. Είμαι πολύ ευχαριστημένος. Thanks very much for the invitation, and really, I'm very, very happy to be here sharing with you some of the findings that we had in the last 20 years. Greece has become my second family, because in my second, actually not family, but my second country, because I'm originally from Italy, but I'm married with a Greek. And let me say that this fact also is because of the people at the University here of Athens that really allowed me to collaborate and really allowed me to have the possibility to join their labs, projects, and not only George, but also the members in general. So again, thank you very much for this invitation. Today, what I'm going to be talking is a little bit specific about application of microbiomes, because when we are talking about microbiomes and when we are talking about how these new technologies can help us understanding a little bit how microbial ecosystems are working, we're always looking at, again, populations, we're always looking at ecology, but the new trend is actually go into more specific information, go deep into the possibilities that microbiome allows us to do, and go really to look the strain diversity. And this is something that I'm going to be showing, and I'm going to be showcasing with two cases that we have addressed in the last, I would say, 10 to 15 years, that is at the level of the food, but also at the level of the gut microbiome. But before that, let me just say that when we really started to look at microbial ecology with these new possibilities that we had that actually came from the technological advancements, because I consider myself, like a lot of others, probably also in this room, very lucky because we have been experiencing in the last 30 years a huge, huge improvement on the way we actually study ecology, in the way we are addressing diversity. And if you see here in this graph, the way the, actually you don't see the graph because I see the graph, but you don't see the graph. So if you see the graphs and you see how it has been developing from let me see if I can find the pointer here. Nope. Is it this one? No. Oh, well. The pointer is not actually working, but anyway, that doesn't matter. So if you see from going from plating, counting, and isolation, that I think that everybody here knows how to do it, and this is the original way we do it, going into culture-independent methods, like, for example, DGG is the first one that we have been experiencing, the first one that entered in our labs when we tried to look at ecology. And then we switched with the advent of the next generation sequencing to RNA targeted metagenomics, and then finally to metagenomics and function target ecology studies. This allowed us to change the way we were studying and we were considering ecosystems, because actually what has been happening is that we are not anymore considering single populations, but we are considering consortia. So we have actually to think about consortia in order to discover and in order to understand how the ecosystem is working. However, what is coming is that this kind of approach, I think that I messed it up, because it's not, let me see. So it's not responding, the computer is not responding to what I'm doing, and I don't know if it is a matter of. Yes, now it's working. And the pointer here is not, the pointer is not here? No. Ah, okay, oh, wow. So this is a system where you have to point the screen, and I'm a little bit getting confused. So anyway, so the approach of all these new methodologies allowed us to understand that actually we need to go back to culturing, so we need to go back to the way we were doing things in the past, but more in a targeted way, because what we see at the metagenomic level needs to be confirmed also at the level of the plating, counting, and isolation. And so that's why the first application, let's say, of metagenomics in terms of culturomics allow us to understand what we have to do in the lab to cultivate and to be able to get out of the sample that we are analyzing, the populations that we are interested in. But what is becoming even more important is the fact that through metagenomics and through bioinformatics, we can go into genome reconstruction. So if we do things deep enough at the sequencing level, we can reconstruct the genomes of the populations that we have there, of the species that we have there, in a way that somehow we can go into a culture-independent genotyping. And this is extremely, extremely important because, as you know, and as I'm going to be repeating it at the end of my presentation, culturing introduces a lot of biases, and the ecology that we can get out of a culture-based method can be very different from the one that we are getting with a culture-independent method. And specifically for genotyping, this is, in my opinion, a very important opportunity that we have, especially when we get into specific domains, like, for instance, food safety. So this is a little bit the way I wanted to set up my presentation, and in order to show how these approaches are important, I'm going to show you, as I said before, two cases. The first one is about fermented sausages. Fermented sausages have been a subject of investigation of my lab for more than 20 years now. So the first thing that we have done with fermented sausages was to actually see how microbiome applications or microbiome studies could be applied in order to better understand what was going on during the fermentation of the sausages. And let's say, expectedly comparing the ecology reconstructed from metagenomic studies on spontaneously fermented sausages and inoculated fermented sausages, you could really see here, for instance, that when we have an inoculation, the strains that are added or the species that are added as starter culture are dominating, while if you have a spontaneous fermentation, you have much more species, you have much more diversity. This is something that we would expect. I mean, no big issue about it, no big news about it. But again, it was nice to see how, there is really this difference. What, in my opinion, is a little bit more interesting is going a little bit more down and really digging down into the metabolic activities of those species or those microorganisms through a network analysis. So here what you see is a network that we have constructed comparing or correlating the activities related to the protein, carbohydrate, and lipid metabolism with the different time points. And what it is rather interesting here is to see how the sample of the inoculated fermentation at 3 days is actually very strongly correlated with a lot of different metabolism, not only carbohydrate, mainly carbohydrate, but not only carbohydrate, while the samples that, while the samples coming from spontaneous fermentation, you see that a little bit are moved and they are not strongly correlated, or at least the correlation comes later. So the samples at the beginning of the fermentation are not strongly correlated with some of the metabolism, like in the case of the inoculated fermentation. So from this graph here, you can really say that, okay, if we inoculate, we have metabolisms that are bursted, and that are burst actually, and the fermentation that comes as spontaneous is a little bit slower. This is something important to understand, especially if we take into consider another important aspect that is actually the sensory properties of these products. So looking into volatilome, what we have seen was like a pretty strong production, especially at the end of the fermentation, of some compounds like acetic acid and ethyl acetate that are giving very, let's say, pungent notes to the sausages, but also other products like metabolites like acetoin. And if we compare those results of the volatile organic compounds with the sensory properties of those products, I mean, you can see that the inoculated fermentations, actually, they are scoring lower than the spontaneous fermentation. So the spontaneous fermentation, for almost all the indicators that we have taken into consideration, are scoring higher than the ones of the inoculated. And also, if you go into general flavor or odor, you can see how the spontaneous fermentations are actually scoring better than the inoculated fermentation. So inoculation of starter cultures is important, especially if we get in terms of safety, for instance. But we do not have to forget that when we are using starter cultures, we really need to manage them correctly, because most probably what has been happening in this study is that the company, because again, I didn't tell you that this is something that has been performed at the company level, at the industrial scale, did not manage well the starter culture. So the starter culture were too fast, they were acidifying too fast, and they were also impacting in this way the fermented sausages. This was the preliminary study that we have done within a European project. Within a European project in the Eranet area, we dig down and investigated a little bit better how specific metagenomics are actually driving the spontaneous fermentation of the Italian sausages. So here we are talking about the same fermented sausages, we are talking about the same plant. What we have done was to ferment, or actually to follow natural or spontaneous better fermentation in three different seasons, actually 3 different months, 3 different batches, and you can see how the ecology is actually different. You cannot read, but just take a look at how those colors are developing. We have specific populations that are driving, or that they are somehow guiding the fermentation in the different months, and specifically the fermentation in March is actually influenced very much by the presence of this Latilactobacillus curvatus that we did not find both in February but also in May. So also here, probably it is expected that if we do things naturally or spontaneously, probably it's going to be hard to replicate, and it's going to be hard to have exactly the same behavior. This is why we're using starter cultures, right? But it was like a kind of surprise in that, we had this great effect coming most probably from the raw materials that the company was using, and that was driving again, or was guiding the development of specific populations that at the end of the day, as I'm going to show you in a little bit, really impacted on the final sensory characteristics of the products. Taking advantage of the metagenomics, what we have done, as I told you before, was to reconstruct genomes. And here you see on your left the genomes, or actually some of the key genes coming from different populations of Lactobacillus, but also from Pediococcus, because Pediococcus acetylactis was actually, the Pediococcus, sorry, was actually pretty much present in the populations, or in a case, in the ecology of those products. But what I would like to highlight here is actually the difference of the genomes and the genomic setup of the Latilactobacillus sakei that were present in the productions of February, March, and May. You can see that there are different profiles. You can see that that strains that are driving fermentation in the different months actually have different setup. And this is important to understand, and this is important to take into account in order really to have a kind of nice view or nice picture of what is happening inside of your fermentation and your fermented sausages, because this is something that can be used further when we go into pathway definitions. And so we reconstructed pathways. I mean, we saw, for instance, that there were differences in the production in the different months of several compounds that are really impacting on the sensory profile of the sausages. Especially here you have this ethylpropanoate, but you have also methylpropanoate and butanoic acid that actually give like a kind of that affect negatively the sensory properties of the sausages. And at the end of the day, what we can do is really, using fancy bioinformatics, go into this kind of correlations where we have the possibility to connect populations. We need to have the possibility to connect also metabolites in order to really understand what is responsible for what, or who is responsible for what. So from this nice representation, we could see that there are some specific compounds that are highly correlated with some populations. And here, what I want to underline is the rather negative impact that Lactobacillus curvatus had on the production of March. That was the one that actually the company rated lowest in terms of sensory quality. You would say, so what? I mean, we have all this information, so how can we then explore further? What we have done, basing our further investigation on these studies, was to go into the selection of starters, selection of strains for starter culture production. What we have done was to select 7 different, actually we constructed 7, you would say, synthetic communities or mixtures of mainly Pediococcus pentosaceus and Lactobacillus sakei. For the Staphylococcus xylosus, we always used one strain that was the one that, in our opinion, was the best. And what you see here is a meta-taxonomic graph where you can see that at the end of the day, when we are inoculating the starter cultures, so the Pediococcus pentosaceus, the Lactobacillus sakei, and Staphylococcus xylosus actually are the ones that we see them at the end of the fermentation. We didn't follow them throughout the fermentation. This is, again, industrial production, so we didn't do anything in the lab, but we did everything at the company level. And when we went into sensory properties of those different cultures, there were some, or consortia, there were some actually that really catch the attention of the company. And now we are in the process of really going ahead with the development of the industrial production of the starter culture in order to implement those mixtures, or in any case, those starter cultures in their production. As I said before, the second part of my talk is going to be devoted to this big topic that we have started to look at about 10 to 15 years ago. This is a very well-established statement because diet influences gut microbiome. So this is, again, established. But through the studies that we have done in collaboration with some universities in national research projects, we really could understand how diet not only, let's say, shapes the microbiome, but also shapes the diversity at the strain level. The first thing that we have done when we started this project was to go probably in the most simple, let's say, case scenario that was looking at diversity in populations that were vegan, vegetarian, and omnivores. This paper here was published by using a very low and, let's say, poor resolution method that was the DGG. However, already from this graph, you can see that there is no clusterization. There is no, let's say, differentiation of the different subjects based on the DGG profiles. So vegans are mixing with vegetarians and are mixing with omnivores. Yes, probably you can see some level of differentiation here, some level of differentiation here, but again, those are very strong speculations. And what it is most important is that this picture that we got with the DGG was actually also confirmed when we applied more high resolution methods that was the metataxonomic. So this is a paper that the group of Daniele, Corina, and Francesca that is here in the audience actually developed using rRNA metabarcoding. And you can see that actually there is no difference when we look at the profiles got from the sequencing in the case of the omnivores, in the case of the vegetarians, in the case of the vegans. And this is not actually possible to correlate it if we go into the adherence of the Mediterranean diet. Here we also looked a little bit on how the diet is adhering or not to the Mediterranean diet. And as you can see here on this cake here, it was interesting to see that also some omnivores actually are highly adherent to the Mediterranean diet. So you don't need to be vegetarian, or you don't need to be vegan to have a high adherence to the Mediterranean diet. But independently of that, your profiles of the population, at least as they were described through metataxonomics, are not different. And this was a little bit surprising to us, because knowing that diet can really affect the ecology of our gut or intestinal microbiome and macrobiota, we were not expecting a result like that. Surprisingly, when we went a little bit more ahead, and so we switched from metataxonomics to metagenomics, at that point we could start to see differences. So applying metagenomics, you can see that we start to have clusterization. So the omnivores are separating more with respect to the vegetarians and to the vegans. Interesting to point out is that the genes that were driving the differentiation were the ones involved in the metabolism of carbohydrates and proteins for the omnivores. And for the vegetarians or for the vegans, we were surprised to see that genes that were involved with chemiotaxis, flagellins, were highly expressed. And this is actually something that you see here very well. Those are all genes responsible for the encoding of flagella, for instance. And this was also something that could be even more, let's say, emphasized going to metaproteomics. Metaproteomics really differentiated the vegans from the vegetarians and from the omnivores, and looking and digging down in the metaproteomic maps, we could see that, in general, pathways that were going into short-chain fatty acid production, but also group B vitamins, were all pushed pretty much, and were all overexpressed. I would not say overexpressed, but they were more expressed when we were looking at the population of the vegans and the vegetarians. We did not stop there. We were happy with the results, but going specifically, and here it comes the strain diversity, going specifically into one population that we saw very predominant, and it is also the literature that says that is very predominant, in the gut microbiota, that is Prevotella copri, we could and reconstructed the genomes from our samples, but also getting genomes that were reconstructing from other metagenomes and metagenomic studies. We could see that there is differentiation. And so strains that were present in the microbiota of vegans, vegetarians, and omnivores are actually differentiating. And this is something that is striking, especially if you see this phylogenetic tree, where you have a clear separation of the strains, actually the genomes that are coming from vegans and vegetarians, and the ones that are coming from omnivores. So again, we do not have difference at the population level or at species level, but if you dig down, then you see that there is a difference. And this somehow is confirming the fact that if you stop at the population level or at ecology level, you actually don't see a difference between vegans, omnivores, and vegetarians. But if you dig down, then you have some species that are really impacting, and they are really driving the ecology or the overall ecology of your ecosystem. I'm just going to finish here by reporting very fast two very exciting news. Not sure if Stefan has arrived. I mean, he said that he was going to be arriving late yesterday evening, but we just got the news that one proposal that we have put in this call of Horizon Europe has been funded. This is a call where we will really look into the link between fermented foods and health by looking at two main domains that are the health. So this part relates to health, this part relates to ecology. You can see that there are a lot of things that we are going to do. So this is a project that will run for 5 years, and so you will hear more about it in the future. The second exciting news is that we are moving in another dimension when we look at the interaction of gut microbiome with our health and with our body. It happened that we started to collaborate pretty well with a group of neuroscientists and neurobiochemists, and we have practically started to design an experimental setup where we can really understand how microbes are interacting with neurons. We know that the classical brain-gut axis is, let's say, bound to chemical compounds or to chemical messaging. However, we somehow hypothesize that the communication does not only happen at the chemical level, it happens also at the electrical level. So what we are going to do is, we are in the process of developing this interface where we are going to be putting together neurons with bacteria, and we are going to be looking at electrical interactions. We are going to be measuring electric signals in a way that we can really understand how the microbiome is actually influencing the neurons. And you know very well that nowadays, especially in the, I mean, can we call it post-COVID? I mean, I'm not yet at the level that we can say that we are out of the COVID. But anyway, in a post-COVID area, when we have all these issues related to mental problems, probably we can modulate the microbiome through the consumption of foods, and then have also benefits at the mental level. So those are all things that I wanted to share with you because I think that they are, again, at least for us and for my group, are really exciting news. I'm just finishing saying that the application of culture-independent methods allowed for a better description and understanding of microbial ecosystems. Again, this is, let's say, very well established. However, what I wanted to point out, especially in the second slide, is that probably we need to go back to the concept of the single strain, or in the case of the strains. And so we have to move, now that we have moved, or actually we have well understood, or let's say we are trying to well understand how the community is working. Probably we need to go back and we need to dig down at the strain level in a way that we can understand how the single strains are driving and are impacting or are influencing the overall, the whole microbial community. And the other thing is that metagenomics really, especially when they are done in a deep way, they allow for, again, I want to stress this term of the culture-independent genotyping. Because here we will really see the strain diversity without the bias of the cultivation step. I'm closing. I just want to thank the group. So this is the core group of my lab is Calliope, Ilario. Irene was a postdoc that did most of the work with the fermented sausages. Christian is involved in the neuron microbiome interaction. Calliope is partner not only in life but also at the lab level, and Ilario is the one that is responsible for the bioinformatics. A long list of collaborations, specific thanks to the University of Naples. Again, Francesca is here, Daniele Ercolini, well-known scientist at the food microbiology level, University of Bolzano, University of Bari, the Bretagne Occidentale University, the coordinator of the ProMed Food, the RMNet project, and all the institutions that gave us funding. I'm just closing with another important announcement. For those of you that are interested, in Aberdeen next year, there is going to be the European edition of the International Association for Food Protection meeting. I'm chair. This year I have the honor of being chair of the organizing committee, so please take a look at the deadlines if you are interested to submit proposals for symposium or for roundtables. There is the 4th of October, while if you're interested in submitting abstracts for technical presentation or poster presentations, the deadline is the 17th of January. And with that, I'm thanking you very much, and I'm here for questions. Thank you.
Thank you, Luca, for the marvelous presentation and such an insightful talk that you just gave us. I think the approach that you were showing and discussing now really allows us to look at the microbiome better of a food product, but even within the human body, at least on 3 different aspects. Who is there, what can they do, and what actually do they do? I think with the several different tools that you were showing, you can actually answer those questions. Linking it to the volatile and some of the proteomes, you actually really deciphered those possibilities. Now, that brings me to the question from the audience, where Chinazo Martina Eze asked, well, she would like to understand, does it mean, in your opinion, Luca, that spontaneous fermentation is better than inoculated fermentation? That's point 1, especially bearing in mind that the spontaneous fermentation are not controlled or less controlled. And then she would really like if you could throw some light from your expertise on that. And how can this spontaneous fermentation maybe be controlled?
No, but this was, I mean, when we saw these ecological maps, so we have 3 productions in different months that actually are rather different. We were a little bit surprised because it's okay, we are in the same setups. Of course, the meat is different because the meat is different, but we have so much influence of the raw materials on the different developments that actually is somehow, I would not say destroy the principle of the spontaneous fermentation is better than the inoculated fermentation, because this is not something that we need to do. Actually, spontaneous fermentation is different because it's spontaneous. So we know very well that spontaneous fermentations are very hard to manage, are very hard to control, and so that's why we started to use starter cultures, right? But what I think it is important here to underline is that we really need to have or to be mindful when we are using starter cultures, because it's not the fact that we are using starter cultures, then our product is going to be marvelous by the quality point of view, by the safety point of view, because if we are not able to manage well the starter culture, then that is a problem. So the fact of going into the use of starter culture is like, let's say, the first step that should make the producer aware that they need to do further implementations or further checks in order to allow the starter culture to work properly. Let's say that, how can we control? I mean, the way that we have been using in the last 10 years is actually go into isolation of autochthonous or indigenous populations and try to reconstruct the populations or reconstruct the consortia or the ecology. So here we get into the very big and developing area of synthetic communities. Synthetic communities is one of the possibilities that we have. The other blowing and I would say exploding possibilities is actually the use of microbiomes. No one has investigated the possibility to store, to propagate, and to reuse microbiomes. So if we have, for instance, a product that has quality characteristics, safety characteristics that are defined by a specific microbiome, why not try to store that microbiome, propagate it, and then reuse it? This is, probably you can go back to the very classical and very old principle of the backsloping. This is exactly what we do. Because if we save part of the product and we reinoculate, actually what we do is actually reinoculate the microbiome, right? But we try, or at least we need to do it in a more, let's say, controlled or scientific way. We have a project that is going to be starting in the fall about it, and we are going to be investigating different food products, but also at the level of the environmental, for instance, and at the level of the human, how this can be done. So, again, like complicated, but I think that there is a lot of room for improvement, also by the point of view of the knowledge.
Yeah. Thank you, Luca. Any more questions from the audience at this moment? Yes, please. There in the middle.
Thank you, Luca, and congratulations on your project. Fascinating talk, really. And I have a question regarding the second part, where you link with the gut microbiome. And I was wondering if there is also a direction in your field from scientists like yourselves or Danilo to close the link by studying also the relationship with the genotype, because there has been some relations, or at least very preliminary relations, saying that the human genome also is driving and interacting with the gut microbiome.
Yeah.
So I'd like to hear more.
This is another. I mean, you know what? When we started to look at the, let's say, influences of diet, we needed to make assumptions and we needed to simplify the system for sure. I mean, we know, for instance, that the diversity here, I don't know how many we are, but probably all of us have a different microbiota and a different, probably, I don't know how different is going to be the microbiota, but the microbiome most probably is going to be different. So for sure, there is an element of complexity that we didn't yet, or we were not able yet to control. When we get into genotyping, I mean, I agree with you that this is an important aspect that has to be taken into consideration. There are some very famous studies about, you know, twins and things like that. But again, when you are moving to population studies, this is something that is very, very complicated to control. What I can tell you is that experiences have shown that when you perturb a microbiome is changing. If you take away this perturbation, microbiome goes back. The typical example is about probiotic supplementation. So if you do, you know, the probiotic supplementation, and you go now, these days is very hot talking about superfoods. I mean, everything is a superfood. So, but when you go on intervention with a superfood, you see a difference. Microbiome is changing. When you take away, microbiome goes back. How is it going back? Is there a memory? Where is this memory kept? Is it at genomic level? So, you know, this is something very, very interesting that, but again, at population level at the moment is very, very complicated.
There are a number of other questions, but I think we are running a little bit out of time. So I'm looking at the organizers. Do you allow a few more questions, or do you think that we have to continue? Well, maybe one short question there, because I see next to George, and I think George takes responsibility for it.
Konstantin, Jenny Georges, University of California, Davis. I'm kind of a prehistoric creature telling you prehistoric stories. And I'll go back to 1974, thanks to a lady who had diarrhea, staphylococcal food poisoning from Italian dry salami. We embarked in 10 years of studying the ecology of those products. And one of the first things we identified was the international cultures could not work simply because they could not adopt that environment. So eventually then we studied the ecology, isolated the bacteria, developed the technology to prepare the cultures, and eventually were able to make Italian dry salami in the desert by controlling the environment. So environment is very important then, because it drives which one is going to come first, which one is going to be second, which one is going to give the flavor compounds.
Yeah, absolutely.
That's all. Thank you.
Thank you. Thank you.
Okay, thank you all. I think the discussion could continue. Luca, that just shows how
I'm
inspiring
going to
the
be
talk
here.
was, but don't miss the opportunity to talk to Luca just before. And before Luca leaves the stage, I have something for you in the name of the organizing committee.
Wow, thank you. So my acknowledgements at the beginning were, well, gave a good result at the end.
That said, I will leave the chair to the next people.
So, welcome to this first session. The first speaker of this session is my co-chair, Pascal Bonnard, who is a research director at INRAE in France and deputy director of the Safe Food Unit. Expert in fermentation of protein-based matrix of different origin and also in food microbial communities. The title of his fermentation, his presentation is Fermentation of Protein-Based Matrix by Synthetic Microbial Consortia as a lever to modulate metabolism and overall sensorial perception, an innovative way for new food design.
Thank you. Do you hear me? Okay. So sorry for the long title. So I will start my presentation on this other work we've been doing on fermentation of protein-based matrices. Oops. Yes, let's start. So I will make an introduction, some element of context, and then I will move to the experimental strategy we've been developing to design PE-based fermented food. So first, I will develop the microbial consortia design strategy we've been used, and sensorial cartography, what we obtained with different kind of microbial consortia, and special things about off-note degradation and masking effect we've obtained also. And we will finish with a food consumer study we've been doing on this product. And some conclusions. So there is a high interest and potential of plant-based diet. There are many reports, including some FAO reports, like related to sustainable food systems, and also sustainable healthy diets, in which they point out that plant-based diet was very good for the earth and for the health too, and it could be tasty as well. And there are advantages of using this protein from plant origin compared to protein from animal sources. They have a lower CO2 footprint. So there is less greenhouse gas emission effect. They require less energy to be produced. They require also less water, and there is an important point is the health effect that they could have, like reducing cancer risk, diabetes, and hypertension. And one very important point is the ergonomic benefit of plant proteins over protein from animal sources. So by replacing 20% of the ruminant meat by to 2030, we could increase agriculture land. We could decrease, sorry, the agriculture land like 500 million hectare reducing would be a target. So on this table, there is you can see that at the top of the table, there is a carbon footprint and land use related to different kind of protein sources. And we see that beef is at the top, so it's worst, the worst protein source to be produced in terms of carbon footprint and land uses. And at the bottom of this table, you can see that you have pulses like legumes, and there is at least 10 times less CO2 effect impact compared to meat, for instance. So legumes varieties could offer an evolution of the food offer, but there are still some obstacles to the consumption of legumes. Main one is anti-nutritional factors like phytic acid, galactoside, saponins, and also that the point is these legumes are not in line with the consumer expectation. They have, since they have sensory defects like off flavors, bitterness, and which limits the use of these protein sources as ingredients. And also there is a lack of availability of the product offer, and there is a question about the total or partial replacement of this protein with other kind of protein to make new product, like a milk protein mixed with legume-based, with pea protein sources. So we have to take advantage of our knowledge on traditional fermented food to design synthetic microbial consortia for new fermented food. That's what we've tried to demonstrate, that fermentation could be a lever to modulate metabolome and acceptability of the pea protein-based gels, fermented pea protein gels. So we've tried to demonstrate a concept of formulating fermented foods enriched with pea protein. We studied the impact of synthetic microbial consortia designed by microorganisms from dairy and or plant origin and the sensory quality of pea-based protein gels. And we also evaluate the consumer behavior on this. So the experimental strategy with that is the following. So the first stage was to mix or not pea protein ingredients with cow milk. So we had two concentrations at this stage, 100% pea protein and 50% pea protein mixed with milk protein. And for this matrix, we've designed synthetic consortia adapted to this matrix. We made ripening, and then we go through a product evaluation analysis through metabolome and essentially, and sensorial analysis, taste and flavor. So just, I will go very quickly on this slide, but it's just a point I want to point out. That's it's a structure of the gel. I think one important point is to be aware that we have quite a work to do on the formulation of the gel and associating. Then when at this step we have, especially when you have a mixture of pea gels with pea protein with protein from milk origin, so this is a mixture of proteins. So we have to test different kind of treatment, so thermal, acid, or enzyme treatment, to get a gel which is adapted to the fermentation like a cheese-like fermentation. Then move to the microbial part. So one example of the strategy we've been developed is to make a different consortia, so synthetic consortia, made from different taxonomic family and functional family also. So there are three of them, so Firmicutes family, acid-sensitive bacteria family, and acid-tolerant fungi family. And then this way, we were able to assemble different consortia made with 9 strains, 6 strains, 3 strains, and we had control with 1 strain. And this way, we made fermentation, and we got more than 100 associations tested. And here is just to illustrate how microbial consortia could modulate sensorial properties. That was the first question we were thinking of. And here there is a sensorial cartography, and depending on the different consortia, which are in blue in the graph, you can see that we distinguish different groups, sensory note groups, like fruity flower groups, cheesy group, and chocolate toasted caramel group, and the control was green, so like pea protein, it was a green flavor. So we can see that modulating the microbial consortia can modulate very drastically the sensory perception of the product. And the most complex association was the one with 6 and 9 strain, which produced the most richer sensorial notes, like fruity, cheesy, and roasted ones. So here, just to illustrate how, just on 2 microbial consortia, how we could modulate and have a masking effect on the perception note by synthetic consortia. So at the top of this table, you have the control, and you say that it has been done with GC olfactometry analysis. So we have different odorous zone, like a grilled odorous zone, which is in brown. In green, it's a green flavor. And odorous zone dairy, it's blue. And odorous zone fruity is purple. No, yes, that's it. So you see that the control is green. So it's the non-fermented gel. And with a Megan consortium, you can have dairy notes. And with another one, with another Megan consortium, we can have grilled and fruity notes. So it's interesting to point out that the impact of fermentation is really important. And we can see that the perception of green note in fermented gel disappeared. And the interesting note appeared like a fruity, dairy, and good note. And it could be modulated by the design of the consortium we can make. And here it's even more complicated. It's how the sensory properties could be modulated we've measured the sensory properties, then amino acids produced, so non-volatile compounds produced, and also volatile compounds produced. And we've made a multi-factor analysis of these parameters. So sensory properties are in red, amino acid in blue, and volatile compounds in green. And on the right-hand side, you have the different consortia which has been using. So just to make it short, bitterness was essentially related to the non-volatile molecules like hydrophobic amino acids. Beanie nuts were related to the presence of volatile molecules like hexanol. Roasted grilled notes were mainly associated with different volatile molecules like furan 2-pentyl and furan 2-ethyl compounds. And fruity notes were mainly associated with the presence of alcohol, aldehydes, and esters. So the last point I wanted to point about, it's another way we wanted to show how matrix composition could modulate sensorial properties. So in this case, we kept the same microbial consortium made of lactobacillus and yeast, and we modified the concentration of the pea protein content. So we produced 4 products this way, and we submitted to a consumer panel made of 241 people. And we wanted to see to which extent the mixed animal and plant protein base was differentially perceived by the consumer. So finally, we compare three products enriched with milk protein, but containing a pea protein. So what we noticed that the difference in assessment depending on the pea content. So we've seen that the less concentration of the protein, 25%, was the most popular, and the highest concentration of pea, 75%, was the least appreciated. So that's what we were a bit aware at the beginning of the study. But there is, and also we found higher individual variability in the sensorial evaluation. An important point is to see that the increase in the purchasing intention when the pea content decreases was really neat. And after the information on nutritional benefits, we can see that the increase in purchasing intention for the product with 25 and 50% P increase. And also after information on the environmental benefits, there is an increase in the purchase intention only with 50% P content product. And there is no effect of the information for the product with 75% P. So it's the main conclusion we've been. So let's go to my final conclusions and future perspective on this kind of approach. So we found that it could be, it's really possible to modulate sensorial properties through the design of microbial consortia. And also mixed gel fermentation could be an interesting option to reduce sensorial defect of pea while keeping some familiar sensorial attributes such as those of dairy notes. So I think that it could be interesting to develop this approach. So we could move to new products inspired by traditional fermented food, as Luca pointed out in his presentation. So we could further develop approach to design synthetic consortia adapted to the new matrices, to design new fermented product meeting consumer expectation, because for the moment, on pea-based protein product, it could be improved. But there are other sources of proteins that could be of leguminous that could be explored. So to explore it could be also possible to explore the largest possibilities for innovation in terms of taste, aroma and flavor and texture, and also to better target the customer expectation about fermented plant-based food products. And again, I think that hybrid product could be an interesting option to develop. So I mean by hybrid products like plant mixes. Plant could be legume, cereals, pseudo cereals, and plant-animal mixtures could be also milk and insect protein mixtures. So thank you for your attention, and I would like to, I have a special thanks to my colleagues who participate to these studies, and also the program related to these studies are cited here. Thank you.
Thank you, Pascal, for this interesting presentation about these new fermented foods that are probably the future of fermentation. Any question from the audience? I don't see. Oh, one there.
Hi, thanks so very much for that very, very interesting presentation. I have a little question, in fact, two questions. I noticed that your sensory appreciation scores were kind of on the I don't know kind of level at 4. Can you tell me what the French consumers' appreciation of that type of cheese might be? And do you have any indication from your consumer study of subcategories of appreciation? Were there vegans in your consumers that might have liked it more, so that you could subcategorize and maybe target your consumer base as well?
So concerning the category of vegan, we don't test this, so it was we don't have the answer for this question at least. And the first question was, for instance, did we have a control with real cheese with real milk? So we didn't do this. So I think they would have higher scores.
Yeah, but do you have
sure,
maybe from another study maybe with a French population cheese score? Like is
no.
cheese typically 6 or 7, or is there anything that in the literature it can be compared to?
I think so. I'm not a specialist in the social report, but I could give you the contact to the person, and she will be able to give you a very precise answer on
That
this
would be great.
typical
Thanks very much. Thank you.
There is another question there.
Very, very fast. Do you have any insights on the nutritional values of those new products?
No, we didn't study this yet. So, but there are people. I mean, we are going to work with another lab on the nutritional part. So they will do it, I think, in different programs. But so far, it hasn't been done.
I don't know if we have the time for a last question coming from the web. So Wellington Pinto is asking, how do these consortia take into consideration the safety of the culture? What have you done to guarantee the safety?
It's a complicated one, but I have some element of answers. The safety, so at the beginning of the project, since we were doing long fermentation, we had some problems with contamination of spore-forming bacteria, like Bacillus ones. And we had to develop a strategy to get rid of this. So there were two strategies, so to adapt our sterilization procedure. So we do this by using tendalization. And also we coupled this with the design of the consortia, and we've seen in some cases that the consortia we've been designed developed quicker, and this way he counteract the development of the spore-forming bacteria. And this is a very important point, especially for long fermentation studies like cheese-like fermentation. But we didn't have this kind of problem because I didn't present this work, but we've also done work on yogurt. So it's really acidification, and it's really short fermentation. And here we did not have this kind of problem. So I don't know if I answer the question, but that's
Okay.
I can say on this.
Thank you, Pascal, again, and we can move ahead.
So we'll move to the second speaker, so Francesca De Filippis. So Francesca is a microbial ecologist and expert in the application of NGS to study microbial communities in complex environments, including food. And she's an assistant professor at the Department of Agriculture and Science in the University of Naples. And she will make a presentation called Metagenomics Unravel, the Microbial Heritage of Italian Typical Fermented Foods.
Thank you, Pascal. Basically, we know that lactic acid bacteria are an important part of the microbiome of several fermented foods, And if we consider the presence, the pattern of lab species in different fermented foods, sorry. No. Oh, that's, okay, I found it. So if we consider the pattern of species, of lab species in different fermented foods, we can see that we can appreciate a different profile in different types of fermented foods. What we know about the cheese microbiome is that it's, as we saw also in previous presentations, the microbiome in fermented foods and also in cheese is extremely important for the production and the ripening of cheese. And usually in each cheese type, few lab species dominate, but at least in natural fermentations, we can often observe several strains, so a high diversity at strain level. So the aim of the study that I want to present today is if we can identify a specific microbiome, a microbiome that is specific for each cheese type, and so if the microbiome is part of the so-called cheese terroir, so the whole all the traits, all the linked with the geographical origin of the cheese that contribute to the flavor and to the specific sensorial profile of each cheese type. And in particular, if there are specific lab species or a specific strain pattern that is specific for each cheese, with the aim to develop a model to trace the origin of the cheese. So use lab species or lab strains as markers of cheese origin. So with these aims, we collected around 200 samples of cheese, of different Italian PDO cheese. We had different, very different type of cheese, so pasta filata cheese, where the card is stretched without water. Before the molding that are fresh, like the buffalo mozzarella that for sure all of you will know, or in some case these cheese are ripened, and so we have provolone and caciocavallo cheese, but also long ripened cheese like Grana Padano and Parmigiano, where the card is cooked before, during the production, and are ripened for more than 12 months. So we collected several samples of different types of cheese. For all of these samples, we extracted the total DNA and carried out shotgun metagenome sequencing. From the taxonomic point of view, so from considering the taxonomic composition of the microbiome, we can see that considering the most important species, we can cluster the samples according to the processing technology. And in some cases also according to the cheese type, like here we can see mozzarella cheese, where Streptococcus thermophilus was dominant, also Grana Padano and Parmigiano Reggiano, where there was a quite high abundance of lactic acid Bacillus rhamnosus, and going more in detail, we can see that Lactococcus lactis was really abundant in sheep milk cheese, so the so-called pecorino cheese, but also in semi-cooked card cheese, so where the card is cooked is heated, not cooked, at a lower temperature, below 48 degrees. But also Lactobacillus helveticus that was dominant in long-ripened cooked card cheese, so Parmigiano and Grana Padano, but also in fresh cheese like mozzarella. And finally, Lactobacillus buchneri that was really low, showed a really low abundance in all the cheese type except ripened pasta filata cheese, so Caciocavallo and Provolone, where it was quite high. We also focused, as Luca showed before, on the functional potential of the metagenome, so on the genes involved in the production of flavor, of the typical flavor of the cheese. We can see here genes involved in the proteolysis, so in the degradation of proteins, that, as expected, were abundant in ripened cheese, while they were really low in blue cheese, so basically Gorgonzola, where on the contrary there was a high abundance of lipase and genes involved in the production of thioesters that are a typical sensorial trait, that are typical sensorial molecules in this type of cheese. We also focused on genes involved in potential probiotic activity, since, as we know, several lab species are recognized as probiotic. And in particular, raw milk cheese showed a high abundance of genes involved in the biosynthesis of the pilus, that is one of the factors contributing to the persistence of a strain in the gut microbiome, so to the bound of the strain to the gut epithelium. While genes involved in the production of gasericin, that is a bacteriocin, were overexpressed in ripened pasta filata cheese. And again, genes involved in the production of exopolysaccharides that are also a factor contributing to the persistence of the strain in the gut were quite high in all the ripened cheese, except for long-ripened cooked card cheese, so Grana Padano and Parmigiano Reggiano. And finally, genes involved in the biosynthesis of vitamins, like genes involved in the production of vitamin B2, were particularly low in fresh cheese, while they showed a high abundance in all the ripened cheese type. And on the contrary, in fresh cheese, we can see a high abundance of genes involved in vitamin B6. As Luca showed before, with this approach, we can reconstruct genomes from the metagenomics reads, so those that we call metagenome assembled genomes, MAGs. And we can cluster these genomes according to their similarity to identify the species-level genome bins. We identified in this cheese around 230 species. Here I reported those showing the highest numbers of genomes reconstructed. And we tried to understand if different strains are associated with different cheese type. Here I showed the example of Lactobacillus delbrueckii, where we identified at least three different subspecies, so 3 different strains present in our samples. And as we can see in these 3, one strain was exclusive of Grana Padano and Parmigiano cheese. Another one was found in sheep milk cheese. And also, when we consider buffalo mozzarella, we could identify 2 different strains according to the production area, because buffalo mozzarella in Italy is produced in 2 different geographical areas. The samples from these 2 areas showed different strains of Lactobacillus delbrueckii. The same applied to Lactobacillus helveticus, where we found 2 subspecies, so 2 strains, and one was typical, again, of Grana Padano and Parmigiano Reggiano. And finally, with Leuconostoc mesenteroides, where we identified 2 strains, and one was, except for a few samples, was present exclusively in Asiago, that is another PDO cheese. So in conclusion, this is one first step towards the identification of the cheese terroir. So starting to consider the cheese microbiome as part of those traits that we have to consider when developing a PDO product, so a PDO label. And a specific microbiome is, of course, linked to the sensorial traits of the cheese, but also to the potential probiotic activity. And as Luca said before, we will go deeper into the link between the fermented foods and the gut microbiome in the next years with the Domino project. And then these results can be useful and can be further explored to develop novel models for the tracking of the origin of PDO cheese, and of course also other PDO fermented foods. Thank you for your attention.
So, other question? Yeah. Where's the microphone? Yes, it's coming. Yeah.
Hello? Yeah. So you link the presence of different genes to different metagenomic samples, in this case different cheeses. But did you also try to link the presence of these genes to the reconstructed genomes? Because I guess it might be difficult when you have different strains in a single mark.
Yeah, this is one, well, the results that I presented are really preliminary because we are going deeper in the analysis. But yes, it's the next step is to understand which strain do what. So which genes are really present in a specific strain, and then we also have the isolates from this cheese. So then the further step will be to understand if what we found in the metagenome, and in the mags is confirmed also phenotypically.
Yeah, there is a question from the web. So can we say that this is possible to have a microbial signature depending on the cheese?
Well, we are trying to demonstrate this. So, in some case, I think it's clearer. Of course, when you try to differentiate really similar cheese that are produced with a really similar technological process, it becomes more difficult. But I think these are the future steps. So trying to implement the microbiome tracing with the purpose to trace the origin of the cheese or any other PDO food.
Are there other questions in the audience? Yeah, Luca.
Go very fast. So the first one, how stable do you think the communities are in order to really say that there is a cheese terroir? Because this is, I mean, this word is actually very, let's say, debated in general. And the other thing is, I was pretty surprised to see how the peptidases in the molded cheese are actually not there. So do you think that is because you specifically looked at peptidases and not more, let's say, general proteinases? And did you get the chance to look at the eukaryotic part also? Because we know that, especially in Gorgonzola cheese, I mean, they play an important role.
Yeah, thank you, Luke. So the first question, of course, we know that there are some variations, but of course, we try to overcome this issue considering a wide number of samples. So now I showed just a preliminary part of the study, but we are going to sample around 1,000 samples. So we hope this will limit the variability according to the factory, according to the raw material, and so on. And then these are all PDO cheese, so you know that there is a clear disciplinary specifying the different steps of production, so all the things should be more or less standardized from this point of view. As for the peptidase, yes, I think your third question is the is the answer. So basically, those are peptidase from bacteria. Because as you will know, it's really difficult from metagenomic threads going to the eukaryotic part. So in that case, it was based on a mapping-based approach. So probably we just extracted the peptidase from bacteria. But yes, for some type of cheese, of course, blue cheese, but also smear ripened, we will move also to the eukaryotic part.
Okay, thanks. So maybe we'll have to move. I had some questions, but maybe... So just one remark. I think it would be very interesting to contact people, because in France they've worked on a meta-PDO cheese program, and it's the same approach as yours, and it could be interesting to share data together just to compare the results. And I also another question is your study on the whole PDO cheese of Italy, or no, it's just a part of it?
No, they are not all of them, but
yes, okay.
At least they are the more, I think, the most important, those with the highest production, because there are really some of them that are really a niche production. Yes. So I think,
okay.
Yeah.
So thank you. So we have to move to the next speaker.
So the third speaker of today is Hannes Deckert. a PhD student at the Vrije University of Brussels in Belgium, working under the supervision of Professor De Wust. And his research deals with the microbial dynamics and metabolomics of Gouda cheese. So his presentation is Tetragenococcus halophilus, a contaminant of cheese production or a potential adjunct culture. Thank you.
Thanks for the introduction, Mrs. Chairman. And good morning, everyone. Cheese is a nutritious and protein-rich food product, but more importantly, cheese makes us all happy. Because when I say cheese, you smile. However, seeing the title of this presentation and more specifically the word contaminant, will you still smile when you see cheese or not? You will find it out in some minutes. First, I would like to introduce you to the 4 different groups of bacteria you can find in Gouda cheese. So mostly the cheese is pasteurized, but if that is not happening, then the raw milk microbiota will of course end up in the cheese and have some effects there. In both cases, pasteurized or not, a primary starter culture is applied, consisting of different Lactococcus and Leuconostoc species. And this primary starter is mostly rotated. So we have different primary starters with another phage profile to prevent phage outbreaks. It is actually known that raw milk cheeses are more flavorful mostly, a bit comparable to the spontaneous sausage fermentations that Professor Cocolin discussed. So to cope for the loss of flavor, some producers add a secondary starter culture. That can be Lacticase or other lactobacilli. And last but not least, cheese is not made in a sterile environment, so you will always have some household microbiota that end up in the cheese coming from the equipment to environment, and they will also have some effects. So in this way, you have 4 different groups of bacteria. 2 starters and 2 non-starter lactic acid bacteria, and of course the non-starters are less controlled, while the primary starter mainly acidifies and also leads to some basic flavors like the buttery notes or the eye formation. The other 3 groups, they lead to more specific flavors which can be desired or undesired. So now that we know the 4 different groups, we go to the research question of our investigation. Is there an impact of the rotation of primary starter cultures with different phage profiles on the microbial diversity of Gouda cheese productions? To answer this question, we investigated 23 different Gouda cheese batches made in the same dairy factory using a rotation of 3 different starters, referred to as A, B, and C. And we sampled them after 36, 45, 75, and 100 weeks of ripening in the cheese core and the cheese rind separately. The metabolites were measured by UPLC-MSMS and liquid injection, GC-MSMS. We characterized the bacterial diversity by culture-independent amplicon-based high-throughput sequencing of the full 16S rRNA gene using the platform of Pacific Bioscience, and we processed the data using DADA2, which results amplicon sequence variants, and that allows you actually to go up to strain level. And last but not least, we also let the cheese course analyzed by cheese sensory panel. So if you look to the average dynamics of the 23 different cheeses, you see in the beginning mainly Lactococcus cremoris, Lactococcus lactis, and Leuconostoc pseudomesenteroides at 36 weeks of ripening. But slowly, Tetragenococcus halophilus is increasing to become the most abundant species at 100 weeks of ripening. And then if we consider each batch separately, we really see a difference between the 3 different primary starter cultures applied, A, B, and C. For example, B has the most non-starters, while C has almost no non-starters. B also has the least Leuconostoc, and probably that's linked with having the most non-starters. The most non-starter actually was Lacticase bacillus paracausae, which is expected. Flash forward to 100 weeks of ripening. There we see in the core mainly Tetragenococcus halophilus. And interestingly, this species was also the most abundant one in the rind at 36 weeks of ripening. Again, here you see that there's less Tetragenococcus halophilus in the C primary starter, again confirming that there is a difference between the different primary starters. We were actually a bit surprised by this high abundance of Tetragenococcus halophilus because it's a quite uncommon species in cheese. It is a very salt-tolerant lactic acid bacterium, actually. It can grow at even concentrations higher than 18%. And to our knowledge, only 7 publications report the presence of this species in cheese, nearly always at low abundances. And 3 additional papers, they report the presence of Tetragenococcus halophilus in cheese brines because of the saltiness. Actually, it has a history of use in oriental fermented foods such as soy sauce, fish sauce, shrimp paste, all very salty products. So it's even sometimes used as a starter and it is considered as safe. Because we found it, publications found it back in cheese brines, we were thinking if that would be the inoculation source in our case. So that's why we plated out the brine of the 23 different cheeses, and indeed, quite high counts on the PCA and the MRS medium, and culture independently on your right, you see that indeed Tetragenococcus halophilus was the most abundant species in the brine. Then we went a bit further on the amplicon sequence variance level. So that's really the exact sequence of the 16S rRNA, the full sequence, with single nucleotide resolution. And that allows you to go really into strain level almost. You see also black dots on these figures. They represent the cumulative relative abundance of Tetragenococcus halophilus in each sample. So for the brine, there was around 37% of Tetragenococcus halophilus. And within this Tetragenococcus halophilus, the 5 orange ASVs were the most abundant ones. And interestingly, these 5 orange ones, you also find them back as the most abundant ones in the cheeses. But you also see in the cheeses that there are a lot of abundant green ASVs. They were almost not present in the brine, so they could have had another inoculation source. So we kept on looking and we plated out the rennet used to coagulate milk. And indeed, we found some Tetragenococcus halophilus in this rennet. So we have 2 different inoculation sources. Now knowing that, we wondered what the effect of Tetragenococcus might be on the cheeses. So here you see the correlations between all metabolites and the most abundant species. It's a heat map, a lot of data always, but you clearly see a different pattern between the starters and the non-starters. The non-starters have more strong correlations with most metabolites, and within the non-starters, Tetragenococcus halophilus really has the strongest correlations with a lot of metabolites, possibly leading to very flavorful cheeses. Interestingly, we also saw a subspecies difference. Maybe you still remember the colors. The 2 most abundant ASVs were green or orange. Now, the copy number of the 16S rRNA gene in Tetragenococcus halophilus is 5. So that means that actually a strain can contain up to 5 different copies of the 16S rRNA gene, and that's the case here. We see 5 different green ASVs and 5 different orange ASVs always coming together, so they represent 2 different strains. And that was also confirmed by shotgun metagenomic sequencing, and we could make 2 metagenome assembled genomes, and they differed quite a bit considering the average nucleotide identity. So interesting to really see these 2 different strains. And they had also different correlations with the metabolites. You see, again, a heat map of the correlations. More specifically, strain A was correlated with a high pH and is probably more acid sensitive, while strain B was correlated with a high cheese dry mass. And only strain A was associated with a low sensory score, so strain B might give a beneficial flavor to the cheeses. So that brings us to the end of my presentation. There was indeed an impact of the rotation of primary starter cultures on the non-starter lactic acid bacteria diversity. That's the answer of the initial question. And in addition to the common non-starters like Lacticase, Bacillus paracasei, we found back Tetragenococcus halophilus in quite some high abundances, especially in the cheese rind and also in the cheese cores after extended ripening. The brine and the rennet served as inoculation sources, and these two inoculation sources probably corroborate with the two different strains of Tetragenococcus halophilus we found. And they had different effects on the cheese, on the flavor more specifically. And that means that in the future, probably a well-chosen Tetragenococcus halophilus can indeed be used as a cheese adjunct culture. So I would like to thank my promoters, Luc de Vuist and Stefan Wex, all the people from IMDO, the financial support, and of course you for listening. Thank you.
Thank you for this presentation. Any question from the audience? I can't see. One there.
Thank you very much. I'm Françoise Leroy from Ifremer, so we know Tetragenococcus in fish sauce. What about the safety aspect of this strain, if you want, of this species, if you want to use it for an inoculation of cheese? What about also the biogenic amine production by this strain in cheese?
Yeah, that's directly the hard subject, the biogenic amines. Well, some strains of Tetragenococcus halophilus are actually used to lower biogenic amine production in these oriental foods because they inhibit other strains that produce a lot of biogenic amines. Actually, in our case, we have indications that one of the two strains might produce histamine. I showed it very briefly, but you could see it in the correlations. So that's, of course, we cannot use that strain, but it's strain dependent and it's only a histamine, so you should use a well-chosen strain, like I said. And if you sequence the strain and you see there is no gene producing this histamine, then it's safe.
And concerning other safety aspects, do you have any idea other safety aspect of the strains?
The general safety, or what do you mean?
No, yes. Do we have, if we want to use it at industrial level, do we have enough proof that this strain is safe for consumption?
I would say because it's used in other, the species is used in other fermentations, it's probably safe. Specifically, if you have your strain, I think you should do additional checks. And yeah, we are still on that process, so yeah, I cannot say more about that.
Thank you.
Any other question? I don't see. No question? No. There is one question online. How did you do selective plating on PCA?
Yeah, it sounds as it's not selective, of course, but actually that's a lie. Every medium is selective. Non-selective media do not exist. You always have a bias. Actually, our project was more culture independently because also not every species can grow or not every strain can grow on plates. But it was just the brine. We were just wondering what the counts might be, and we were quite surprised that these were so high. And indeed, yeah, plating on PCA, maybe it's not that selective, but it was the goal to have as many different species as possible, but still, you should know that you miss a lot as well, because it's media.
Yeah, thank you. Okay.
So I think we have one last question, maybe, unless there is some question from the audience. Yes, you suggest the use of this microorganism as a starter. So my question is the following: which flavoring properties it could give to the cheese? Because you've shown some data on the flavoring compounds, but I didn't.
Yeah, which properties? So it has a lot of high, strong correlations with a lot of free amino acids. So they are key to have a lot of different flavors. They are the start of a lot of cheese flavors. So at least it could be used to fasten maybe ripening, and then we don't really know specifically which flavors it could exactly give, because also here you have the combined effect of the two strains, while then here you have the different strains separately. So we are not there yet to know exactly which flavors, but yeah.
But for instance, they could liberate amino acids, and then the other strain could degrade them in flavor or something like that?
Yeah, that's the idea. Or also, they're also quite linked to some short-chain fatty acids. So maybe in high concentrations that's not desired, but in medium concentrations that might give an extra cheesy flavor that is desired.
Okay, thanks. So maybe we have to move to the next.
To the next one.
Sorry. One last question.
Could I ask a question?
Okay.
Thank you.
Yes.
Okay, so first of all, I would like to acknowledge you for a very good presentation. So we were also one of those that investigated different brines and different dairies, and we find a lot of interesting halophilic microorganisms, also Tetragenococcus halophilix, but also yeast as Debromyces hanseníae. And if you look into the dairy sector, you will see that some dairies, they look upon the brine as the holy grail. I mean, they never pasteurize them, they never do anything.
Was the case?
Then there will be others that microfiltrate the brine in order to get rid of all kind of microorganisms. So what I want to ask you is about what is your general recommendation to the dairy industry in general on how to treat that brine? I mean, how could we maybe open the eyes of people in the dairy sector to the possibility that the brine could have very relevant inoculations cultures? So could you please reflect a bit on that?
That's a very, very interesting issue in our project, actually. I think you mainly have two approaches. Or you can go for an extremely sterile brine with nothing. And then, of course, you cannot have beneficial bacteria or yeast, but in our case, we almost had no yeast in the cheese. Indeed, in the brine, Debromyces hanseníae also, but not in the cheese. So very sterile, but then, of course, it has to remain sterile. You can, maybe it's even quite unprotected if it's completely sterile for some infections. And the other approach is that really you could inoculate it, maybe indeed by some safe strains of Tetragenococcus halophilus and Debromyces and some others, that you know what is there and that it's stable and that it's safe. But, however, it is important to know brines are not, they evolve in time. And that's very important. You always have leakage of nutrients of the cheese going into the brine. So slowly but gradually you have a buildup of all nutrients and you will have microbial changes. So it's important that you follow your brine up.
Thank you. So we will have to move to the next speaker now. Thank you again.
Thank you.
So we move to the next speaker with Louis Delaunay from the Laboratory of Biodiversity and Microbial Ecology, LUBEM, at the University of Bretagne Occidentale. He's currently collaborating. Where is he? He's not here?
Yeah, here I am. Can you hear me?
Okay. I did not see you.
Yeah.
I thought you were here.
Yeah, I'm online.
Sorry for this. And you are currently collaborating with an industrial partner to develop cleaning procedures to eliminate biofilm formed by spore-forming bacteria. Sorry.
Yeah.
So your presentation is entitled Assessing Resistance of Anoxibacillus biofilm to Optimize Sanitation Process in Dairy Powder Processing. So sorry for the beginning, I didn't know that you were
Doesn't matter.
So.
Okay. So thank you for your presentation. So I would like to thank all the organization team to give me the opportunity to present part of my PhD today. So here is a chapter of my PhD to focus on Anoxibacillus and spore-forming bacteria, mainly on dairy product processing. So as you may know, world production of dairy products in 2021 represent more than 13,000 tons, mainly produced in Europe to Africa. Many of these products are exported, like quite half of the European production are exported mainly to Asia. These dairy products are exported because of their low water content, so it could be stored during months and years without any spoilage of bacteria. During the production of dairy products, so you have the milking step, then the storage, the collection, the creaming to remove fats, then the pasteurization at 72 degrees during 15 seconds. So it will destruct the pathogenic bacteria, but this step, this pasteurization step, don't have any effect on spore-forming bacteria. During the evaporation step, you have an increase of temperature close to 50 or 60 degrees. And these temperatures are favorable for the proliferation and the formation of biofilm by thermophilic spore-forming bacteria. These spores are then atomized in milk, and then you have the packaging process which put the powder in content. It's well known in the bibliography that you have mainly three different strains which are worldwide founded in dairy products. So you have Anoxibacillus flavitamus, Geobacillus stautomophilus, and Bacillus licheniformis. During my PhD, I focused on French dairy products, and I found also these three species. So these bacteria can form biofilms in in dairy powder process. So it can form biofilm and decrease the heat treatment of heat plate exchanger. It can clog also the ultrafiltration membrane and also the falling film evaporator, which will decrease the efficiency of the milk powder production. So the first step was to know if our Anoxibacillus flavitamus found in French dairy products can form biofilms. So the first step was to use 31 isolates from French dairy products for my thesis. We added the type strain, the DSM 2641. And we make some culture during 6 hours at 55 degrees Celsius in skimmed milk with stainless steel coupon, as you can see on the left, which is a little piece of stainless steel. We put it in milk during 6 hours. And after the 6 hours, we take this coupon. We make just a little wash to remove all the planktonic cells, make a coloration with acridine orange, and take 10 random picks per coupon to know if the bacteria can form biofilms during these 6 hours. Here you have some pictures obtained. So the red ones are those taken directly with fluorescent microscope. And then we use a software which is ImageG, which transform all the biofilm pictures like this to two colors pictures, white and dark. And what we would like to know if it's the percentage of coverage of all the white pixel compared to the dark pixel. So there you will have a better coverage of the biofilm compared to this one. So here we have the first result. So Anoxibacillus flavitamus. So we tested 32 isolates, and we decided to set a threshold on if the bacteria can cover more than 5% of the microscopic field, it will be a biofilm former. So then on the 32 isolates tested, more than 60% are biofilm former. Mainly of them are 3D biofilm formers. We can show this by the picture taken under the fluorescent microscope. And for the maximum covering yet on Anoxibacillus, we get a percentage of covering close to 35%. We compare this data with 15 strains of Geobacillus stautomophilus So only one strain can form biofilm in this condition at 55 degrees Celsius. But this former can form a biofilm with a high level, with 75% of covering. So the first result was that Anoxibacillus can form a fast biofilm during 6 hours at 55 degrees Celsius. So it can be the first biofilm former in the industry. But the test obtained on Geobacillus tautomophilus can form biofilm more than 75% of covering. So these results was in accordance with one study did by Burgess in 2009, known that Anoxibacillus is a fast former biofilm, and Geobacillus need a higher temperature, close to 60 or 65 degrees Celsius to form biofilm. So the second step, here we have biofilm formers, so Anoxibacillus and Geobacillus. So we would like to know if these biofilm are resistant against cleaning in place solution. So what we did, we took the 7 best biofilm formers, so the worst case, industrial case, from the first part, and we did some biofilm with the same condition, 6 hours in 55 degrees Celsius, in stainless steel microplates. So we form the biofilm, we wash the biofilm to remove planktonic cells, and then we make some cleaning in place treatments using caustic soda or nitric acid with different conditions, different parameters, with modification of time, temperature, and concentration of this solution. After a little wash, a destruction of biofilm using ultrasonic bath, and counting using the microcolony method. So here we have the parameter tested. So we have a modification of temperature when you are looking about caustic soda treatment, a modification of concentration, of duration of treatment, and the same for nitric acid. We add also 3 Geobacillus tautomophilus to compare the results obtained from Anoxibacillus flavitamus. What will be shown on the next slide will be the inactivation. So the delta between negative control and the result obtained. So here we have the result for sodium hydroxide, for Anoxibacillus, and this is the log inactivation. So if the result is low, that the effect of the cleaning in place solution is very, very low. So here on Anoxibacillus flavitamus, you have quite low effect of the increase of concentration of nitric acid, of caustic soda, of the temperature, and of the time of treatment. When you compare to Geobacillus tautomophilus, it seems that this bacteria is more sensitive to the increase of concentration of sodium hydroxide. There is an effect of temperature, but when you increase the temperature, the inactivation will decrease, and it quite have no effect on the time of treatment. When you see there the nitric acid treatment, on Anoxibacillus flavitamus, all the parameters have an effect of the inactivation. So more you increase the concentration of nitric acid, temperature, and time of treatment, you will remove more biofilms when you increase these parameters. It's quite the same on Geobacillus tautomophilus. When you increase the nitric acid concentration, the temperature, and the time of treatment, you will remove more biofilm attached to the stainless steel. So in conclusion, we have a high and fast biofilm formed by Anoxibacillus flavitamus in the condition tested, so in skim milk at 55 degrees Celsius. And it seems that we have a better resistance on Anoxibacillus biofilm, again cleaning in place solution, with low intensity, so with low nitric acid and low caustic soda. In perspective, we would like to form biofilm in flow systems with a removal of media, a removal of skim milk, to mimic industrial condition. At the same time, also to mimic industrial condition, we would like to try the resistance of multi-species biofilm against cleaning in place. So we would like to add Anoxibacillus, Geobacillus, and Bacillus lecaniformis to form biofilm maybe during 12 or 25 hours, and then test the resistance of biofilm against cleaning in place. Here are some references, and thank you for your attention. If you would like to watch more about biofilms, what we did in our lab, you can visit the poster session from Anne-Gabrielle Matteau, poster 1.47.
Are there questions in the audience? No? So I will ask one, maybe several. So can you imagine an efficient way to get rid of this spore, like high pressures, or because you were talking of the treatment of biofilm, but what about the treatment of spores? Is it possible to get rid of the spore just by a special treatment?
I think it will be difficult to treat all the worldwide production, like with high pressure. And in the industrial condition that you can't work with in sterile condition. So I'm thinking that you will always have a contamination by thermophilic spore-forming bacteria. The main objective is to remove them and to try not them to implant in the process.
And another question: do you have an idea of the parameters inducing the spore formation? Just
maybe the content of salts in the media. It's well known that what you would like to produce spore in the lab, you have to add a lot of salts like calcium, like potassium, but it's very difficult in milk because you have a lot of salts in milk, so it will be very hard to reduce the spore formation in dairy products.
And maybe I have one stupid question. It's just what about the use of enzymatic treatment to clean the devices?
Yeah, there are some. It exists, but it costs a lot. So it's not very. Yeah.
But apart from the price, would it be possible to do this?
Yeah. They did this. They do this. They use enzymatic treatment in industry, but it costs a lot, and all the industry can do it.
Okay.
There is a question there.
Yes, hello, Pierre Nielsen. I would like to ask you, have you looked into other, normally when you do a SIP, you also add some other surface active components to it in addition to the sodium hydroxide, and also quite often in industry you rinse it afterwards with, for example, oxonia, like a peractic acid treatment. Have you looked into some of these things in combination? Because I think that it could be interesting to actually, I think that peracetic acid could probably be one of the ways where you can get rid of the spore formers in combination with hydrogen peroxide, for example.
Yeah, we would like to see the effect of chemical solution without any additive. So only nitric acid and caustic soda. It's well known that peracetic acid have a huge effect on spores, on the inactivation of the destruction of spores, but I think that it will be forbidden to use peracetic acid in industry, in dairy industry for the next years. So we've mainly focused on nitric acid and caustic soda. We tried also to make some multiple treatments using caustic soda plus nitric acid, and we have a better effect on the inactivation and removal of biofilm.
Okay. Thank you, Louis. And I think we have to move to the next presentation. We are really on delay.
Okay. Thank you.
So the next speaker is Christian Diaz-Munoz, PhD student at the Vrije University of Brussels in Belgium, under the supervision of Professor De Vuist. And his research is focused on the application of starter culture to enhance the flavor and quality in cocoa beans fermentations. His presentation is called Strain Level Monitoring of Functional Starter Culture During Cocoa Fermentation Process.
Thank you for the introduction. So indeed, today I will be talking about the different approaches that we followed to monitor at the strain level resolution, when possible, the starter cultures that we inoculated during cocoa fermentation processes. But before that, I need to introduce you to the fruit of the gods, which is the translation of the genus name Theobroma, from which the cocoa tree belongs to, Theobroma cacao. The cocoa beans, which are embedded in a mucilaginous pulp, as you can see in the picture, need to be fermented prior to the production of chocolate. This cocoa fermentation is indeed of paramount importance, as it contributes to the liquefaction of the pulp, the development of the typical flavor and color of chocolate, and to the reduction of bitterness and astringency due to different biochemical conversions that take place during fermentation, which are highly influenced by the microbial groups that take place during fermentation, which are mainly yeast, enterobacteria, lactic acid bacteria, and acetic acid bacteria. However, the fermentation process is still a traditional on-farm process, which means that contamination by unwanted microorganisms can occur, and therefore not a reproducible end product may happen. To overcome these difficulties, one solution is the application of starter culture mixtures. Thanks to those, we can steer the microbial dynamics, we can get a consistent quality of the end product, and also we can speed up the fermentation process and improve the flavor of the cured cocoa beans, especially by the production of volatile organic compounds. Therefore, in our lab, we already developed an efficient starter culture mixture composed by a Saccharomyces cerevisiae yeast strain, Lignosilactobacillus fermentum LAB strain, and Acetobacter pastorianus AAB strain. However, the focus of my research is in the use of non-Saccharomyces yeast species to improve the flavor profile of the cured cocoa beans. Therefore, we tried two different strains of Pichia cutrapsevi and two different strains of Hanseospora puntia. In this presentation, I will show different parts of my research, but in all of them, there will be a negative control, that is to say, a spontaneous cocoa fermentation process, a positive control, which is inoculated with the Saccharomyces cerevisiae IMDO 050523 strain that I just introduced in the previous slide, and also a number of AFSCs, adapted functional starter cultures, which will vary in the yeast strain that is inoculated as the sole yeast or in combination with the Cerevisiae strain that I introduced. All these starter culture initiated fermentations will be also inoculated with the LAV strain and the AAV that I introduced in the previous slide. We will not change that, we will only change the yeast composition. With this experimental, wait 1 second, just doing. Yeah, with this experimental setup, we went to Costa Rica to perform our experiments in a small scale in plastic vessels with the Trinitario variety, the local variety, the local hybrid variety that is in Costa Rica. This fermentation consisted of 20 to 36 kilograms of cocoa pulpy mass, and indeed the curing consisted of 4 to 5 days of fermentation, and afterwards 7 to 11 days of sun drying, depending on the weather conditions. Back in Belgium, we extracted the DNA of the whole community, the whole microbial community, and we perform an amplicon-based high-throughput sequencing strategy for the bacterial and the fungal communities. For the first one, we amplified the 16S rRNA gene. We first focused on the B4 hypervariable region of it through Illumina short-read sequencing, and afterwards we tried to sequence the full-length 16S thanks to the PacBio long-read sequencing. For the fungal communities, we targeted the ITS1 region, which depending on the microbial species will vary from 100 to 400 base pairs. All these amplicon sequences were imported into the Data2 pipeline that already introduced my colleague 2 presentations ago, and we infer the highly accurate amplicon sequence variants, or ASVs, that can putatively resolve differences as small as 1 nucleotide. by aligning these ASVs to the genome sequence of the inoculated strains, we can effectively monitor the starter culture supply. Further, to study the influence of these strains into the fermentation process, we also perform a metabolite target analysis by using different chromatography and mass spectrometry techniques to focus specific compound groups, as you can see here in the picture. Therefore, the first result that we got when we saw the main microbial community dynamics of the bacteria in the left and the yeast in the right is that the application of the starter cultures steered the fermentation process. As you can see by the NC, F1 and F2, I don't know how to use, voila, perfect. So, I forgot how to use it again. Yeah. So the spontaneous fermentation is characterized by high relative abundance of Weissella as the main LAB group and Gluconobacter and Acetobacter as the AAB group. However, all the fermentations that were inoculated with Limoncillolactobacillus fermentum were characterized by high relative abundances of Lactobacillus, and at the end of the fermentation process of Acetobacter, also highly influenced by the inoculation performed. If we look at the fungal community dynamics, we can also see a similar picture. As at the beginning of the fermentation, Hansenia spora prevailed over other microorganisms, which was then taken over by a more ethanol, acetic acid, and high temperature tolerant Saccharomyces. All the starter culture initiated fermentations were again influenced by the inoculated strain. In other words, Saccharomyces prevails in the PC, Isacchenkia in the AFSC1, and a mix of both strains inoculated in the AFSC2. But we can go more in detail to see, for example, here, the Saccharomyces composition. So if we look at the ASBs that belong to the Saccharomyces genus, we see that there are different ones according to the different fermentation process. So the ones that were inoculated with the SRBC IMDO 50523 are characterized by high relative abundance of Saccharomyces 07. While in the fermentations that were not inoculated, other ASBs occur. Furthermore, it's only Saccharomyces 07, the one that shows 100% identity to the sequence strain. So we already know that we can steer the fermentation process, but can we influence the metabolite composition of the fermentation? To do that, we perform a principal component analysis in which we gather all the metabolic data. The first thing that we saw, if you see at figure A, is that there is a clear difference between the metabolites quantified in the cocoa pulp and those quantified in the beans and drying beans. And also we can see that this differentiation goes higher as the fermentation proceeds. If we then color by the fermentation process, as you can see in the figure B, we can start to see some clusters. First in the cocoa pulp, but more importantly in the cocoa beans, as this will be then used for the chocolate production. In other words, the yeast strains that are inoculated affect the metabolite composition of the fermenting beans. As we can see, some substrates that are consumed are produced consumed to produce other metabolites, and some of which are volatile organic compounds. We will add like esters or higher alcohols or aldehydes that will add some fruity, floral, and creamy notes that are desired for the fermentation. As we can see, there is a high reproducibility between duplicates. And here I show you a visible proof of it. For good and bad curing processes. As we can see, the inoculation by some yeast led to properly fermented and high-quality beans, whereas other yeast led to under-fermentation. Now we wanted to go one step further, and to do that, we make use of long-read sequencing technologies. We used PacBio circular consensus sequence, which are highly accurate, and we also made use of an adapted Data2 pipeline. We went to Costa Rica again, we performed our experiments, in this time with 12 different fermentation processes, again, the negative control and all the other inoculated fermentations, and we first have a look at the general picture at genus level, and we see that Limoncillolactobacillus and Acetobacter are the main microbial genera present in the inoculated fermentations. But if we go a little bit more in detail, if we analyze the lactic acid bacteria community, we see that this Limosilactobacillus is actually Limosilactobacillus fermentum, which is the species to which the starter culture strain belongs to. But if we look also at the spontaneous fermentation, there are also 3 samples that they also have high relative abundances of this Limosilactobacillus fermentum. So we want to differentiate between background and inoculated strains. To do that, we again look at the ASBs. The ASBs that belong to Limosilactobacillus fermentus, there were 21, but the same 4 were found in all those starter culture-initiated fermentations, so from F3 to F12. And they were always found in the same ratio of relative abundance. Furthermore, those 4 ASBs showed 100% identity to the corresponding region of the inoculated strain, to the 5 different copies of 16S that this strain has. Therefore, we are sure that this is the inoculated strain. Another proof is that the spontaneous fermentation that before we saw that it was the same species, we can see that the ASBs and therefore the communities of Limosilactobacillus fermentum, putatively the different strains of Limosilactobacillus fermentum are different from the inoculated one. To sum up, the application of an amplicon sequence variant approach contributed to the follow-up and to accurate monitoring of the starter cultures that we applied, and we could see, thanks to this approach, that they prevail over the background microbiota. We could get at strain-level resolution by inferring the ASBs generated with long-read sequencing technologies, and we could discriminate between closely related microbial communities. Also, we could see that Saccharomyces cerevisiae and Pichia kudravtsevi showed an enhanced production of desired metabolites, in particular volatile organic compounds. And they resulted in richer aromatic profile in the cured cocoa beans of those fermentation processes compared to the spontaneous fermentation. Therefore, this multiphasic approach that we follow allowed us to correlate the metabolites that are produced during fermentation with the yeast strains that were inoculated, untangling the functional role of the yeast in the cocoa fermentation process. Finally, I would like to thank my professors, Luc de Vest and Stefan Wex, as well as all the other colleagues that contributed to this research of the Vrije Universiteit Brussels, and all the colleagues also from the Universidad Nacional de Costa Rica, and of course you for your attention.
Thank you for... Okay.
I think it works.
Now it works. Any question from the audience? There, Luca Coccolin. Is the microphone?
So thanks for the presentation. I have two questions. One is very brief. If you can share with us, if you know the actual initial load of yeasts in your raw material before the inoculation of the starter cultures. The second one, there is a strong debate, or at least there was a debate initiated in the past, then it faded away, about the actual role of bacteria in the cocoa fermentation. Yeah. Do you have any kind of insights, or did you try to inoculate only yeasts without any bacteria and see what happened?
Okay, thank you for the questions, yes. Well, to answer the first question, yes, we did selective plating, and we also monitored the counts on YPG with antibiotics to follow only yeast. And we could see that the log at the beginning, it was around log 4, log 5, and after our inoculation, it went almost to log 6, because we attempted the inoculation of 5 log 5. So we increased the counts, and that was another proof that the inoculation was successful. And regarding the second question, that I forgot. Yes. So indeed, there are some debate, and there was some debate in the past. Indeed, that if they were necessary or only yeast. So yeast are very important, and that's why we focus on different yeast strains. But we believe that we want to have a consecution of different microbial groups through the fermentation process. Probably if you only inoculate yeast, the fermentation will follow a good trend, and therefore desired LAV and desired AAV will also grow. But we want to make sure that the dynamics are the appropriate ones. For example, the inoculated strain that we use of LAV, we know that it's a citrate positive yeast, LAV, and this is very important also to the consumption of citrate to therefore produce lactic acid, ethanol, acetic acid, and other metabolites that are also important for the flavor, like diacetyl, for example. And well, when we did the correlation analysis, we could also see that Limosilactobacillus fermentum was correlated to many desired metabolites. Not because Limosilactobacillus fermentum directly produced these metabolites, but we believe that it contributed to that the fermentation went to a good end.
Any other question from the audience? Okay, I think we can
yes,
go
I
ahead.
think we could move to the next
Thank
speaker.
you, Chris.
Thank you again. So we will move to the next speaker of the session. So Alina Kirilenko. She's a brilliant PhD student at MISO under the supervision of Marjon Wells-Benick, and she will make a presentation. I wanted to point out that she is making her PhD in the framework of EMUS project, which is a European program. And she will make a talk on the fermentation of plant-based ingredients using lactic acid bacteria as an emerging alternative to dairy-based fermented products. So,
thank you, Pascal. Thank you for everyone who made it till the end. Hope you will not regret. I will try to be short. So as Pascal mentioned, I'm a PhD candidate from Wageningen University, but I'm also employed by NIDSO, and today I'm here to tell you about the potential of plant-based fermentations to produce high-quality dairy alternatives. Because this product category has become extremely popular within the last 10 years, and with this trend continues to grow, many manufacturers seek to broaden their plant-based dairy portfolios. And as such, we can find many new products appearing on the supermarket shelves as plant-based cheeses. However, not all of these products can meet consumers' expectations in terms of organoleptic profile and also nutritional value. So, in order to overcome these problems, fermentation can be a really powerful tool, as it can help us to mask or remove the typical greeny or grass-like notes which are present in most of the products of plant origin. Also, with fermentation, we can improve shelf life stability and microbial safety of our products. Because with the lowering pH, we can suppress or even inhibit the outgrowth of some spoilage and pathogenic microorganisms. So, as you can see, the fermentation is quite a cool thing, and this is what our research is about. So, in total, we have analyzed 7 different ingredients of different origins. So, there were pulses, legumes, cereals, and a selection of lactic acid bacteria strains. The fermentations were performed for 48 hours, and as the outcome, the acidification rates were measured, and also the presence or absence of the major volatiles, like favorable or maybe undesirable aromatic compounds was measured, and also with the addition of different sugars, as glucose, sucrose, and raphenols, we were able to collect the carbon source utilization profiles. For acidification and carbon source utilization, the experiments were done in the high-throughput screening system, and the pH was measured continuously for 48 hours, and the decrease in the pH corresponded to decrease in fluorescent intensity of the pH-dependent indicator. For volatiles present, we used the same concentration of the plant-based emulsions, as you can see, and their presence or absence was measured at the end of the fermentation, but also in the beginning, in the raw uninoculated ingredients. And this was done with the GCMS analysis. What takes us to the most interesting part, results. Well, obviously, lots of data was generated, and due to this time shortage, I am not able to present all of it. However, there will be quite a few interesting examples. For example, this one is where we can see how the carbon source is important for the acidification. And on this slide, you can see two different ingredients, rice flour and fava bean. And you can see the acidification curves for 11 Lactococcus cremoris strains. Well, obviously for glucose, we see quite a nice acidification, and it was needed mostly as a positive control to see if the strains are really growing. However, for sucrose and raffinose, we see quite a change. Well, it also brings us to the thought that not only the carbon source is important, but also the composition of the ingredient itself. Because as you can see in rice, more strains were able to perform better with sucrose and raffinose, while in fava bean, almost like 10 out of 11 strains were not able to acidify really well, and there was only one strain we were able to isolate, which performed good in both ingredients and demonstrated the similar behavior. And as I mentioned before, this brought us to the thought that most probably composition of the ingredient itself plays a very important role here. That's why on this slide you can see two different ingredients, oat protein and amaranth flour, no sugar added in both. Well, because we don't need it, we know like with glucose, acidification will happen, with sucrose and raffinose most probably it will, but not that well. But when no sugar is present, we can really assess the influence of the food matrix itself. And here we can see three different species, Lactibacillus plantarum, Lactococcus lactis, and Lactococcus cremoris strains. And for example, for oat, you can see acidification, first of all, it's not that fast. The second, the pH doesn't go that low. And for amaranth, the trend is the same. Like for all of the strains, they were able to acidify quite well, and the pH dropped as low as 4.2, 4.5, which is incredible. I remind you, no sugar added, no carbon source extra added. And the last interesting example I would like to show you, the last one for acidification, obviously. This is the difference between the species which we were able to capture. So overall, we can acknowledge that Lactibacillus plantarum strains were doing much more faster and better than Lactococcus cremoris strains. So this, on the slide you can see for graphs, all are in the same ingredient. This is the rice flour. The first two graphs, the first top two graphs are rice flour supplemented with raffinose, and the last two graphs are the rice flour with no sugar added. And this is really a great example of demonstrating how plantarum strains are able to utilize oligosaccharides like raffinose, which obviously indicates that there is some enzymatic activity, and they are able to cleave these glycosidic bonds between fructose, sucrose, and so on. So, well, that was a very interesting observation, which is definitely worth to take into account if you design your microbial consortia for the fermentation. And as I will move further, another part of the research was dedicated to measuring the aromatic compounds present at the end of the fermentation. But it doesn't really make sense to measure only at the end because we are kind of curious what is in the beginning, right? So what is the initial levels of desirable or maybe undesirable compounds present? So on this heat map, you can see key, let's say, off flavors like straight chain aldehydes, the first 5, pentanal, octanal, nonanal, hexanal, butanal, and also more desirable compounds, more desirable for the cheeses, obviously. So such as 1-octane-triol, which gives the product nice mushroom smell, and also the 2,3-butanedione, also called as diacetyl. So all these compounds were measured in raw, uninoculated ingredients before the fermentations to evaluate the initial levels. And again, interesting observation was made that some ingredients initially have quite low levels, for example, hexanal. As such, amaranth and fava bean were among like all of 7 ingredients, the ones which had the lowest hexanal levels present initially. And at the same time, the quinoa, letter F, you can see that it had quite high levels of 3 strain chain aldehydes, which give it like green and grass-like of flavors. And if I move further, after the inoculation with the strains and 48 hours fermentation, we measured what was there. And there are 2 heat maps on this slide, one dedicated to Lactobacillus plantarum strains and another one for Lactococcus strains. And here I would like to draw attention to the first 5 compounds, because this is where we can see the potential of the fermentation itself. We can see the first column is again the raw ingredient uninoculated, which gives us an indication of the initial levels of the aromatic volatiles. But all other from 1 till 11 and from 34 till 44 on the second heat map, these are numbers of the strains. So with the color changing from green till red, we can see that the levels of these off flavors, they decrease. What is, well, pretty amazing for me, because it means that fermentation really helps us to reduce these not nice aromas. And this is the second example I would like to show you. And this slide will help us to see the difference between the species, like Plantarum strains and Lactococcus lactis strains. Again, all is the same heat maps. First column is dedicated to levels of compounds in raw ingredient, and all other numbers are the strain numbers. So first of all, we can see that Plantarum strains were better in reducing the hexanal levels, they're better in reducing off-flavor levels. However, if we take a look at compounds like diacetyl, or like it's 2,3-butanedione, the same, which are highly desirable in cheese, which gives it like buttery and Scotch flavor, like, yeah, maybe a golden standard for the cheeses, we can see that Lactococcus lactis strains were much better in producing this compound than Plantarum strains. And, well, it takes us to the conclusion that maybe, again, if you're designing the microbial consortium for your fermentation, you need to consider including both of the species in your starter culture. And again, maybe a few more conclusions, but they were, yeah, I did them throughout the presentation just to sum it up. So first of all, you saw it yourself, and we saw it that fermentation with lactic acid bacteria can be a very, very powerful tool to improve the quality of plant-based dairy alternatives. And maybe not only dairy, also meat. Then, again, Lactobacillus plantarum strains, they were much better in utilizing the oligosaccharides. They were much more better in acidifying medium. faster and the pH drop lower than Lactococcus strains. However, for Lactococcus strains, we noticed that they produced more aromatic compounds like diacetyl at the highest levels. And we also found the relationships between the ingredients and the amount of the compound produced, because in some ingredients, the amount of aromatic compounds were always higher, and in some ingredients, the amount of aromatic compounds were always lower. And, well, obviously, it also should be important to take into account the composition of your ingredient, because as we saw, some ingredients support the good growth of lactic acid bacteria without any supplementation, without any sugars additions, and some ingredients do not. And I would like to thank you for your attention. I would like to thank all of my colleagues who are involved in this project. And yeah, these are some QR codes which can help you to find me, NITSO, and the IMUSE project, which my research is actually a part of this ITN Marie Curie project. Thank you for your attention.
So I'm afraid we've exploded the time limit of this session, but maybe one question to the audience, because it's hard to finish without any question. One question? Yeah.
Thank you very much for a good presentation. I might represent a little bit more the users. I think that a project like yours is made maybe to reach a consortium that could be used in practice. The plant-based products are very interesting. They might have an effect that you have not been covering because it's not mainly microbial, but some people, myself included, do sense a bit of stomach issues when you eat products like this, which is probably due to complicated carbohydrates and other compounds. Some microorganisms could also do a thing on this. And when you would make a final nice product, it could be a good thing to include that. I know in a PhD you would not have that many different aspects to cover, but thinking about when you would reach a final very good product, that is an aspect one could take into consideration.
Thank you very much. I'm not sure if it was a question or...
It was not a question. Well, it could be a question. Did you also cover some of the long chain carbohydrates that could give stomach upsets and be difficult to degrade for our own systems?
Oh, not yet, but obviously this can be a good plan for the future. Thank you very much.
So thank you again, everybody, for the presentation, and sorry for the time limit, which has been.
But it means that there were a lot of interactions in this session.
So the next session will restart right now, I think. Yeah. Sorry for this.
Yes, thank you very much. Welcome to session A2. We have a number of very interesting talks to listen to. I would like to invite the first speaker, Dr. Krag, from the Technical University of Denmark, who is going to talk about Listeria monocytogenes strains and their ability to adapt and resist biocides, and how this resistance is affected by the presence of soils and other parameters. The floor is yours. Thank you.
Yes, thank you, and thank you to the scientific committee for giving me this opportunity to present some of our work. So, as you may know, Listeria may survive and persist in food processing environments, and there are several factors for this. One of them being that improper cleaning or the following disinfection might be insufficient. And some of the reasons why this can be insufficient is that, Listeria may hide in badly designed equipment or surfaces and materials that have worn out or cracked, which again makes the cleaning more hard, but also makes the biocides less likely to hit the cells. Thirdly, Listeria may form biofilms together with other bacteria, and also Listeria do have some biocide resistance that, together with other bacteria's resistance, may make them able to survive disinfection treatments. So for this specific part of our project, we set up 3 research questions. One being, does biocide resistance vary among Listeria isolates? And how do biofilms and soils or other organic matter affect the biocide susceptibility of Listeria? And lastly, does Listeria develop any resistance to commonly used biocides? So first, we needed a strain collection with sufficient phylogenetic diversity to make us able to come up with some more generalized conclusions in the end. So we ended up with a collection of 240 isolates, mostly from different foods and also from the food processing environment. And using whole genome sequencing, we were able to identify 137 different CG-MLST types, and also this included 53 different sequence types. So in all, we concluded that we had a sufficient diversity to come up with some more generalized conclusions in the end. So the initial phenotypic testing was the classical MIG testing, which many of you also know for antibiotics especially. So we did twofold dilutions, meaning that of course we have a large test range, and we chose to do this in what we call more realistic conditions. So we incubated at 15 degrees and also used a 10 times diluted media to again more mimic the food processing environment. So if we look at the first biocide, prussic acid, a bit disappointing, but also nice to see all the isolates had the same MIC value. For sodium hypochlorite, we saw that a few of the strains, or 30 of the strains, were a bit more sensitive, but all in all, very much the same MIC value for all the isolates. The same picture continues for ethanol, not much diversity here. But however, when we look at benz and conchloride, where we know there are resistant differences for Listeria, we saw a clear division of a sensitive group and a more resistant group. So we see a 4 to 8-fold diversity between a sensitive and a more resistant group. And this correlates with the resistant genes found in these 88 of the resistant isolates. So just one of these, so to say, resistant isolates did not have any of the known resistant genes. And this was a specific sequence type 328. And of course, more work has to be done to actually conclude what could be the mechanism for this specific strain being resistant then when it doesn't have one of the known resistant genes. So with this on place, in the big MIG testing of all the isolates, we decided to focus on a smaller section of subselection of isolates to make us able to do a bit more work and still have sufficient diversity. So we decided to use just 19 strains, including strains from the 4 major serogroups, and of course then covering both lineages. And we also made sure to have both isolates that had been seen to be sensitive to benzene chloride in our first test, and also some of those that were resistant, and also making sure that all the resistant genes were included as part of these isolates. And lastly, also that these 19 strains, as you can see in the tree with the small red dots, were spread throughout the tree to have as much diversity included as possible when selecting fewer strains. So the next test was more like a disinfection test as you would see it in the industry. So we did a shorter exposure of 30 seconds to each biocide, and first here starting with the benzene chloride at 50 parts per million. So it is lower than the industrial concentration, and that is because simply if we use the concentration used in industry, they all died. So at least that's a good result, but we wanted to look for diversity among the isolates. So we had to lower the concentration given. And here you see that there's no big difference between the isolates. However, there is one interesting observation, and that is that those strains with red dots that had some of these resistant genes that gave them resistance at the low concentration in the MIC test, these do not have any survival advantages at this high concentration. So both groups of sensitive and resistant have the exact same log reduction on average. And this also correlates with the minimal bactericidal concentration that we also tested for the strains, and it was also the same when tested at 48 hours. Then we went on to do the same test for sodium hypochlorite, and again we saw no diversity among the isolates, and again this correlated with the MPC also being the same for all these 19 isolates. And going back and looking at the tree again, and now with the MIC values for those isolates that were, so to say, more sensitive, at least in the MIC testing for sodium hypochlorite, as you can see by the green color bars in the tree, in the circle, they are all at the, you can say they are all in different areas of the tree, so there doesn't seem to be any specific adaptation or sensitivity among one group of isolates. And lastly, if we look at the peracic acid, as you remember, maybe in the MIC testing, they all had the same value, all 240 strains. But with the shorter exposure time and the high concentration, we actually see that there's a difference between the two lineages. So lineage 1 seems to be way more tolerant, whereas lineage 2 is definitely more sensitive. And yeah, this was in contrast to the MIC and MPC observations. And lastly, if we go and look a bit more at the biocide tolerance of Listeria under different conditions. So to make it simple, I have normalized the MIC values to just be represented by a 1x. And if we compare this with the MIC when we tested under, so to say, soil condition, so that is the presence of any organic matter that might be left after cleaning or insufficient cleaning, you see that for peracic acid and ethanol, the MIC is the same, so the biocide is just as efficient. Whereas for sodium hypochlorite and benzene chloride, we need an 8-time higher concentration to achieve the same effect. So really underlines that cleaning is very important for an efficient biocide treatment. Just to say the MPC again, we saw again peracic acid just as efficient, killing the cells as inhibiting the cells, whereas for the other biocides we need a high concentration. And when we allowed Listeria to form a biofilm in microtiter plates, we saw again peracic acid being the most efficient, that the same concentration as in the MIC test was efficient, whereas for the other biocide, and especially benzene chloride, we needed a way higher concentration to achieve killing of the cells. And then what about adaptation to biocide? Because now we have just been looking at what is present in our strain collection. So we decided to do an 18 days adaptation to biocides. And what we saw this was for prussic acid and sodium hypochlorite, we were not able to increase any resistance further. Whereas for benzalkonium, we saw that those strains that were originally sensitive, we were able to increase their MIC value 8-fold, whereas the already resistant strains could not be further improved. And if we recall that these, so to say, resistant strains in the MIC test to benzalkonium, they did not have any advances at higher concentrations. And yeah, this also correlates with the findings in the initial MIC test, where we didn't see any diversity towards prussic acid and sodium hypochlorite, whereas for benzalkonium, clearly there was a diversity. So to sum up on the research questions, I mean, we do see some variations in resistance in the MIC testing to benzalkonium, which, however, did not have any advances at high concentrations. We did see some variation in prussic acid at higher concentrations, but we could only identify a difference between the lineages and not have any isolates that seemed to be very resistant compared to others. And we saw that non-clean conditions may increase the biocide tolerance of Listeria more than actually resistant genes does. So this kind of shows that we should focus, or we should at least not forget to focus some of our work on the cleaning aspects, because insufficient cleaning might be just, if not more important, than actually keeping control of resistance development towards biocides, for Listeria at least. And with this, I'll just say thank you for listening, and if you have any questions.
Well, thank you very much for this very interesting talk. Any questions from the audience? No? I have one question for you. Have you thought any mechanistic perspective of the effect you see that the soil decreases the efficiency of biocides? Is it that biocides are blocked by soil, or it could be that soil induces some state of physiological state to Listeria and it is become more resistant?
Yeah, so it's at least to my knowledge known that, especially for sodium hypochlorite, that it reacts with any organic matter, and it happens quite instantly. So if you have any organic matter being media or some leftovers from a fish processing slicer or whatever, it will inhibit or it will decrease the efficiency, especially for sodium hypochlorite. So I think we can generalize this to all, I mean, settings, not just Listeria, at least for sodium hypochlorite. Yeah.
Okay. And are you planning to look into the transcriptome of the strains that are already highly resistant to the biocides you showed? Because you said they reach a plateau and they cannot adapt any further. So.
Yeah. I think what would be, and what other people have been doing in other stresses, is to actually see if you can isolate survivors from higher concentrations treatments, because at least other people have tried similar things using this, I didn't say, but we use this sub-MIC adaptation evolution, and it seems like at least this is not a way to identify higher resistant strains. So we need to look at high dose treatment and see if we can find any surviving cells from these treatments. I think that should be the next focus if we want to find specific isolates that are higher or more resistant.
Okay, thank you very much. Yeah, there's a question, I see.
Thank you, Martin, for the wonderful presentation. Francis from the Institute of Food Safety, Zurich, University of Zurich. So I have probably two questions. One is, do you know the history of exposure to any of these biocides in your strain collection? Because this might affect the tolerance levels you detect. And then the second question, did you see any clonal complexes that showed a trend towards higher resistance compared to the other ones?
Yeah, so for the first question, we don't have the specific data, but we did discuss a lot that we would actually, as you would expect, those strains, isolates coming from the food processing environment to have an advantage because they should have been pre-exposure. But I guess the fact is that those isolates coming from food products, at some point, they have usually also been in the food processing environment. And then there's the last thing that maybe they don't have any resistance development, so that's why we cannot see any differences from the source. And to the other question, we only saw clonal complex correlations, so to say, or higher resistance by the fact that these resistant genes that I showed are especially found in the sequence type 121, or at least that's the most dominant one, and that's clonal complex 121 as well. But it comes down to whether or not they have the resistant genes or not.
Okay, thank you very, very much for the nice talk.
Now we would like to invite Dr. Emily Lang from the University of Campinas to present us the work entitled Survival of 400 Salmonella enterica strains isolated from Brazilian food industries to environmental perturbations.
Before to start, I would like to thank the organization of Food Macro, which gave me the opportunity to present my work today. This study is a part of a larger project where we are focused on milk powder industry, because milk powder is a common ingredient in numerous food products as powdered infant formula. And these products are not sterile foods, and microbial contamination can occur along the food chain production. And I think everybody remember the outbreak with Salmonella enterica in 2017, which led to the infection of 36 infants and led to 18 hospitalization. The origin of this contamination in this outbreak was the food production environment, and Salmonella enterica were found on the surface inside the factory. And this is the reason why we try to look at the behavior of Salmonella enterica facing stresses commonly found along the food chain production in the environment. To do this, we use 400 strains of Salmonella enterica, for which we know the therovar. We have 24 therovar in this study. And all the strains were isolated in factories. And for this study, we have 87 factories who will participate to furnish isolate. And we divide each factory in sampling area where we found Salmonella enterica and the isolates. The area in contact with the initial food product, the area of transformation of the product, which contains the sterilization procedure and also the spray dryer, and the area in contact with the final food product. For each strain, we decide to apply the traces, which mimic different environmental perturbation commonly found in food industry. The first one is heat stress. We perform at 62 Celsius from 0 to 1 minute. We also perform the desiccation for 84 hours at room temperature and room relative humidity. And finally, we perform an osmotic stress at 0.8 water activity for 5 minutes in milk. To observe the impact of stresses, we perform classical plating method on triptych soya agar. And we express the result as the log of the final count over the initial count. For the heat stress, we also decide to calculate the D value, which represents the time necessary to divide the population by 10 during a heat stress. All the results were talked together to be analyzed with a principal component analysis and an analysis of variance. I will start to present you the result of the PCA. The PCA gives us the expression of the result as function of new dimension, and we see that the impact of heat stress and desiccation are the same and not correlated with the result obtained with osmotic stress as a function of dimension 2. And on dimension 1, we have a positive correlation for for all the stresses. So for the analysis of further graphic, we will see on dimension 1 stress-resistant strains and sensitive strains in the other side. We also have the dot plot which represents each strains as function of the dimension 1 and dimension 2. So we see we have great dispersion of each strains on these two dimensions. And we can also color each point as function of their qualitative variable or supplementary variable. Here we have one color per sampling area, with in red sampling area corresponding to the area in contact with the initial food, in dark the transformation area, and in green final food product area. So we can also draw the confidence ellipse, and we see we have the ellipse in green, which is slightly on the right, suggesting that we have in this area strains which are more stress resistant. We can do the same for the serovar and factory, but we have a lot of different serovar and factories, so it was a little bit confusing on the graphic. So we decide to perform and present ANOVA for each stress independently. And the ANOVA reveal for each stress a significant influence of the strains, serovar, and sampling area on the stress response of Salmonella enterica, but no influence of the factory origin of the isolation. And the ANOVA reveals significant difference. We decide to perform a two-case honest significant different test to have a ranking of resistance of the strains and also of the serovar and sampling area. So for the serovar, we can have this following ranking with in the top the resistance serovar and at the bottom the sensitive serovar. We found a common serovar between heat stress and desiccation, for example, which is the serovar Seftenberg, which is already known to be a resistant serovar for heat stress, particularly. And also we found a common serovar in resistant group, which is Indicam, for which I have less information. And also, we can find common sensitive serovar for heat stress and osmotic stress. So we can think that some serovar are more resistant or more sensitive than others, depending on the stress. For the sampling area, we can also make a ranking regarding the resistance, and we found more resistant strength in final product area. And if we look the evolution of food production chain At the beginning, we have the most sensitive strain. To the end, we have the most resistant strains. So we can maybe think to an acclimatation or adaptation of the strains along the food chain, or selection of the most resistant strains along the food transformation, or when we arrive, there is cleaning procedures which are more drastic. To resume, we use 400 strains of Salmonella enterica, which come from food factory. And we observe the survival of these strains after environmental perturbation. We see an influence of their biology, so the strain and also serovar, for which we have serovar more sensitive and more resistant. And we see also an impact of the stress and also an impact of the origin of the strain inside the factory. And all these results reveal a really complex behavior of Salmonella enterica. And this study is a part of a largest project, and this first study will help us to make a strain selection to go further by biomolecular analysis of the strains to try to understand the difference we observed here, and also to observe the behavior of Salmonella enterica during the spray drying process. And I would like to thank all of the people who work with me, my lab team and my supervisors. And thank you.
Thank you, Mrs. Lang. We have time for a couple of questions. Anyone? There's a...
Congratulations for your interesting presentation. I want to ask, have you done any genotyping analysis to be sure that you have different strains? Because I suppose that some of the strains isolated from the same factory, they are probably the same strain.
We have already not done that. I hope to do that for the next step of the project.
Hi, thank you. That definitely would, the methodology, a lot of work, I imagine, with all the plating. I'm just wondering, because I work with Stephenberg myself, you say you're going to spray, is that going to be on the milk powder itself? Okay, and like, would you be doing survival studies on that?
Yes.
Or packaging? Okay, very interesting. And like, how do you, I suppose, plan to go about that, if you don't mind me asking?
We will use a pilot, and we will be able to manage different parameters as the rate of milk, the rate of air, the temperature. I think.
Any other question? I would like to ask something myself. What about the occurrence of the different serotypes? There were a lot of each serotype, because if there is only one serotype present, then maybe the analysis may be biased. Have you seen a trend?
We don't have the same occurrence for each serovar. We have serovar which are more represented, more isolated. For example, moorheads and Typhimurium are two serovar which are the two dominant, and we have more rare serovar. Infantis was isolated so 5 times, for example.
Okay. And how many industries exactly? How many industries were implicated in the study? The factories? Approximately. The factories, the industries.
We have 87 factories,
Okay.
So 400
that was
strength.
interesting. Okay. Thank you very much.
Thank you.
You may proceed.
Okay, thank you very much. Now we would like to invite Dr. Leborman. Dr. Leborman is going to talk about us about the impact of phages on the dynamics and metabolism of a model microbial community inspired from a fermented beverage. Okay? Can you hear us well?
Hello,
Everything
everybody.
all right?
It's all is right for you?
Yeah, perfect.
Okay, so I will start.
Thank you very much.
Hello everyone, I'm Pierre Dormant from the University of Caen in France. And today I'm going to present you the impact of phage on the dynamic and the metabolism of a model microbial community inspired from a fermented beverage. So first of all, bacteriophage, also called phage, are viruses specifically infecting bacteria. They are present on 10 to 31 particles on the earth, so 10 times more than bacteria. Bacteriophages are non-living organisms but biological entities because they don't have the machinery necessary for their replication. Their size is comprised between 45 and 170 nanometers with a DNA or RNA genome. Two main life cycles were known about phage. So the first one, you have the lytic cycle here on the right, leading to the death of the bacterium host after the lysis of the bacterium and the replication of the bacteriophage inside the bacterium. And you have also the temperate or lysogenic cycle, where after the infection of the bacterium host by the bacteriophage, the genome of the phage was integrated inside the bacterial chromosome as prophage, and this prophage is replicated in the same time as the bacterium. So now what about phage in food industry? Phage in food industry are mainly studied for their positive aspect, like antimicrobial agent to destroy pathogens or spoilage bacterium, for example. But they are also known for their aspect like killers or food starters against, for example, in the dairy industry, against Lactobacillus lactis or Streptococcus thermophilus, but also about Enococcus honey in the wine, for example. However, what is known about phage-host interaction in fermented foods is very poorly described. Few molecular studies are available about Lactococcus lactis in the dairy industry and also about Liquor Lactobacillus mali in cider during my PhD project. But no study take into account the impact of phage in microbial community model. While phage infection strongly deregulates infected bacteria, and particularly cell function like motility and diverse metabolism, for example, during the infection of Liquor Lactobacillus mali strain. During this infection, we have shown that the glycolysis was downregulated and the lactate dehydrogenase was highly induced. So there is a need to take into account the impact of phage in foods to understand their role in the functioning of microbial communities. So now phage shape microbial communities in the gut and particularly in the mice gut microbiota. For example, a study showed that phage reshaped the mice gut microbiota equilibria after a dysbiosis caused by an antibiotic treatment. Another study showed that phage also impact the microbial population fluctuation and also the metabolism of a model of gnotobiotic mice colonized by human commensal bacteria. So we have to know what is happening in food ecosystem. So very few data are available about phage-host interaction in food ecosystem and especially in cider. Cider is an alcoholic fermented beverage based on apple juice, and cider making generally occur from November to March. The first step of cider making begins by the apple collection, and after apples were washed, ground and pressed to obtain the apple must. The apple must was then put in different tanks for the fermentation. The fermentation is divided in two different steps: the alcoholic fermentation performed by yeast here, and the malolactic fermentation and the maturation performed by lactic acid bacteria from genus like Enococcus, Lactobacillus sensuato, Pediococcus, and Leuconostoc to obtain the final product cider. So the objective was to study the impact of phage after a phenomenon of dysbiosis on a model microbial community inspired from cider. So now what about the methods of the study? The first step was to design a model microbial community inspired from cider, and 3 lactic acid bacteria were selected: Leuconostoc mesenterides, Licorylactobacillus mali, and Enococcus honi, and 1 yeast Saccharomyces uvarum. These different microorganisms were inoculated in MRS broths, and when the population reached around 10 to 5 CFU per ml, this inoculum was divided in 3 conditions: a control condition and 2 conditions subjected to a disturbance to establish a dysbiosis. The objective of creating a dysbiosis was to disrupt microbial equilibria without totally eliminating a microbial population. So different disruption factors were tested, like different concentration of sodium sulfate, ethanol, and a combination of sodium sulfate and ethanol. Different temperatures were also performed for each shock, and at the final, the disruption factor chosen was heat shock at 50 degrees for 10 minutes. After, 2 bacteriophages targeting Leuconostoc mesenterides and Licorylactobacillus mali were added only on the bottle P, and these 3 conditions were monitored over time at different temperatures, at 25 degrees and 15 degrees. High-performance liquid chromatography and gas chromatography mass spectrometry were performed to analyze the metabolism of the model, and also microbial counts were performed to analyze the microbial population fluctuation during this experiment. So now what about the result of the study? Here the result of the microbial dynamics at 25 degrees are presented. So if we looked at the control condition, we globally can say that until 72 hours, the different microorganisms grow at the same way, to reach 10 to 6 to 10 to 9 CFU per ml. After 72 hours, there was a decrease about Leuconostoc mesenterides in blue and Saccharomyces uvarum here in green, to have a codomination of at 168 hours of Licorylactobacillus mali and Enococcus honi here. So after, about the condition without phage and with phage, we first verify if dysbiosis will occur. So 24 hours after the heat shock at 50 degrees, we can observe a decrease in the different concentration of the microorganism. So a dysbiosis will occur. After, a significant trend showing that the condition with phage was very close to the control condition is demonstrated, because if we looked at 168 hours, for example, we globally have the same equilibrium of the community, with the codomination of Licorylactobacillus mali and Enococcus honi in comparison to these 2 other strains. Now, if we look at the condition without phage here, after 72 hours, we have a codomination of Leuconostoc mesenterides in blue and Licorylactobacillus mali in orange here, in comparison to these 2 other microbial strains. So this result is very different than in the control condition. So here, the presence of phage seems to contribute to reshape the microbial equilibria of the model community after the dysbiosis. We also looked at the metabolic profile of the model microbial community. So here, the result of the experiment at 25 degrees and 168 hours are presented. So according to the statistical criteria mentioned here, 32 compounds were followed over time by HPLC and GCMS to observe the sugar consumption, the organic acid production and consumption, and also the volatile organic compound production. So principal component analysis were performed, and we can see on this PCA that then principally on the dimension 1, we have 2 different groups. So here on the left of the graph, you have the condition without phage, and on the right, you have the control condition and the condition with phage here. For example, the control condition and the condition with phage were characterized by high concentration of isoamyl acetate, octanoic acid, or isoamyl alcohol, for example. But while the condition without phage here was characterized by high concentration of lactate, ethyl lactate, or glucose, for example. So we also demonstrate here that the phage can have an impact on the metabolism of the community. So, like I said just before, we also performed the experiment at 15 degrees. So in summary, at 25 degrees, we have shown that the presence of phage seems to participate to microbial equilibrium of the community and also impact the metabolism with a control condition and a condition with phage very close together, while a condition without phage far away from these two others. So what about the result at 15 degrees? These results are totally different because the presence of phage at 15 degrees seems to enhance microbial imbalance of the community with a total destruction of Leuconostoc mesenterides strain. These results are also visible on the metabolism because the condition with phage here was far away from the condition from the control condition and the condition without phage here. For example, the condition with phage was characterized by high concentration of citrate, non-anal, or fructose, for example. So, in conclusion of the study, the initial objective was to observe the phage addition after a phenomenon of dysbiosis on a model microbial community inspired from a fermented beverage in here cider. We have shown that there was a temperature-dependent effect with a more positive effect at 25 degrees and a more negative effect at 15 degrees. We also show that phage addition impacts the microbial dynamics of the model and also the metabolic signature of the model community. So in perspective of this work, a further characterization of this type of model, for example, using methods like metabolomics, could be a way to have better pictures of what is happening in these different conditions. After, it could be also interesting to apply this type of study to more complex models and to other food matrices more rich in phage, for example, I think to cheese. And the global message of this work is that it is very important to consider the impact of phage in fermented food matrices because also impact the microbial dynamics and the metabolic signature of the different model with a possible impact on organoleptic properties of different foods. So now I would like to thank all the people who have worked around this work, and I thank you for your attention.
Well, thank you very much for this very interesting talk. Unfortunately, we're running a little bit late, so there is time for one question.
Yes.
Is there someone who wants to ask something?
Over there.
Yeah.
Yes, thank you very much for the presentation. I just wanted to ask if you measured the multiplicity of infection and what was the multiplicity of infection when you spiked with phage?
Yes, we have put different multiplicity of infection because in cider ecosystem there was not lots of bacteriophage, so we chosen very low concentration for mimicking what is happening inside the community.
So it was a low. So it was a low multiplicity of infection.
Yes, it's a very low multiplicity of infection because, like I said just before, in cider there was not lots of bacteriolytic phage, so the few lytic phage were mimicking with very low concentration of phage.
Okay, thank you.
Okay, thank you very much.
Thank you.
We would like to invite Dr. Liu from the Wageningen University to present the impact of phases on the dynamics and metabolism of a model microbial community inspired from a fermented beverage. Welcome,
Yes.
Liu.
Hi.
Hi.
Thank you for the kind introduction and welcome to my talk of the day. And I'm really happy to follow up on the topic from the previous speaker. Only here I talk about the bacteriophage and host interaction with insights of prophages in Lactococcus lactis strains from a complex dairy starter culture. All right, when we talk about bacteriophage-host interaction, it's unavoidable that we talk about this classic model, as also shown with the previous speaker. I will be brief here. So with at least 10 phages, they have two distinct life cycles, namely latex cycle, lysogenic cycle. And in the latex cycle, the phage replicates, assemble into progenies and lyse the host. And to zoom in this lysis, it's good to know that this lysis is a combined action of phage-encoded choline and endolysing system. Both are enzymes. So choline makes holes on the bacterial cell membrane, allowing the endolysing to access and degrade the cell wall of the bacteria, and working together, they can basically lysis the host and cause the phage progenies to release. And then, alternatively, we have this lysogenic cycle when after infection, phage DNA is integrated into the bacterial chromosome, where they are referred to as the prophage. The prophage just replicate along the bacteria, seem to do not so much, until there are what we call the induction condition. There are triggering factors to activate the prophage to enter the latex cycle. I don't know how it is for you, but when I learned this model from textbook, it seems to be quite a black and white image in my mind. So with latex cycle, it's all active situation. You have killing of cells, lysis of cells. But then when you go for the lysogenic cycle, it's all latent, nothing much happens, no killing, no cell lysis whatsoever. So, but hopefully at the end of my talk, your idea of this picture can also change a little bit, like it did for me. So our study focused on prophages in Lactococcus lactis strains from a complex dairy starter culture. This starter culture is to make artisanal Dutch cheese. And it's a result of backslopping. So many generations of propagation, it is a long-term evolution. So it's nice to keep in mind that what we're looking at is probably a success, a result of a long-term evolution. And this starter culture is really featured for its stability and robustness. When we looked into the strains, the Lactococcus lactis strains, we found that they're mainly lysogenic strains carrying prophage in the genome. And also the prophages, they all carry phage defense systems, and they appear to be functional. As we see, if you compare the wild type with the phage-cured strains that we created in the lab, the wild type have higher sensitivity, resistance towards phage infection. And then when we look at those prophages in the genome, we found that actually all the elements are there. It's complete, everything is there. But we find in almost all cases, there are disruptions in the phage tail encoding genes. It's a disruption by mobile elements like transposons, etc. And I show you two examples here. The red elements are the mobile elements to disrupt the tail encoding genes. And in the end, when we look at the phage particles, indeed, we don't see any tails. It's when we look at the microscope, electron microscope, we see all the tailless phage heads looking at us almost like a caviars. So the excitement not only stop here. When we look further, we also discovered that the phage particles seem to be released spontaneously at a quite high amount, even without any prophage-inducing elements. But of course, if you add prophage-inducing elements such as the antibiotic mitomycin C, you see a further increase in release of phage particles. However, in spite of the high amount of phage particles released upon the induction, we do not observe massive cell lysis. That is shown by the growth curve here, reflected by OD. So this gray curve is when we add mitomycin C for the prophage induction. You see, in comparison to the condition without induction, there's only, let's say, a bit inhibitory of growth instead of this drop where you typically see with phage-induced cell lysis. And if we take a cell sample at the very end of this induction process, we also observe that the cells seem to be still intact, but you do see extracellular particles on the surface of the cells. From these observations, we will lead to another observation that the prophage activation influenced extracellular vesicle release in Lactococcus lactis. Let me go into this new term in more details. So extracellular vesicles, EVs for short. These are lipid bilayer enclosed lumen-containing particles. They are produced by organisms in all domains of life, and bacteria is no exception. They come in the nano-sized range, and they carry a variety of molecules, DNA, RNA, protein, you name it. And that enables the EVs to play important roles in the cellular life, exchange of material, etc. And in recent years, there is increasing evidence in gram-positive bacteria that they also produce EVs. And our work added Lactococcus lactis to this ever-growing list of EV-producing bacteria. So what we found is that it doesn't matter if you have phage-inducing conditions or not, all the time we see that there are EV-like structures, as shown here. They look like wrinkled bags a little, and released along those tailless phage head particles, as we mentioned before. But then, if you do the prophage induction by adding mitomycin C, the extracellular vesicle EV quantity is stimulated by 10-fold or more. So there seems to be really a role of the prophage in this EV release process. And we could further confirm that by some mutants we made. First, when we take this prophage cured strain, prophage removed, and then you see mitomycin C induction, no induction, the EV quantity remains constantly low. And we further zoomed in the effect on EV production to the prophage encoded choline lysine system. Remember, that was normally used to lyse the host by the phage. So with a mutant, we created a choline lysine deletion strain. All the rest phage elements remain. And then you also see without mitomycin C, the EV production remains at low quantity. So there is really a role of the phage choline lysine in the EV production. Further, if you look at how these bacteria strains and mutants grow, we just described, under the phage-inducing, non-inducing conditions, etc., you still remember this wild type. Okay, phage induction, there's no lysis, but a little inhibition. Okay, this is the phage cured strain. It doesn't care about mitomycin C because there are no prophage whatsoever. And interestingly, when you look at this choline lysine deletion strain, it seemed to have a reduced fitness in growth. They grow slower without this choline lysine system. And that's both under the phage induction or not induction conditions. I will come back to this later. And then when we take a close look at those EV particles released, we seem to find that inside of these EVs, we find phage particles enclosed inside as cargoes, as you can see. And besides the pictures, we also further confirmed that this observation with flow cytometry. We stand all the extracellular particles with different dyes, and to keep a long story short, this flow cytometry analysis allowed us to identify at least 3 different populations of particles. First, we indeed identified a population that seemed to be the EVs, vesicles, with phage particles as a cargo inside. And another population is just the phage particles, not enclosed, naked. And then we also have a population of just the extracellular vesicles. I wouldn't call them empty because they can carry other cargoes, but at least the phage particles are not inside. All right, when I combine all the previous observations, etc., we come to propose a new mode of action of the phage and the host with EVs as a player also. So what we propose is that there is a situation that the phage particles can be released in a non-lytic way, as we observed for this strain. And this non-lytic release is when the phage-encoded choline and lysine are expressed at, let's say, a moderate level and speed, that then they would only compromise the integrity of the cell envelope in a local manner, and these small wounds locally would allow the extrusion of extracellular membrane vesicles, and sometimes phage particles become cargoes of it. Although we do not exclude the classical model of cell lysis, when choline lysine are expressed in high amounts in fast speed that would just cause cell lysis, which is also a source of EV formation. In the end, I would say there seem to be quite some new interesting elements in the phage-host interaction here. I mean, if you recollect all the results, it seems that the phage has been continuously expressed. So the phage defense system is also continuously expressed to reduce the chance of infection by other phages. So we're here with this model, and we showed indeed there is phage resistance. But then when the phage are continuously expressed and formed, the cells need the opportunity to get rid of all those defective phage particles, which are cellular waste. And this choline lysine-induced EV formation allow the opportunity to dispose such. But when you do not have this route, like when we delete the choline and lysine, you see the fitness of growth indeed was reduced. We cannot get rid of the cellular waste. And further, it's interesting to further investigate whether there are mechanisms that promote EV encapsulation of phage particles in a very specific way. Sometimes it seems to be attempting to assume such. This needs further research. And also, if you look at the cell surface that we showed, there seems to be a hot spot of EV release. You see, they seem to be around the cell division site, which could be a combined action of autolysine and the prophage encoded choline and lysine. In the end, when you look at the vesicles released, they could act as decoys for phage infection. So that would again protect the host from foreign phage infection, contribute to the resistance even further. So now hopefully I could change this black and white picture of bacteriophage and host interaction a little bit, because I believe at least that this non-lytic release of phage particles mediated by extracellular vesicles would definitely add to some shades of gray to this whole picture. And that also points into the diversity and ecological significance of phage-host interaction and co-evolution. Hopefully our findings can stimulate further applications in fermentation and probiotic formulations. The described work are published in the papers showing here, and I would really thank to all my co-workers and co-authors. The described work is also part of my PhD thesis that was completed a couple of months ago. If you are interested in a copy, you're very welcome to contact me. With that, I thank you for your kind attention.
Thank you, Mrs. Liu, for the very interesting information and this nice presentation. It was very informative. We have time only for one quick question. Anyone? There.
Hi, Liu. Great talk, great work. Congratulations. I'm interested to know the resistance mechanism that you said that your phage expresses. Is it a broad host resistance, or is it just specific for the type of phage that you used?
Yes, thank you very much for your question. The phage defense is really, I would say, it's quite specific for the phages that infect Lactococcus lactis, and they turns out to be, of course, strain specific. We know that. But you see, with phage and without the prophage, there is a difference in resistance. But I'm talking about phage that target Lactococcus lactis strains.
Okay, thank you.
Thank you.
Thank you again. Dr. Liu?
Okay, now if there is no other question, we can move to the next talk. We welcome Professor Lundstad from the Institute of Marine Research in Norway. He is going to talk about Klebsiella pneumoniae in the marine setting, a challenge for seafood safety.
So, thank you for introducing me, and thanks also to the organizers for letting us do this as an oral presentation. So my talk is about Klebsiella pneumonia in the marine setting, and if there is any reason for concern or if this is without any problems of any kind. Our research is a part of the largest project. It's called CLABGAP. And it's a One Health approach where we collect samples from the clinical settings, from patients, from hospitals and so on, and from animals, wild and farmed animals, and also from the abiotic environment, including the marine setting, and try to compare to see if there is any overlap. The work is in its way, so we don't have all results now. The work is mainly done by our PhD student, Fredrik Håkonsholm, in cooperation with researchers at the Arctic University of Tromsø in Norway. I think it's correct to say that Klebsiella is a neglected possible pathogen in foods. It's well known as a pathogen. There are some publications on Klebsiella in food, and when it's found, it's usually in poultry or salads. There are fewer publications, or only a few ones, showing Klebsiella in seafood, and so far only are in seafood from cold waters. And as far as I know, it's only one publication showing Klebsiella pneumonia in seafood as a source for infections in humans. So much is to be examined in this field. So the systematics in microbiology is constantly changing. You all know this. Klebsiella is now in the Enterobacteriaceae family, together with 6 other families. They constitute the order Enterobacterales. So this is just to put it into context. And when it comes to Klebsiella pneumonia taxonomy, it used to be quite easy. Not easy, but more easy than today. Until the mid-1980s, Klebsiella pneumonia was considered to be one species. And often today, if you don't do enough typing, it could also come out as Klebsiella pneumonia if you do just analyze and isolate. But the recent taxonomic updates, they propose now 5 species, which constitute what we call the KP species complex. And it's, of course, Klebsiella pneumonia in the strict form of the meaning, the word, sensu stricto. And then there are 4 other species. They all constitute Klebsiella pneumonia species complex. So the pathogenicity of Klebsiella pneumonia is linked to several factors, like most other pathogens: adherence, the lipopolysaccharides, and the siderophores. But what is maybe special for Klebsiella pneumonia is the ability to produce very resilient capsules, protecting the microbe from our attacks. And it's typically seen that for the so-called hypervirulent strains, you will be able to have a positive string test. It's very mucoid, so you can pull a string from the plates. And these hypervirulent strains, they can also be a challenge for healthy people. So how do we see Klebsiella pneumonia as a pathogen? It gives a variety of extraintestinal infections, like pneumonia, of course, from its name, urinary tract infections, surgical wound infections, endocarditis, septicemia, and last but not least, liver abscesses. Very serious could be. And it has been documented that it accounts for around 1/3 of all gram-negative infections, and is also among the so-called escape pathogens that require special attention. This is a typical opportunistic pathogen causing infections in hospitals, especially in patients with underlying disease. And it's the most common cause of healthcare-associated infections. And then, as I mentioned earlier, we have the hypervirulent strains that also can give infection in otherwise healthy individuals. So this is reason to concern. And then it's also worth mentioning that there is quite high carriage in healthy persons. These are just some selected studies, and in Norway we found approximately 60%, in this study at least, carriage for the complex, and 10% for the sensu stricto Klebsiella pneumonia. In Australia, maybe 6 to 19%, and then it's quite, it's not easy to explain, but there is always higher amounts in samples from Southeast Asia. This is in fecal material, so up to 88% in some countries. Okay, so back to our studies. We sampled fish, seawater, sediments, and marine bivalves. And this is the locations. You can see that some of the locations are along the coast. All the bivalves were along the coast, close to the coast. Some fewer samples in open waters. And I can say at once that we did not find any Klebsiella in the samples of fish or in the samples of sediments, but we found in seawater and in bivalves. So this would of course indicate that since we don't find in open waters, this is connected to contamination from land. Okay, what about the samples? More than 500 batch samples of bivalve mollusks, 53 fish samples, water samples, and some sediment samples. And since the bivalves are the key target for us, we found this to be especially interesting because they are filter feeders. And for instance, a blue mussel could filter, let's say, 80 to 100 liters of seawater a day or during 24 hours and retain particles. So they will retain everything that's found in the sea and that's worth eating, including bacteria and also virus. And they are easy to collect, and some of them are intended for raw consumption. For instance, the scallops and the oysters, making the the challenge even larger for humans. So what about the methods? Initial homogenization and one step of multiplication in buffered peptone water, and then transferred to the Siemens citrate agar with inositol, a new incubation and identification by Malitov, and also short read sequencing. So what do we find? We find that in this study, 14% of the bivalves have Klebsiella pneumonia in the strict form, sensu stricto, and 35% of the seawater samples have the same, have this species. And for other species are found in bivalves and seawater and bivalves again. But it seems as the Klebsiella pneumonia complex is quite common, I would say. So what about antibiotic and heavy resistance, antibiotic and heavy metal resistance? We examined 87 isolates and found antibiotic-resistant genes in 6 sequence types, and they were genes encoding to sulfonamides, chloramphenicol, quinolones, cephalosporins, aminoglycosides, and the most common resistance genes were towards beta-lactams and tetracycline. And when it comes to heavy metal resistance, this was very common. 71% had genes, mainly resistance to silver, copper, and arsenic. So it's a good array of genes found. When it comes to the initial sequence typing and comparison that we have made, we find some sequence types that intermingle with isolates from other sources, for instance from the clinical setting, sequence type 17, 20, 29, and 37, and especially the sequence type 25 is very common to what we also find in the clinical setting, in hospitals and hospital outlets, in hospitals. So you see that they're very similar, and they are almost identical to isolates from Germany and Denmark. Okay, so next part of the study was to see if any of the plasmids that we could find could be transferred to other organisms. And in this case, we made filter mating. The recipient was a green fluorescent protein marked E. coli, which you can identify under UV light. And we chose 3 resistant KP isolates that we have found and described, mixed the donors and recipient 1-to-1, and let the filter be overnight, 3 hours at 37 degrees. And then afterwards we saw what we found. And so so we found 8 plasmids all in all, 6 with antibiotic resistant genes, and they carried genes for clinically relevant antibiotics. And 5 of these plasmids also carried genes encoding resistance to copper, silver, and arsenic. One of these plasmids, carrying tetracycline and resistance to the heavy metals, were transferable to E. coli. And the conjugation frequency were approximately 5 to the power of -4 transconjugates per recipient cell. So how should we conclude about this question? Is this any reason for concern, or is it not? First of all, we have to realize that Klebsiella pneumonia is an important pathogen among humans and animals, giving severe disease in some cases. We also know that there is a considerable carriage rate among healthy people, and there is a great lack of understanding in the pathogenicity of Klebsiella, especially how symptomless colonization develops through infection. We don't know much about this. So, next one, we say that Klebsiella pneumonia is found in seawater and seafood from coastal areas, and some isolates are quite similar to what we find in infections, and they have antibiotic resistance genes that can be transferred. So you could say that bivalves that are consumed raw could contribute to colonization, making seafood a possible source. But I think it's also correct to be fair to the seafood that other food items are more in danger, especially the poultry and the salads. But anyhow, we have to follow the situation to see if it changes. And if this is published now, these two publications are out now, and several others are on their way, so we will continue working on this challenge. So by these words, I thank you for listening.
Thank you very much for your talk. We have time for a very short and quick question, if there is anything from the audience. Otherwise, we will move on, and perhaps we will discuss afterwards.
Let's do.
Okay?
Thank
you very, very much.
Thank you. So, last but not least, Dr. Margini is going to present us bacteriophages, biodiversity and quality of natural whey starter cultures in Trenti Grana cheese production.
Okay, so good morning, everyone. Of course, first of all, I would like to thank the organizer to select our work for these important congresses and to present our data in these really interesting morning sections. So what I'm going to present to you are some results that concern the lactic acid bacteria and phage community in cheese-making processes. In this term, it is well known that there are several actors that are involved in the microbial consortia that is typical for the cheese-making processes. So from the farm through the dairy implant and so on. All these actors, they contain microbes, so this means that they contain also phages. The presence of phages in cheese-making processes is in most of the case considered as a negative characteristic due to the strong economic issue, for example, for the loss of entire batches of products, rather than in the reduction in the production of certain products. We can think about products that can contain just only or few probiotic strains. So there are consistent literature available for the phages and the dairy environment. And nowadays, through the next generation sequencing, is even more clear a constant interaction between phages and lactic acid bacteria in a cheese making process, in the maintenance of starter or natural starter. And recently, it has been also shown how certain phage can remain into a starter without affecting at the end the performance of the starter itself. So the presence of phage in the cheese making is, of course, a negative characteristic, but due to the coevolution process that drive the presence of these two actors is still an utopian goal, a complete phage eradication in this kind of environment or ecosystem or industries. So in our project, the aim was to study the phage and lactic acid bacteria community in the natural whey starter for the production of the Trenti Grana cheese in order to find a potential solution for the phage contamination and to protect the excellence of this cheese. So this is a PDO grana-like cheese that is produced in the Trentino province, in the north of Italy, between the Alps. Basically, the cheese-making steps are the same as for the other two famous grana cheese in Italy, or the Grana Padano and Parmigiano Reggiano. And we focus particularly on the natural whey starter that is produced starting from the whey that is collected in the vat after the cardan precipitation, and that we call, in this case, cooked not acidified whey. So a natural whey starter for the production of this kind of cheese is a culture of different lactic acid bacteria that are daily maintained in the dairy implants. And four are the main species that are present: Lactobacillus helveticus, Delbrueckii, Fermentum, and Streptococcus thermophilus. This kind of starter is really important in terms of acidification, so for the reduction of pH that allows the right card formation, the whey purge from the card, and to inhibit the growth of pathogen bacteria or spoilage bacteria in the subsequent cheese-making steps. So the reduction of the fitness of this kind of starters in terms of acidification, it is in most of the case related to the phage attack. So we have considered 6 dairy implants spread on the territory. We collect a sample during 4 period in a year of cheese making, collecting 100 samples of milk, cooked not acidified whey, and natural whey starter. And so by both culture-dependent and culture-independent approaches, we studied the microbial and phage community, especially in the natural whey starter. So we were able to isolate 160 phage. Most of them were able to form lysis plaque on the media, and we built a lactic acid bacteria biobank. So some data from this work. During the sampling period, we collected also data that concern the technological parameter for the production of this kind of starters. Here on the left, you can see the typical trend of temperature of a fermenter where the natural whey starter is produced. The blue line represents the external temperature, the orange line is the temperature inside the fermenter. And we have 2 typical stages: a thermophilic stage where the temperature drops from 50 to 40 degrees, more or less, and then we have a mesophilic stage where the temperature drops from 40 to the room temperature. Of course, the length of these 2 stages are something that can drive the kind of microbiota that is present inside the starter, as I'm going to show you. Between our 6 dairies, we found that 3 of them have a longer mesophilic stage. With respect to the microbial composition, the majority of lactic acid bacteria that we found belongs to the Lactobacillus helveticus, as is expected because it's one of the main actors in this kind of starters. But as for the first time, we found also as a codominant Lactobacillus brevis, that is a mesophilic strain. And in this case, the majority of Lactobacillus brevis were founded in the dairy implants characterized by the longer mesophilic stage in the fermenter. Of course, some data also again from the metataxonomic analysis on the lactic acid bacteria. The main difference were found in terms of abundance of strain, especially with respect to the dairy implants rather than the sampling period. The phage community, we found phages in more than half of our sample. They were all lytic for Lactobacillus helveticus, and the majority of biotype of these phages were founded in the dairy implants that were characterized by the higher Lactobacillus helveticus biotype. So from this data, we can assess the importance of the technological parameter, at least in this case in terms of temperature for the daily maintenance of the natural whey starter, that they are able to drive and shape the biodiversity inside these starters. That of course, as much as we have biodiversity, it can represent a way to counteract a potential phage infections. So now we have these two biobanks of phages on one side and Lactobacillus helveticus on the other side. In the first case, we selected 11 phages belonging to the main phage biotype. We performed some transmission electron microscopy on them, and also we sequenced the genome through nanopore technologies. For the Lactobacillus helveticus, we selected 26 strains that were able to resist to the phage attack and that do not contain prophage. And we are just doing the genome sequencing on them, and we start to perform some acidification performance tests on experimental way in the presence of different phage community. This last part, the mean of this part is, as I mentioned at the beginning, the Trentin Grana is a PDO cheese, so the production must to follow the PDO rules. That doesn't allow the introduction of a strain from a commercial starter rather than GMO microorganism. So the idea in terms of protect the production is to consider a strain rotation of lactic acid bacteria, and in this case Lactobacillus helveticus, that must to be resistant to the phage attack and really performing in terms of acidification. So these are some, let's say, shots that we got from the transmission electron microscopy on these 11 phages. So all of them belongs to the Myroviridae family, and this was also confirmed by the genome sequence data. These are really, really preliminary data. I'm talking about a few days ago. And all of them have high coverage with an available Lactobacillus phage genome. And just to conclude some data on the phages, the idea is to go deeper to study the phage community in this kind of sample through next generation sequencing approaches. And also because the majority of the data that we have are related to phages able to infect Lactobacillus helveticus, as we use Lactobacillus helveticus for their isolation. So we perform some preliminary tests just to look for phages that are able to infect the other main actor as a lactic acid bacteria in this kind of starter. These are a table that report the result just for one of the dairy implants. Of course, we have confirmation of the presence of phages for the Lactobacillus helveticus. We didn't find phages for Lactobacillus delbrueckii, and in some cases, we find just, in 2 cases, phages for Streptococcus thermophilus. So, of course, this is just underline the need to go deeper in the knowledge of this phage community. So just some conclusion about all this data. Of course, so following the aim that I mentioned at the beginning, so which can be the main way to protect the production of this kind of cheese in terms to maintain the natural way starter activity. For sure, something that is really important are the technological parameters, just to shape high biodiversity of the natural way starter itself. Of course, there is the need to understand more the different phage and lactic acid bacteria community in these starters. Our hope is to increase the knowledge in terms of history of interaction between these two actors in the starters. And in this kind of cheeses, of course, considering that they must follow specific rules of production, one way to overcome potential problem related to the phage infection could be strain rotation approach. So with the isolation of lactic acid bacteria really performing from a technological point of view and resistant to the phage attack. So between the different, of course, different questions can be addressed now, but one that I think that is quite important could be, which is at the end, the effect of this kind of phages that, as I show you, are still present in the natural way starter, for all the year of sampling, no one of these dairy implants, fortunately for them, unfortunately for our point of view, have problem in terms of acidification. So the product was good. So which is the effect of this strain in terms of flavors, texture, and at the end, the consumer's preferences. So should we start to think about this natural restart and not only as a culture of lactic acid bacteria, but a culture of lactic acid bacteria and phages? So just to conclude, I would like to thank my colleagues at the Institute, my colleagues Dr. Elena Franciosi, the head of my group, Stefan Martens, and the people at the Computational Biology Unit, and all of you for your attention.
Thank you very much, Dr. Marcini, for the nice presentation. There's time for one quick question. Anyone? Everything was well understood, I guess. Thank you very much again.
Okay, thank you.
Well, thank you very much for following this session. Now we have to move to the poster session, I think. It's not here, it's in... It's outside, anyhow. Okay? Yes. Όλο φάγει, ρε παιδί.
Ladies and gentlemen, can you take your seats, please? We need to start this afternoon's session. I'm going to introduce our speaker, keynote speaker this afternoon, Professor Jacco Aby. He works at Wageningen University, and he has a personal chair there, and his research is focused on the functional genomics and ecophysiology of food-related microorganisms, including starter and spoilage bacteria and foodborne pathogens. And you can see the title of his talk now. So welcome, Professor Aby.
Thank you, Janet, for a nice introduction. And happy to be here and to present this afternoon. And I want to thank the organizers for the invitation. So I actually fit very well in the additional subtitle of the conference, Next Generation Challenges in Food Microbiology, because I hope to present you some challenges, and also because I think there's indeed room and space and energy for new generation scientists that can actually pick up some of the current ideas that are running around. So today I will discuss diversity and substrate utilization capacity in Listeria monocytogenes, impact on transmission and violence. And while thinking about this presentation, I realized it would be very broad and decided to focus on one of the favorite topics we are dealing with at the moment in the group, and that is bacterial microcompartments. Bacterial microcompartments are actually organelles and prokaryotes in bacteria, which have gained a lot of attention in certain research areas in nanotechnology and biotechnology. There's also an interest in foodborne pathogens, especially E. coli and Salmonella. But there's a lot of studies and approaches used in bioinformatics, whole genome comparisons, and there's very little information actually about physiology. So I was very much intrigued by these protein organelles. And so there's organelles like in eukaryotic organisms, but these organelles are not enclosed by membranes, but by proteins. So this is really unique, and I hope to share my enthusiasm and inspire you for further analysis and thinking about it, probably maybe future research in this area. So transmission in the food chain is supported by substrate utilization, both in aerobic and anaerobic conditions. And in this presentation, I want to zoom in on anaerobic conditions, because that's where the microcompartments, these special organelles, come into view. And they actually facilitate metabolism of non-fermentable substrates. They actually allow accumulation and condensation of certain enzymes into the compartments, so it makes the metabolism more efficient. And as I will show you later on, there's another reason, and that's because there's volatile intermediates that are toxic. So there's several reasons why the microcompartments are essential for supporting metabolism of certain compounds and anaerobic conditions. And I will show that these systems are actually activated in Listeria monocytogenes under certain conditions, despite the multiple level control. And I will also give some examples about the growth performance in anaerobic conditions, and actually link this to conditions that they may encounter in modified atmosphere or vacuum-packed chilled stored foods, like cold smoked salmon. So we see a happy guy here because he defended his thesis in December. He actually did his PhD on these microcompartments, addressing several topics that are interesting. And he actually managed to submit 7 papers. So 5 of them you can find in online journals, and 2 of them are in bioarchives. And of course, if you're interested after this presentation, you might contact me for a PDF of the thesis or a hard copy. So this is Andy, and he did a great job. And we actually picked up, starting with a cartoon of Gerald Kervel's lab in Berkeley, that shows these compartments in a bacterial cell. And as I mentioned, they contain metabolic enzymes, enclosed by a protein shell that actually allow metabolism of compounds that actually have volatile toxic intermediates. So in this way, they are separated from the cytosol, and bacteria can actually consume and metabolize these substrates and actually produce energy that allows them to grow under these stressful conditions. And the basic composition is given in the middle. So there we have this enclosed microcompartment. There's a wide variety of proteins involved in the shell. That's also why I has drawn the interest of biotechnologists and nanotechnologists, because they actually like to play around with proteins and then create novel nanoreactors. So what happens is that the substrate can be taken up via specific diffusion proteins. Inside, the substrate is converted by a couple of enzymes. The volatile intermediate is retained during the, let's say, the existence of the compound and before it is transferred to the next compound. And then at the end, the intermediate, because there's further metabolism in the cytosol, actually will proceed. And in the cytosol, the ATP will be generated. So this shows one of the papers of the Kerfeld group, showing the diversity in bacterial microcompartment clusters. And what you actually see is that the largest cluster that has diversity in these compartments is actually the firmicutes. Well, Listeria is in this group. And the representatives are predicted to include over 20 distinct type of microcompartments. That doesn't mean that it's 20 different substrates, but it points to a wide diversity and the type of clusters and the type of enzymes and the type of shell proteins that are involved in the different species. And as mentioned in several talks this morning, also in the keynote by Luca, and so you can actually look at the diversity and try to translate it to functionality. But the biggest part of these microbes have not been cultured. So there's still an enormous reservoir of information we have, and that has to be applied to what are they actually doing and what is their role. So in the analysis, it turns out that Listeria contains membrane compartment dependent utilization systems for ethanolamine and propanediol. And these clusters are mainly present in human pathogens, among other Salmonella, and only very limited and beneficial bacteria. Up to recently, the only studied bacteria that actually produces microcompartments is Lactobacillus reuteri. And we recently provided evidence and also have shown that Propionibacterium freudenreichi, a well-known probiotic and also starter bacteria, also produces these microcompartments and can actually metabolize propanediol. So this is the overall design. On the left, I gave the structures of ethanolamine and propanediol just to visualize it. They're very simple compounds, C2 and C3 carbon. And normally they cannot be metabolized without oxygen. So you need these special features and combinations of characteristics of the cell to be able to metabolize these compounds and generate energy. So if you look to the right, we can actually see the catabolic so-called metabolism, because they actually metabolize substrates. So the substrate, it could be ethanolamine or propanediol, is going in. There's a signature enzyme that actually produces the aldehyde, and then the aldehyde is rapidly converted into alcohol by alcohol dehydrogenase. And to maintain redox balance, another substrate that is converted to an aldehyde is actually going the other way. That's via aldehyde dehydrogenase and a phosphotransacylase. And this produces SL phosphate on the bottom of the carboxysome microcompartment. And then the phosphorylated compound moves out, and this is a high energy phosphate bound. And actually ATP can be generated by SL kinase. So what happens is that you have a unique situation where you have non-fermentable substrates. They can actually be metabolized in this very efficient way by combining and concentrating all enzymes. And actually the energized compound, the S-cell phosphate, moves into the cytoplasm and is actually converted to an acid, and ATP is formed. And at the basis you see the simple, let's say, construction of the gene cluster. But you have to realize that there are multiple shell proteins and there's multiple components that make up the enzyme. So these are also 3 or 2 or 4 component enzymes. So that brings me to Next slide. So these compounds, ethanolamine, and also glycerol, as you'll know in a moment, they are present in membranes, and all living forms have membranes. And both bacteria, plants, animals have large amounts of phosphatidylethanolamin in their membranes. So it's one of the main components in let's say, double-layered membranes. So it's present everywhere. It's present in food. It's actually present in the GI tract because we have a shed over of the epithelial layer, and then also lipids are released. And these lipids, actually indicated in the center, can be attacked by a number of enzymes, diacetyl esterases or phospholipases, phosphatases, and they can actually cut off the acyl chains, release glycerol, and that can also act as a substrate and release ethanolamine. And so that's actually indicated on top. Yeah? Glycerol can be converted to propanediol. So there we have the two substrates that can be actually utilized by Listeria putatively and in a pathway via the microcompartment. Propanediol is actually also produced from rhamnose metabolism. And rhamnose is one of the main carbohydrates, one of the main hexamers, and plant cell walls. So there's several levels where propanediol and ethanolamine can be produced or released in foods or in our intestine. So if you then look at the current status of the research, on top I indicated there's several food products and also where you can actually encounter these compounds. So ethanolamine, more in lipid-rich foods, propanediol also because of glycerol conversions. Dietary fiber, and that contains large amounts of rhamnose, so rhamnose metabolism could supply propanediol. And 1,2-propanediol is also a food additive, so it's also added to food. As far as I know, it's a sort of a stabilizer. So the current situation is that there's BMC-dependent propanediol utilization and ethanolamine utilization gene clusters identified. There's a role suggested in transmission in foods based on gene expression studies. And that was a very nice study from the group at Cornell already in 2015. So that actually showed transcriptome analysis of the adaptation of Listeria monocytogenes to growth on vacuum-packed cold smoked salmon. And what they actually showed was activation of expression of PDU utilization and also ethanolamine utilization genes. So they hypothesized that it could perhaps indicate that they were actually using, let's say, these compounds as a growth substrate. But this was just hypothesized based on gene expression. And together with what I mentioned about the microcompartment studies and biophysics and also nanotechnology, this paper really triggered me to think about a project where we could actually analyze and confirm the role of microcompartments in Listeria. There was another indication that they could play a role in violence that was described in a study by Chart et al. They actually constructed a mutant in the PTU cluster. And in mouse model studies, they actually could show that the wild type had a longer, let's say, perseverance time in the mouse GI tract. So actually, with inactivation of one of the enzymes in the pathway, the transition time was actually shortened. So it actually was shed earlier. And there were also some indications that maybe intracellular growth was affected, but that was not very clear. So these were some indications, but no proof of the pathway. So that brought us to three, let's say, approaches. First, we wanted to analyze the distribution of these type of clusters in Listeria species, because now there's more, let's say, Listeria species known. And it was interesting to see whether there would be any additional, let's say, distribution there. We also wanted to investigate the role of vitamin B12 or cobalamin. This is a cofactor. It has been suggested that some of the utilization pathways via microcompartments are B12 dependent, whereas others are not. And there were indications that the Salmonella B12 cluster was involved in this conversion. So we want to see whether this was the case also in Listeria. And we combined growth analysis, metabolomics, proteomics, transmission electron microscopy to investigate whether indeed growth was supported by activation and reproduction of microcompartments, and also, of course, we check metabolomics. And finally, we actually decided to investigate a possible role in violence using CACA2 model system, which is, of course, just one model system, one model in vitro system that you can use. But what we actually wanted to see is how do the cells behave if they are non-induced and if they are induced for the system. So we really knew that we have control cells that were not induced, and we had control cells that had produced these compartments and could metabolize these lipid derivatives. So that was actually the final part of the study. And for today, we'll, of course, finalize with the discussion and a short outlook. So this is the B12 cobalamin cluster in Listeria. There's a very nice recent paper in Frontiers in Microbiology by Anasta et al., so I advise you to read it if you're interested. And so this is the propanediol cluster, the ethanolamine cluster, and the vitamin B12 cluster. And why is this apparent, and why is this to a certain extent logic? Because the key enzyme, the signature enzyme involved in these pathways is actually B12. So it makes sense that if you activate these utilization clusters and want to actually activate and use a substrate, you also need a B12 cofactor, otherwise the signature enzyme doesn't work. So the assumption is that, okay, and the whole system is activated depending on the substrate, and so there's activation of PDU if you have propanediol, there's activation of ethanolamine if you have ethanolamine, but anyway, and you need activation of the B12 cluster. So that's a very interesting concept, because if you look at the vitamin B12, it's a very, very complex molecule. So it also indicated if you look at the genes, and this is not all the genes that are actually involved, you have to tune up a significant enzyme machinery to produce this compound. And on the other hand, if you look at all the other genes, you have to tune up a very significant machinery to produce all the shell proteins and compartments and also the enzymes. So, and I won't go into detail, but there's at least 8 regulatory levels of regulation. And so there's the sigma B, there's other sigma factors, there's at least 4 small RNAs, there's a 2-component system, there's a B12 riboswitch, and this indicating that the cell is really acting to have a fine-tuned control whether to activate or not the whole system or only parts of it. So that's why we took up actually another topic, and that's what I relate to this, that we wanted to see whether addition of B12 is actually needed to activate the system and support growth. And this was inspired by a paper in 2018 from University Groningen, where they actually characterized a B12 transporter from Lactobacillus delbrueckii. And it turned out that it had similar, probably similar transporter system in Listeria, but it was of course not confirmed at that time. So there were several options to analyze regulation of these metabolic pathways. So just very quickly, Andy looked again for distributions and including novel Listeria species, and actually we still find it's only present in Listeria sensu stricto. And so that actually indicates it's not unique for monocytogenous, but this also fits in the analysis that it might support growth of Listeria species and a wide range of environments where these compounds are probably available or produced. So the first experiments Andy did was actually looking at we first focused on the ethanolamine utilization cluster. And this shows a data set where we actually analyzed anaerobic growth in a defined medium with ethanolamine as a control and with added B12 to see what the effect was of B12 addition. And that's the blue symbols. And what you see is that if you look at the colony-forming units, there's a massive increase in the colony-forming units if B12 is present. So defined medium ethanolamine requires B12 addition and to support growth. And if you look at ethanolamine metabolism, top right, then you see that ethanolamine is only metabolized when B12 is added. And if you look at the bottom, you see that the typical expected products of ethanolamine metabolism, acetate and ethanol, are indeed produced in these conditions and not the control conditions. And what we find is a ratio of about 2:1, whereas the prediction will be 1:1 based on redox balance. But I will come back to that shortly later. So we did proteomics, and what you actually see is that if you look for the EUT-induced cells, so they are again with B12, and you find massive upregulation of almost all proteins and that are, or genes that are in the cluster, and also translated into, let's say, very high protein levels. And when we combine this with transmission electron microscopy, what you see indeed in the control cells is that, and that the several microcompartment type as in line with how they are actually found in other microbes, and you can actually see these microcompartments appearing in these conditions. You may have noted that the shape and also the size is about 40 to 80 or 100 nanomolar, mimics bacteriophages. So it actually mimics bacteriophage heads, but there's no evolution constraint between that. So it's really microcompartments and protein shells and with the enzymes contained inside. So this is our final model for the utilization of ethanolamine by Listeria and via microcompartments, and this shows some additional information. And you see this sort of a threads that actually connect certain of the proteins to the shell proteins indeed. And there's still an ongoing discussion, how is this formed, how is this synthesized in time, and do we first have a sort of a shell already and then the enzymes are put in, or do we first have aggregates of enzymes and then they are combined with the shells? There's a lot of interesting studies going on that part. And what you get is ethanolamine goes in, B12 dependent conversion into aldehyde. What you also note is that ammonium is produced because ethanolamine has an amino group. And this also may play a role, and I won't go into that any further, but it could also play a role in pH control. So because of the release of ammonium, it might affect in close connection to the cell that actually the surroundings become more alkaline. So it also might link to maybe a bit of a sort of a acid defense. So we find acetate and ethanol, but we find 2 acetate and 1 ethanol. That actually means that there are 2 ATP generated from 3 molecules of ethanolamine, whereas the prediction will be in a 1-to-1 ratio, you have 0.5 ATP per ethanolamine. So this is a 33% increase, which is of course massive. And we actually found out the mechanism, and if you're interested, you can read it in the paper, because this links to extracellular electron transfer connected with the microcompartment. So I think this is a very nice addition to the information contained to these kinds of metabolites and pathways. To connect it to the work of the Cornell group, and we actually produced filter sterilized cold smoked salmon food medium. So we have a sterile medium based on cold smoked salmon. And we actually looked for growth of Listeria monocytogenes at 7 degrees in anaerobic conditions. And we find indeed, that's the green and red lines, we find growth in this medium. But if we add ethanolamine, we see that the blue and purple line actually show massive increase in OD and also massive increase in coliform units. But here you see that there's very good growth with the addition of ethanolamine without additional supplementation of B12. And notably, B12 levels are very high in cold smoked salmon. So there's, of course, a couple of questions coming up. So is ethanolamine released in cold smoked salmon? And that's the first one. And how? And the other thing, the other point, of course, is what kind of enzymes or what kind of factors would allow Listeria to release it? And for this, actually, it brings into the topic that was discussed many times this morning, and so microbiota. And of course, you have co-microbiota in foods, and we also have co-microbiota in the GI tract. And if you look into the spoilage flora in cold smoked salmon, so vacuum-packed cold smoked salmon, and that's important, and storage at low temperature, there's a very typical spoilage flora, and that's mostly lactobacilli. And there's several lactobacilli that actually are lipolytic. And so, of course, we all know they are proteolytic, but they also are lipolytic. So we actually wonder, we're actually still working on that. And so how is actually ethanolamine released? Is the system activated in foods, or is it indeed sort of a cooperation, and that actually is derived from the spoilage microbiota? And of course, as always, further study is required. So we used a similar approach for the propanediol cluster, and of course I won't show all the data, but we actually confirmed growth activation using all the techniques I mentioned. And also here, there's no growth in aerobic conditions, so a bit of oxygen already blocks this, this activation. And also here we find that addition of B12 is required. So we can find a situation where the B12 cluster in Listeria is activated and that it actually produces sufficient B12 to, let's say, give active microcompartments. So the question was, can BMC PDU, the next one, be activated and stimulate anaerobic growth with rhamnose? I already mentioned that rhamnose can produce, rhamnose metabolism can produce propanediol. So actually, if propanediol metabolism could be activated in this situation, maybe the propanediol could be converted, let's say, to these other compounds and generate extra energy. So for this you have to know that Listeria is very well capable of growth on rhamnose. So, so this is, has been partly published before, but we checked the genome and also looked for the proteins. So actually our proteome analysis showed activation of the enzymes encoded by the LMO numbers and also by the Ra-M and Ra-A. And these enzymes are all activated during growth on rhamnose. So you can grow Listeria in aerobic conditions on rhamnose, but also in anaerobic conditions. But nobody ever checked what happens if you grow them anaerobically with addition of B12. So that's what we did. So this is actually how Listeria is growing aerobically or anaerobically with rhamnose. So it produces acetate lactate in 1,2-propanediol. And we actually incubated Listeria in defined medium with rhamnose, without and with B12, with oxygen and without. And I highlight the anaerobic results because only in anaerobic conditions, with the addition of B12, we indeed see that propanediol levels are decreased and propionate and 1-propanol are produced. So this indicates that if you supply B12, Listeria can now convert propanediol indeed to propionate and propanol. So we again checked microcompartment synthesis, etc. And our model now is shown here. So in the appropriate conditions, anaerobic conditions, with B12 added, we actually see a shift in metabolism of Listeria on rhamnose. So we have propanediol reduced, but still produced. And now we see an increase in propionate and propanol next to lactate and acetate. And this actually means that Listeria can enhance its efficiency on anaerobic growth by producing microcompartments if B12 is there. And as indicated, and the conversion to acetate, or sorry, to propionate, produces additional ATP, which is of course very important under energy limitation conditions and anaerobic conditions. And I will skip this part and move to the conclusions. And this was about the violence, and we actually did violence studies using transwell systems, where we could actually look for invasion, adhesion evasion, and also translocation of Listeria. And what I want to conclude is that so what we actually saw was that upon the activation, and we actually checked all our results again. So if you then look at the proteomics, what we actually see there's activation of stress defense. We find upregulation of CLIP chaperones, which are involved in protein quality. We find SOS damage repair proteins, and this all makes sense because, as you know, if you activate a pathway with a toxic intermediate, it has to be a very efficient fine-tuned system before it's optimally working. So on the way to success, there might be collateral damage, and that is what we see here. And we also find typical proteins upregulated that might be associated with the enhanced violence of these type of cells. So to conclude, BMC, UTE, and PDU support anaerobic growth with these compounds. Activation of BMC and PDU in Listeria requires addition of B12. We never could find a condition where its own synthesis was sufficient to support growth. And BMC-induced cells show activation of stress defense and enhanced in vitro violence. And based on the conditions and environmental niches, BMC, UTE, and PDU may enhance competitive fitness and anaerobic niches. And I indicated on the right, this is from an opinion paper that Andy published together with Alexander Dunk, another PhD student. And there's some very interesting aspects in the co-microbiota that may play a role in this whole process of this interaction, and also vitamin B12. So is vitamin B12 exchange required or not? And with this, I would like to go to the final slide. Thanking Annie again, was supported by Chinese scholarship and the co-supervisors of the thesis, Richard Notenbout and Eddie Schmidt, and of course, MSc students and also cooperators that are on some of our papers. So thank you very much.
Thank you, Professor Abe, for very very much inspiration in these lectures, and this would need now at least a couple of hours of discussion. But unfortunately, your time is over, and we should be polite with our keynote speakers to give at least possibility for one question. Who is going to be the one? Everybody understood everything.
If nobody has a question, I have one.
Please.
Jacko, I'm a mere food microbiologist, but anaerobically, is there a relationship between the temperature and an anaerobic situation? Does Listeria grow better or worse at low temperatures? And can you do something? I mean, I'm thinking of vacuum-packed smoked salmon.
Well, like most microbes, it shows reduced growth rates at low temperature, but
Yes.
it can still grow very efficiently at low temperature, and also even in anaerobic conditions. So that's the exception is it's well tuned to also grow at low temperature.
Yes, but can you put it at sufficient, without freezing it? Will it help if you have it at a very low temperature?
Yeah, it delays growth, so let's say the onset of reaching critical values or levels of Listeria is much delayed. And of
Yes.
course, you try to find other ways with preservatives, etc., to control it.
Yes.
Yeah? Okay. Thank
Thank
you.
you once again.
Oh,
yeah,
there's
we
one
have
more
one
thing.
more. Okay.
Yeah.
Fantastic talk, Jacko. I just had a kind of a naive question for you on these microcompartments. One thing is, can they be isolated? Could they work in vitro? And also, when you show those TEMs, you know, when we do TEMs of different organisms, we see them as well. But we see now what looked like what we thought were phage heads. And I like the analogy with the phage heads inside them. And so inside those TEMs that you saw, like, are they just the ethanolamine ones, or are they multiple different ones? So I had a few questions there.
You mean the numbers of the microcompartments? Or?
Yes, the microcompartments.
Yeah, what we see is it changes, maybe varies between 3 to 6, 8, something like that.
Can you speak to microphone, please?
Oh, sorry. So what we see 3 to 6 or 8. So of course, if you have a phage burst, as you discussed this morning, you see massive loads of phage heads. But it looks very similar. So you have to do density centrifugation or isolate them, characterize them with the proteomics.
And will they work then if you feed them substrate?
If you isolate them and actually, you can actually feed them substrate, just then it has not been done with Listeria microcompartments, but the nanotechnology is actually aimed to produce them in vitro and actually use them for production.
Yeah, that sounds real exciting.
Yeah.
Yeah.
Thank you. And now, organizer would like to express our thanks to the professor, Professor Corey. Thank you. And thank you to everybody. Show goes on, and we leave the stage.
Very welcome to this session. It's quite a diverse session, I think, on the various topics that are very interesting to us as food microbiologists. The first presentation, but I don't see the presenter just yet, is by Lerato Mogozzi, and she's from Bloemfontein. I hope she's in the room now and can start. Yes, there you are. So the floor is all yours.
Thank you. Good day, ladies and gentlemen. As being mentioned, I'm Lerato Mogozzi. I'm from Central University of Technology in Bloemfontein, South Africa. And the research center that I come from is Center of Applied Food Sustainability and Biotechnology. So the title of my talk this morning is Microbial and Associated Contamination in South African Craft Ales. Beer is the most consumed alcoholic beverage worldwide, followed by tea and coffee, as well as water. Czech Republic is a country that consumes most beer. However, South Africa is not the biggest drinking nation in the African continent, with about 56% consumption coming from beer. Contemporary trend of focusing on micro-manufactured produce, also known as a craft beer development, is known as a microbrewer movement as well. Craft beer, as compared to industrial beer, is more prone or subject to microbial contamination. This is because most of the craft breweries don't have resources in place to pasteurize as well as filter their beers. So this results in the proliferation of gram-negative bacteria as well as wild yeast. However, majority of spoilage incidents, they come from lactic acid bacteria. Microbial contamination can affect the beer quality, and this can result in disastrous financial consequences in the microbreweries. So this microbial contamination can be associated with mycotoxins that come from molds, as well as off flavors that can be associated with lactic acid bacteria. So most of the craft brewers, as I've mentioned, they don't have resources in place. Most of their expenditure, it goes to the following. 4% can go to the brewery margin, packaging 18%, 15% to labor, 9% raw materials, 23% due to license fees, and 11% distribution, and 20% to retailer margin. Mycotoxins are secondary metabolites of fungi that can be associated with cereals such as your barley, your maize, as well as your wheat. These are thermally and chemically stable compounds that can contaminate the finished processed foods such as your beer. Mycotoxins, as a result, can cause health disorders when ingested in high concentrations. So in order for us to understand how these off flavors end in the beer, we first have to understand how they come about in the beer, as well as their metabolism as well. So in the beer, we have what you call hydrocyanamic acids, like your p-coumaric acid and ferulic acid. Those are two abundant hydrocyanamic acids that can be found in the beer that can be extracted from your grain during brewing. So this hydrocyanamic acid, your coumaric acid and ferulic acid, normally they come in high threshold concentrations in your aroma and your flavor. That can affect your aroma and your flavor as well. So these hydrocyanamic acids, they can be decarboxylated to your vinyl products as well as your ethyl products. So your vinyl products and your ethyl products, they normally come in low threshold concentration in your beer. However, their presence can become, they're not very likable to the drinker as well. They're not very good in terms of their presence in the beer as well. So they're not acceptable most of the times. They shouldn't be found there. So sometimes they can be associated with some form of contamination as a result of an enzyme called phenolic acid decarboxylase. So if there's a presence of a contaminant in beer that can decarboxylate this phenolic acid, that can decarboxylate the hydrocyanamic acids, it's not good also in terms of the beer quality. Thus, the aim of our study was to determine the level of microbial contamination in South African craft ales as well as to detect any associated compounds that could prove hazardous to consumer health or influence the flavor profile. Now, these are the materials and methods that we followed. We sampled different L beers in terms of the different styles. But in brewing, we have ale beers. We also have what you call lager beers. So we actually focus on ale beers. So we sampled different kinds and types of ale beers from 30 different microbreweries around South Africa. So if you can look at the map on your right, this is the map of South Africa. We've got about 9 provinces. So we sampled different beers from those different provinces. But most of our craft breweries are mainly located in the Western Cape. The Western Cape. So that's where most of our beers were actually bought from. And the different kinds of types of beers that we sampled that are ales, as I've mentioned. We have your IPAs, your English ale, your stout, your whiskey beer, imperial porter, blonde ale, American pale ale, and amber ale. So once we got these samples from these different provinces, we stored them at 4 degrees Celsius until further analysis. So the analysis that were done, we actually were looking for pathogens that can be associated with the beer. We looked also at the spoilage bacteria as well as the yeast, mold and yeast. And we used different selective media in order to be able to detect them. We also took about 50 mL of both for mycotoxins as well as volatile compounds. So our mycotoxins of interest were deoxynivalenol, aflatoxin, nivalenol, fumonisin, ochratoxin, and zearalenol. So according to literature, deoxynivalenol, nivalenol, and as well as fumonisin are more common in your grain that we use for brewing. The volatile compounds that can be associated with contamination and that are normally good for sensory analysis or that are not good for their presence in a beer. We looked at 4-vinyl guaiacol, 4-ethyl guaiacol, 4-vinyl phenol, and 4-ethyl phenol to see if there's any association between them and lactic acid bacteria. So according to our results, this is for yeast and mold contamination in craft ale beers. For our beers, IPA, which is Indian Pale Ale, we only found about 45 coliform units per ml. So this is how we actually graphed this. On my right, we have different styles of beers. So we sampled from 30 different breweries, but we actually grouped them according to their styles to see which beer contained how much of the coliform units. And we saw that IPA, we have the highest of 45, and your stout is 28.3. So most of the beers didn't have any yeast and mold contamination, which is quite a good thing. And we couldn't even associate the presence of this yeast and mold with any of the mycotoxins. So for lactic acid bacteria, as well as other microorganisms as well that I've mentioned, your pathogens as well, we didn't detect any pathogens. We're talking about your coliforms, we didn't detect any of those, your salmonella, we didn't detect any of those. But we actually found your lactic acid bacteria, which is also very interesting according to literature because it's very resistant to intrinsic conditions of the beer. So in this, the range of what we actually saw here was between 20 to 177,500 coliform units per ml of lactic acid bacteria, which means 63% of all the craft beers that we actually looked at were contaminated with lactic acid bacteria. Mycotoxins, according to literature thus far, we don't have any maximum tolerable doses of mycotoxins that has been found in beer. In the grain, like the barley, yes, we have. But according to the literature that we went through and our studies, there was no significant amount of mycotoxins that should be of concern that was detected in the beers that we actually sampled. Now, we also wanted to look at the off flavors, as I've mentioned. Everything that you see highlighted in red, it shows that all these concentrations in these different types of beers were beyond or above the threshold limit. So if you can see on my table above, everything that is highlighted in black in those blocks above, it just show you the the accepted threshold that is needed to be in those different beers. And in the first column, we have 4 vinyl guaiacol. We can see that in most of them, they have this specific compound as well. So also we wanted to see if there's any correlations at all between those off flavors and lactic acid bacteria, because it's known that lactic acid bacteria can produce a specific enzyme that can result in these off flavors. But not only lactic acid bacteria, other organisms as well, like your wild yeast, can have this specific enzyme. But according to spear correlations that we did here, we saw that there was no correlation at all between lactic acid bacteria and those 4 off flavors, that is 4-ethyl guaiacol, 4-ethyl phenol, 4-vinyl guaiacol, and 4-vinyl phenol, as well as vanillin. So there's absolutely no correlation. So everything that went beyond 0.5, we saw it as a correlation. Anything beyond the coefficient of 0.5, then there was no correlation at all, as you can see. Now to conclude, we say microbial quality of South African craft ales needs attention due to the fact that 63% of all our craft beers were contaminated with lactic acid bacteria. So quality assurance, not only of the final product, is vital in our craft beer industries, but the raw materials as well, which can carry mycotoxins, is crucial up until throughout the craft brewing process. And according to literature, mycotoxin levels are within the allowed limits. And also we found that there's totally no correlation between this lactic acid bacterial contamination to the off flavors that we detected in the beer. Now, for our future research, we need to do the next generation sequencing in order to understand the lab community, lactic acid bacterial community, as well as detect the presence of wild yeast, because we couldn't understand what caused those high levels of off flavors. So taking another step to do a next generation sequencing, we revealed if there's any wild yeast that we can associate with these off flavors. And also our future plan is also to look at other chemical compounds that can be associated with any form of contamination, like your major volatiles, for example, your esters, and look at the sugar profile as well. Thank you.
Thank you very much. Are there any questions from the audience? Okay, let's see it.
Thank you for such a wonderful presentation, and I'm from Hungary. I'm interested, I want to ask 2 questions because you were talking about lactic acid bacteria. Can you just specify a little bit which group of lactic acid bacteria was more prevalent? And you said that there was no yeast present, right?
No.
Yeah, but
wild yeast.
I mean, I don't know much about the craft beer, but all I know is that the yeast actually gives a flavor to the craft beer. So why was this difference?
Okay.
From your observation and that.
All right, thank you. I was actually referring to the wild yeast. The wild yeast is the yeast that's not supposed to be there. We have the brewery yeast and the wild yeast. The wild yeast is a contaminant that can come through maybe when you're inoculating your beer or during the brewing process. That's the wild yeast that I was talking about. And then your first question referred to, sorry? Oh, we haven't done the profile yet. That's the next step that we need to do in a study to see what type of lactic acid bacteria that we need to, that we're talking about here. Yeah.
Okay. Thank you very much for your presentation. If there's no further questions. So the next presenter will be the co-chair of today, and this is Aspasia Niziotou, and she works here in Athens at the Elgo Dimitra.
Thank you very much. Good afternoon, everyone. In this presentation, we will show our work on the development of novel molecular markers based on microsatellite analysis for genotypic Metschnikovia pulcherrima which is a common wine-related species, but hasn't received much scientific attention till now. Let me start with some information about wine quality and yeast. Wine, we all know that is among the most valuable and prized agricultural commodities. It is a huge market that is projected to grow even more within the next years. And the top drivers of its value include the vintage quality, the provenance, the rarity, and the ability to age. It is a highly complex product that contains more than 1,000 chemical constituents, which originate from the grape vineyard, the specific grape vineyard that is used to make the must, and of course, a microbial activity. The close connection between the wine quality and the place of origin is summarized into the term vitivinicultural terroir. This term describes the influence of the place of origin in the special features of a specific wine. Traditionally, typically, the main components of the terroir in wine include the climate of the region, the soil, the terrain, that includes the altitude, the slope, and other characteristics, and the tradition that include the vitivinicultural practices that are in this region. Of course, the question that the food microbiology asks is what is the role of the native microbiota? Has any role in the terroir notion? If that is the case, then the term microbial terroir emerges and implies that, hypothesizes that the geographically distant viticultural regions may possess different yeast communities. That means that the first criterion to be met for microbial terroir to exist is that the wine-associated microbiota exhibits geographical patterns, geographical distributions. This is not enough. The second criterion that has to be met is that this microbial geographical differentiation results in metabolic differentiation that is translated to phenotypic differentiation in wine. This is a critical factor in consumer preference and economic appreciation and needs to be confirmed. So which microorganisms can be included in this notion? Of course, the microorganisms that have a significant metabolic imprint in the wine product, and that are typically the fermentative yeasts and the lactic acid bacteria that conduct the alcoholic and malolactic fermentation, respectively. We focus more on yeast due to their decisive role in the alcoholic fermentation. We can divide the wine-related yeast in two major groups. One is the Saccharomyces cerevisiae, of course, the prime wine yeasts that conduct the alcoholic fermentation with its numerous strains. And the other group includes some diverse yeast species that have different metabolic activities compared to cerevisiae and to each other. With these ideas in mind, we started back a few years ago a large-scale sampling. We screened the central and the southern Greece, the main protected designation of origin regions in Greece, 42 vineyards, 3 sampling points per vineyard, and we followed the sampling for 2 or 3 years when grapes were available. We collected more than 2,500 yeast isolates, among which 500 belonged to Metchnikovia pulcherrima. So, of course, the first step was to check whether there was any geographical regionalization of the yeast communities at the species level. And we performed several statistical analyses and found that species composition of yeast communities actually differed significantly among the different regions. This is depicted in the three diagrams, as can you see, where the yeast community is depicted by the dots, grouped together when they belong to a specific region, as shown by different colors. The next observation was the high abundancy of Metschnikovia pulcherrima, which was, along with Hasseniaspora ovarum, the most prevailing species in the Greek vineyards. So we decided to study more this species because it seems to be important, since it has some important metabolic activities. But due to its low to moderate fermentation power, was not considered till now for technological applications. Among the other metabolic activities, the production of hydrolytic enzymes that influence the sensory character of wines, and then the antimicrobial activity due to the production of pulcherrimic acid is some of them that show specific interest. Other activities important for improving the wine aroma are the production of phenylethanol, of several esters like isoamyl acetate, activity on terpenes, and these all characteristics have been found to be largely strain dependent, as often happens in yeast species. So we consider we couldn't find any dedicated tools for typing our isolates, so we consider to do genotyping by microsatellite analysis. This method, microsatellites or simple sequence repeats, are tandem repeated DNA sequences of normally 1 to 10 BP. They are highly polymorphic because they are prone to errors during DNA replication, and they show high reproducibility among laboratories. So in the figures, you can see an example of repeat units consisting of 1, 2, 3, up to 6 nucleotides, and the common practice is to amplify these regions with primers and then detect the differences in the length polymorphisms in a genetic analyzer. To do this for Metschnikovia pulcherrima, we use the genome sequence of strain APC 1.2 that is available at NCBI. And we analyzed for microsatellites of 2 or more nucleotides using the tandem repeat finder program. Only microsatellites of at least 10 repeats were selected in order to increase the possibility for them to be polymorphic. And we designed primers that flank the microsatellite regions, and we use them in amplification experiments. This is a table showing the 15 microsatellite loci that were selected within the gene of Metschnikovia pulcherrima and retained for further analysis. You can see the nucleotide motif that is, ranges from 2 to 4 nucleotides, the nucleotide motif, the chromosome that is located, and the specific primers that were designed flanking the microsatellite region. Of course, then these microsatellites had to be tested. We used a panel of indigenous Metschnikovia pulcherrima strains along with the type strains to test the efficiency and the differences in the sizes. And we select the 9 ones, which you can see the highlighted ones, to perform the analysis in the total number of isolates that we have in our lab. 9 of them were found to show allele variation. They were located at 4 different chromosomes. The strains were first typed with a generic method for Ascomycetes to have groups of isolates, and then we applied them to 66 vineyards isolates from the different regions. So as we speak, the genetic analyzer is running. So for the future goals, I will show you an example of a similar analysis that we have performed in our lab. And what we want to do, we want to take the fragment lengths and analyze them, so to find the genetic diversity between Metsnikovia pulcherrima isolates from the different regions. And this is an example from a similar experiment that was conducted on Laissezia thermotolerans, another wine-related yeast that is well known for its ability to do biological acidification in wine, with very relevance to climate change in winemaking. So in this case, we found that the genetic analysis of thermotolerance based on microsatellites showed a clear structure between the populations of isolates originating from two different regions, from Nemea and Peza, in PDO regions in Greece. The next step is to evaluate the metabolic profile, the metabolic activity of these isolates, of the different isolates, with the purpose to compare the metabolic profiles between the strains from different regions, from different vintages, from different vineyards, and to draw some results. And this is again an example from Laissezia thermotolerance that was discriminated based on kinetic and phenotypic profiles according to the regions they originate. Why would we do this? Of course, these have significant applications in winemaking. We can succeed in differentiating wines to give special characters. We can introduce a terroir effect in wines. We can use strains, specialty yeasts to ferment difficult masts, to increase the volatile profile of the wine or decrease any specific faults, and of course to adapt to the recently emerging climate change. And this is an example of another experiment performed in our lab, where we used indigenous strains to differentiate the producing wine. Thank you very much, and I'm happy to answer any questions you may have.
So I'd like to invite people in the audience if anyone has a question. Yeah.
Hi, thank you for the talk. We used, like 10 years ago, SSR also to identify ploidity levels. So if you have a triploid or tetraploid, you see a few alleles on the same strain. Depends if it is homothallic or heterothallic. So.
Yes, yes. This is why we selected microsatellites also to check the ploidy of the yeast, because it's very important, especially in wine strains, because they show high level of ploidy. And this is an advantage of this method for yeast isolates. Thank you for your comment.
Okay, in view of time, I would like to ask anyone who has further questions, perhaps to do that after the lecture.
Thank you very much.
So, thank you. Then the next lecture will be online via Zoom. So this will be presented by, I first have to change glasses here, sorry. Presented by Raquel Nogueira, and she works at the Marine Research Institute in Vigo. So I hope the connection is working. Yes, hello, welcome.
Hello, thank you. I'm sharing my screen. Do you see it?
Yes, perfect.
Yes. Okay.
Thank you. Go ahead.
Good afternoon, everyone. I'm Raquel Nogueira, and I'm going to present to you the principal component analysis as a tool to cluster Listeria monocytogenes isolated from food-related environments based on their physiological and three-dimensional biofilm features. As you know, Listeria, as foodborne pathogen rose a worldwide concern due to his constantly outbreaks. In 2020, based on the EFSA information, were reported 1,876 cases of listeriosis and 167 deaths within the European Union. Listeria biological features allow it to thrive and survive in food and food processing environments. And regarding risk assessment, different classifications have risen along the years to sort listeria according to one or another biological features. In spite of its accuracy, it is needed to have a wider approach, taking into account more than one feature to give a broad vision of the current situation. The aim of our study has been to cluster listeria isolates according to the tolerance to benzalkonium chloride, the biofilm three-dimensional structure, and the transference by contact to a solid food matrix by using principal component analysis. And why the PCA? PCA decreases the number of the variables in a study system, grouping them into new variables or dimensions. This tool is very useful in food safety risk assessment as it sets and simplifies a wider number of variables into a few dimensions, making the most of the study of the bacterial pathogen into the food chain. The isolates were provided by the EMCC collection and from the Food Technology and Science Department of the University of Córdoba. PCR serogroup was performed following the OFMID protocol, and the benzalkonium chloride resistance was assessed following minimum inhibitory concentration and minimum biofilm eradication concentration in accordance with the classical microbiological protocols. The three-dimensional structure was carried out following a confocal laser scanning microscopy analysis, and the imaging stats were processed using CONSTAT software. The determined parameters were the biovolume, surface area, surface to biovolume ratio, average and maximum thickness, and the roughness coefficient. Finally, we tested cell transference by contact from biofilms on stainless steel coupons to a food matrix, specifically smoked salmon. The results were expressed in percentage of transference in the first 3 contacts. And as a result, 11 variables and 22 isolates were included in the PCA analysis, which was conducted using the FactorMind R plugin and RStudio. From the PCA output, the explained variance was closely to 8%, following the first three dimensions, and the cos-squares values show the weight of each variable in each dimension. Thus, the first dimension is depicted by the structure parameters such as biovolume or thickness, while the second one relies on the percentage of transference and the surface area. From these scores, a correlation circle is built up. In the correlation graph, the vertical length shows the contribution of each variable to the total, while the position displays the grade of correlation among variables. Taking this into account, we can observe that for benzalkonium chloride, MIC, and MBAC, lack of correlation between each other, which underlines the relevance of the biofilm matrix structure to hinder the disinfectant effectiveness. Also, the MBNC correlates positively with rugosity, which would be accounted for the hindering of the disinfectant diffusion across the biofilm. On the contrary, it has a negative correlation with surface area and biovolume. In third place, the percentage of transference have a negative correlation with the thickness. And this could be explained as the thicker the biofilm is, the higher number of cells could be in the inner layer, ergo less active and incapable of being transferred. Another output was the factor map, which allowed the isolates to be classified in 3 statistically different clusters. No significant correlation was found between the cluster definition and the PCR cell groups. And the layout mirrors a population distribution in which the majority of the strains stands in the middle of the distribution of the first dimension. In addition, by overlapping the correlation circle, we can observe the contribution of each variable to the clusters, as well as showing what variables define better each isolate. Thus, cluster 1 is depicted by biofilm tolerance to benzalkonium, heterogeneity, and low values of surface area and biovolume. Cluster 2 is depicted either by transference percentage or biovolume and surface area. And finally, cluster 3 is characterized by guantonic tolerance to benzalkonium chloride, the thickness, and low values of the percentage of transference. It is also really important to remark that the factor map represents a multidimensional space catching just 2 dimensions. And for that reason, the isolates located in the middle of the distribution have no representative variable at all, as all weigh the same in the dimension. To summarize, PCA can provide significant advantages for the microbiological risk assessment, considering that it allows to simplify data display and points out the variable correlation. Also acts as an open system, allowing the input of a large number of variables and isolates. But also, in the particular case of Listeria, it provides an accurate picture about isolates distribution, helping to deepen the knowledge in the bacteria population and features. The cluster characterization in terms of predominant variables pre-selects the isolates more suitable for the study of a particular risk trait, as disinfectant tolerance or third transference, making easy the accuracy of future works in food risk assessment associated with Listeria traits. At last, the simplicity and reliability of the PCA makes possible an integrative approach to risk assessment, leaving the door open for future analysis. And that's all. Thank you very much for your attention.
Thank you very much. Do you have any questions from the audience, please? Yes.
In one of your introductory slides, you list some characteristics of Listeria, one being low water activity. All other conditions being optimum, what is the lowest water activity at which Listeria will grow?
Thank you very much for your question. The truth is, I don't really know the answer, because my knowledge about Listeria biological features is very thin right now. So I'm so sorry if I can't answer you right now, but I hope I will be answering you in the future if you are so kind to contact me.
Thank you.
Okay, if there are no other questions, we can move to the next presentation. Thank you very much. It's Mr. Omer Sambach from Israel?
From the Technion in Israel. Okay.
You may.
So, thank you for the introduction. My name is Omer Sambach from the Technion, and I'm here today to present my study regarding the microbial community's interactions in sourdough starters using a genome-scale metabolic modeling. So to start from the beginning, we need to start from sourdough bread. It is a staple food for human nutrition that made much earlier to the discovery of microorganisms. It is composed of 4 main ingredients: flour, water, salt, and of course the sourdough starter. If we will take a closer look inside the sourdough starter, we will see that it is composed of 2 main ingredients, the flour and the water, that are mixed together and then let to ferment. After a while, it will achieve a stable microbial community composed of both yeast and bacteria, mostly lactic acid bacteria. Now, from sourdough starter to sourdough bread, among some other technical parameters, we have the metabolic interactions that are responsible for the bread flavor, aroma, volume, acidity, and much more. But how much do we really know about the exact metabolites exchanges between the bacteria and the yeast, and also among the bacteria themselves? The answer is that we don't know much. And basically, we can refer to it also as a metabolic black box. And that was exactly the aim of our study, to shed some light into this black box. We chose to do it using a genome-scale metabolic modeling, which is a way to represent the cellular biochemistry in a mathematical formal way. In more simple words, we need to take a DNA sequence, a whole genome DNA sequence, convert it into a system of reactions, enzymes, and metabolites that can be illustrated in a nice way, or also in the mathematical formal way of matrix as a system. We solve the system using linear optimizations that actually maximize the objective function of the system. Now, since we are talking biology, the objective of any living cell is to maximize its growth. And this is also the case here in the models. So the first step in our research method was to build the bacterial panel. Here you can see the panel composed of 10 of the most common lactic acid bacteria that occur in sourdough starters worldwide. We build in silico communities, each one composed of one yeast, Saccharomyces cerevisiae, and 3 lactic acid bacteria from our panel. So if we will do it, we will end up with 120 communities to model, and this is exactly what we did. We choose the one yeast and 3 bacteria species to resemble a real sourdough starter composition. So we look at the data, and the first thing that we notice is a correlation between the bacterial community members to the growth rate of the yeast Saccharomyces cerevisiae. Here in the heat map, each row represents a bacterial species, and each column represents one modeled community. So by default, in each column, we have only 3 colored spots, and the color represents the growth rate of the yeast. Now, you can see the diagonal trend pointing to the Pediococcus species and to Lactobacillus sacchae in a way that correlates the percents of those species to enhancing the growth rate of the yeast Saccharomyces cerevisiae. If we will do another analysis, this time the color represents the community CO2 secretion flux. Again, we have here the diagonal pattern showing that the Pediococcus species and Lactobacillus sacchae also correlated to a better lavaining ability of the dough. So we ask why, what is so special about the Pediococcus and about Lactobacillus sacchae? And more specifically, we ask, can similarity in the consumed metabolites among community members outline a competitive relationships? To answer this question, we use the consumed metabolites profile of each of analysis using a principal component analysis, PCA, of each one of the community members. So in green, you can see the cluster of Saccharomyces cerevisiae, and right on the other side of the main principal component, you can see in shades of orange the clusters of Lactobacillus sacchae and the Pediococcus species indicate that they don't share the same consumed metabolites and they don't compete on the same nutrients. However, the other 6 lactic acid bacteria in our model cluster pretty much above the cluster of Saccharomyces cerevisiae in a way that indicate a positive competition on the same nutrients. So to answer this question, we know that a significant difference in the consumed metabolites profile benefits the microbial community through a non-competitive interactions, and of course, vice versa. The other question was, do food chain interactions shape the microbial community properties in sourdough starters? Now, when we talk about food chain interactions, we're looking for a metabolite that's secreted by the bacteria and are later consumed by the yeast. And when we looked on the model and searched for such metabolites, this is the network analysis that we've done. And for example, you can see that the Pediococcus parvulus is secreted histidine to the medium, and the histidine is later consumed by the yeast. And if we look on all of these metabolites, most of them are actually amino acids that are secreted by the bacteria and consumed by the yeast. So we went back to the lab and we did show that if we add amino acids to a defined growth media, it is support the yeast growth in terms to a defined media without any nitrogen source. However, you can see that both histidine and lysine are actually inhibit the yeast growth. Now that was done on the laboratory strain S288C. However, we also expanded to include some more Saccharomyces cerevisiae sourdough isolates that we have in our lab, also Kazakhstania humilis isolates that we have in our lab, and also to some other commercial baking strains. And all the time we saw the same trend. And once again, lysine is actually inhibit the growth yeast. So we went again back to the models and we classified the bacterial panel into two groups. One group indicated in blue as an average of a positive lysine flux, meaning they tend to secrete lysine to the media. And the other group, here you can see them in red, as an average negative lysine flux, meaning they tend to uptake lysine from the media. And when we did a co-culture assay in our lab, it is actually support the classification. You can see in gray the monoculture of only Saccharomyces cerevisiae alone in a defined media supplemented with lysine. In blue, you can see the co-culture of Saccharomyces cerevisiae and one of the bacteria that was predicted to secrete lysine to the media. We have no difference. And on red, you can see co-culture of the yeast Saccharomyces cerevisiae with one of the bacteria that was predicted to consume lysine from the media and reduce lysine concentration. And when it's happened, we can see that the Saccharomyces cerevisiae is now able to grow to a much count, to a higher CFU to ml. So to sum this up, we know that the bacteria influence the yeast growth also by altering the amino acid flux. But what about the other interaction, the other direction? How is the yeast support the growth of the bacterial growth? So what you can see here is all of the metabolites that we detected that are secreted by the yeast to the media and later consumed by the bacteria. The y-axis represents the Pearson correlation between the bacterial growth to the bacterial uptake flux of the specific metabolites. And we can notice that in red we have amino acid that we weren't detected earlier. In green we have B vitamins, riboflavin, B2 vitamin, and also B5 and B1 that's secreted by the yeast to the media. And we also have in blue some carbon sources such as glycerol and L-malat. So if we have the full picture on these interactions, we can conclude that we have a bidirectional nutrient dependencies between the bacteria and the yeast in sourdough starters. Now the last, and honestly the most challenging question was, can the model's findings help us to predict an actual sourdough behavior? So to answer this question, we took all of this is the CO2 secretion analysis, and we divided all of the 120 communities into two groups. One group that is predicted to have a high CO2 secretion rate, and the other that was predicted to have a low CO2 secretion rate. We randomly choose four communities from each group, and then we synthetically build them in our lab. We inoculated sterile flour and sterile water with the specific bacteria and the yeast, and let it ferment for over 1 week with daily feedings, daily refreshments, just like you make a sourdough. And at the final day, we did our analysis. Here you can see some photos from the experimental setup. You can see how we grow the sourdough starters in our lab, and also the gas production system, where we put in a sealed glass bottle leavened dough made with a specific sourdough starter and let it ferment in a controlled environment for over 20 hours. And we have a success in predicting the CO2 production rate. You can see it both over time, where we can distinguish between the red line that resembled the group that was predicted to achieve a higher CO2 secretion rate. It did achieve a high gas production rate. And we can see the rates in the box plot where we have a significant difference of 20% in the means, meaning that the green block spot represent the communities that were predicted to achieve a high CO2 secretion rate, indeed achieve a higher gas production rate compared to the orange box plot. So to sum it up, if we started from the black box, we now shed some light into it. We know that if we have a similar consumed metabolites, it indicates a competition, and of course the other way around. We demonstrated that we have in sourdough starter bidirectional nutrient dependency that help to shape the microbial community. And we also showed some in vivo experiments that support the model's findings regarding the Levanian grades. Now, last slide. During the study, my PI always asked me, what about the taste? And at the end of the day, people should eat this bread. So I have here only the preliminary results regarding 30 of the most common aroma and taste compounds that appear to be in sourdough starter and sourdough bread. You can see in the upper green heat map the relative flux, and at the bottom you can see the community member by per column, per community, and we can see some relationship between the metabolites. But still, once again, it is the preliminary result, and we are going to work on it more. To finish, I would like to thank my PI, Professor Yechezkel Kashi, also to Dr. Shiri Freilich from the Volcani Center, to all of our lab members, and for you for listening. Thank you.
Thank you very much for the presentation. Are there any questions? If there are any questions, we have a question from the audience.
Is there no? I can't see it. Oh, yeah. Okay.
Thank you very much for the very interesting talk. I have two questions for you. The first is, I couldn't understand the first PCA graph you showed, if it was the result of your in silico predictions or was it experimental predictions?
So it was the result of the in silico modeling, yeah.
Okay, okay. And the second question is, why do you have a mechanistical perspective of the influence of so many amino acids produced by lactic acid bacteria in the medium that may help Cerevisiae to grow? Why is lysine, for example, shutting down the growth of Cerevisiae while the other amino acids stimulate the growth?
So specifically to lysine, there is, it is a well-studied amino acid that we know that impact in a negative way Saccharomyces cerevisiae. The exact mechanism, for me, it is unknown. I read some articles that suspect it was some byproduct of the degradation of lysine. Some other suspect it was the lysine itself that has a toxic effect on the yeast. But once again, I don't have a specific answer for the mechanism. Thank you.
Okay, thank you very much. We may now proceed with our next presentation, which is going to be via Zoom. Okay, it's by Beatrix Stacey from the University of Veterinary Medicine, Vienna. Yes. Hello. Hello, can you hear us?
Yes. Hello from Vienna.
Hello.
Now back to the Listeria topic. I would like to show you some post-outbreak management data. And we had a problem with a meat processing facility in Austria related to a food outbreak, to a human cluster. And we have the question, when could food production be considered under control again? Sorry, I have a problem with the slides. Outbreak situation, you can also read it by Piecka, which was published in 2019. The outbreak related to meat products, first initially 7 patients got ill from November 2015 to September 2017, and the SNP analysis, so the genetic relatedness between these isolates, differed only by 1 to 4 alleles, genetic alleles. Food and environmental isolates from a huge sort of companies, of Austrian companies, was collected by the reference laboratory, and they compared everything to the human cluster, to this cluster outbreak isolates, and they identified one company, let's call it company A, where they identified a genetic relatedness, and there were only 0 to 7 alleles difference. And at the end of this whole comparison, they identified the industrial slicer as a source of recontamination. So in meat industry, most equipment which is related to recontamination are slicers. And there were also, in the whole comparison, were also other companies included, also isolates from fish companies and other ready-to-eat products, and also from deer meat products, but they were not considered to be related. The analysis included one scheme applied in Austria, developed by my colleague Werner Rupic. You can also read it within this article, and there is not the whole genome included by this comparison between European reference laboratories. It's only 1,700 genes included from the core genome. The standard process is to compare it by software and to get a minimum spanning tree, what you see in the middle of the slide. And there is the comparison of human isolates from these outbreak isolates and also from the different companies. And the scheme, Originum MLSD scheme by Rupic, identified one clonal type, CT. I marked it in green. It was CT1234, which is corresponding to the Institut Pasteur scheme, and this clonal type calls 100-117. In Austria, we have only this data for the human comparison and also the food-related isolates with this scheme. And when we go to the Pasteur scheme to get a hint how, on a global level, this type connected to clonal complex 155 is present, then we can have a look at the database. And if you have a snapshot on this clonal type, which was related to this human outbreak, you see that the Pasteur site includes isolates from different countries, and the source was, for instance, wild animals, wild pig, deer, tonsils, and also the pork processing facility in one country in Spain. So with this type, wild animals and pork processing environment is related or was related in the past. The genetic type, clonal type, has special features, strain features. For instance, adaptive element, stress survival 1. This is you can find it under attributes, and also has a special adaptation to disinfectants applied in processing facilities. So our situation was a post-outbreak scenario. So with the company, which was then related to the outbreak, 4 food lots were identified positive for Listeria monocytogenes, and different genotypes were already at that situation identified. At the onset of our investigation, the outbreak was already traced back, and there were no human outbreaks. First takes could be attributed to from 2018. We had a critical question. We had a lot of interviews or talks to the management team and also to the agency, and we saw that we all had a critical question that how could a food business operator manage such a post-outbreak situation if he is not aware of its role in the outbreak, and also to work with that whole genome sequencing data. The FPO tried really to manage the situation very actively, so however it became clear that there is in Austria, it's not an authority-approved guideline for the management of a listeria contamination after this outbreak. So the agency decided that if the company is getting rid of all the contamination and if active batch release is applied, that each batch is controlled before it is set on the in the market at retail level, and that this is negative tested for Listeria monocytogenes, then the processing facility might be active with producing food lots. So we started together in 2018 environmental sampling, and also tried to map it on transmission maps together with Company A. The processing facility processed different kind of ready-to-eat products, but also convenience products, which, for instance, schnitzel, meat products which have to be heated at the, for instance, at home, and so that it's not ready to eat. So different kind of meat products were processed in this processing facility, and you see that there is a blue line where you can follow the uncooked products, and the red is the ready to eat. At the beginning of the outbreak, only the slicer was identified that this should be related to the outbreak. But then we established a sampling plan and tried to get the hint of the entrance of Listeria monocytogenes and to get rid of Listeria monocytogenes from the environment. If you have a look at the table, we identified the outbreak strain still in 2018. And these are 19 sampling events, and ST155 was present until sampling 7. The general Listeria occurrence in all the environmental samples was 2.4 for Listeria monocytogenes, so it got better through the time. If you have a look at the genetic diversity, in green you see the outbreak-related clones, so it's clonal complex 155 or clonal type 1, 2, 3, 4, and this was detectable at different kind of sampling sites, for instance the hygiene lock, different sites of the hygiene lock, the hallway, then also men locker on the shoes, so it was really widely distributed. Also in the raw material tumbler site, at the schnitzel site, so it was not only identified at the slicer. So we tried really hard to reduce this contamination, and we also identified other genetic types, clonal types, at different sites of the convenience production line and also the raw meat cutting site. So this is the diversity. In different colors you see different genotypes. I will not go into detail with that. So you see the heterogeneity of all the isolates present in the meat processing facility. And then you see the minimum spanning tree. So we identified through these years 4 different clusters. Then one major cluster was identified at the beginning, 2019, and then we got a lot of isolates of clonal type 9, which is very present in pink, and this is very present at meat processing facilities, and it is strongly related to the raw material. So every day, if you work with different meat types, Listeria is entering meat processing facilities at small amounts. So we have to solve the problem at the raw material side. We had some management points, how we reduced Listeria prevalence with an easy way. So we identified gaps, hygiene gaps, and so you saw a lot of isolates from the hygiene log, so we repaired together with the company the hygiene log. This is often a hygienic gap, because this is sometimes the disinfectant does not really get into the hygiene log. Then again, we saw that the slicer team has to be one team, not different persons who are maintaining the whole hygiene standard of the slicer. So at the slicer, we did not identify any Listeria from 2019 on. Then we had to work further. So we saw that conveyor belts were not easy to clean. So we tried to set the sampling plan that we always had the information before and after cleaning and disinfection, that we had a comparison if Listeria is in some niches present still after sanitation. So then we also identified problems with the conveyor belts, and we tried to interrupt some transmission, Listeria monocytogenes transmission cycles occurring between raw material, intermediate product, and slicer. We also saw that the crate washing system was not working very well. The temperature was not fitting. It has to be about 80 degrees, but it only achieved 50 degrees for the main washing with sanitizers and so on. So I will not bother you, but these are some easy ways to interrupt heavy contamination in a meat processing facility. And we have to, this is our outlook, but we really have to go into detail. It is important to track genotypes at the processing facilities, but many types are really entering from the slaughterhouse and also the primary production. So we have to source attribute these types which are very present in the primary processing. So for instance, we get the hint that clonal type 9 and 121 is coming from, for instance, pork producing facilities, as you see here, published by colleagues. But this is applicable for France, but we also have to know this for Austria if it's the same situation, and to compare this then with our colleagues from the reference labs at other countries. Okay, so the outlook is also to go into detail with the adaptation of these strains to the sanitizers and disinfectants, and also to compare it with, to share alternative monitoring concepts, to make tailor-made sampling plans for each company, because in these small and medium small scale enterprises, it does not fit to each company size what you think about monitoring is fitting. So this is the plan. Thank you for your attention.
Thank you very much for the presentation. Is there any question from the audience? Yes, please.
Thank you very much for the presentation. Looking at all the things you found at the production site, it doesn't look like a new and modern and, very well-kept production site. So my question would be, were they certified to any food safety standard?
The company, yes, of course, they have IFS audits, and they are controlled, and they have also to, in Austria, they have to sample food lots and also slicer dust, for instance, for meat products, and to verify that they are negative for Listeria monocytogenes. They also have to show that they have a hygienic situation for personnel and also for the processing facility, and they provide those data, but still they get a huge diversity in Austria. They buy a lot of raw materials. So, for instance, this company is diverse. They have poultry meat, pork meat, and beef meat, and this is daily bought by different slaughterhouses. So there's a high diversity entering of Listeria entering this company.
Okay, thank you very much again.
Hmm?
Okay. Thank you. We may move to our last presentation from the chairperson, Dr. Marion Wells-Benny from Ninso, the Netherlands.
Yes, hello. Once again, good afternoon. I'm going to talk today about an organism that is, by most of you probably known as a spoilage organism and not so much a foodborne hazard. But despite its general profile, we did look at its toxinogenic potential, and I'm going to talk about the spore former Bacillus licheniformis. Let me see how I do this. Yep. So just a very short recap on foodborne illness. So most of you know the pathogens like Salmonella, Listeria, Campylobacter, and these are typically causing foodborne infections. And you probably also all know the more typical foodborne intoxication causing agents, such as cereulide produced by Bacillus cereus, botulinum toxin produced by Clostridium botulinum, and Staphylococcus enterotoxin. However, I'm going to talk today about some less well-known toxins, and particularly produced by Bacillus species. So when I talk about these toxins, it's going to exclude Bacillus cereus. And what I would like to highlight is that, particularly in relation to Bacillus licheniformis, there have been a number of outbreaks reported over the years. Not many, but it is actually happening. And there are a couple of cases where an organism, Bacillus licheniformis, was isolated from infant food and raw milk. Also from contaminated milk powder in a kindergarten, and this actually led, sadly, to the death of a child. And there are other instances where this organism has been found in products that have led to foodborne illness, so mostly the vomiting type. Now, just a little bit on Bacillus licheniformis. It's a spore-forming organism. So as you all may be aware, the organism is growing, and once it reaches a very high cell density, or when it senses nutrient deprivation, it tends to convert, or the mother cell tends to have an asymmetric cell division, and endospores are produced. And you see them here on this little movie, which was made by my colleague Robin Eilander. And once these spores are produced, they are super stable. So this is a good thing to remember. So the spores can be dormant. They can be dormant for even many years or decades even. And not until they experience very favorable conditions, and usually this is due to nutrients, they germinate. So they go from phase bright to phase dark. And then from this phase dark spore, a cell emerges, and then growth can reappear. So this is just a very short recap again on sporulation and germination. And when we look at Bacillus licheniformis, it is an organism that is ubiquitously present, so you find it everywhere really. So in decaying plant material, in soil, in water. And it is quite a robust organism. It can grow at a very broad temperature range from, let's say, 7 degrees up to 60. And when you're culturing it, it's actually sometimes very difficult to handle on plates. It's a slime former, it spreads very easily, it can overgrow plates. And actually this ability to spread on plates and to swarm is very likely related to the compound that is actually also causing foodborne illness. And that is the compound, the compound lichenisin. So lichenisin is very similar to another compound produced by Bacillus subtilis, and it's actually, the structure is shown here. So these compounds are surfactants. They have a very potent ability to reduce the surface tension, of where they are growing, essentially. So these compounds, they are produced, they're non-ribosomally synthesized, so it's a very large gene cluster that produces different modules, and these modules actually couple different amino acids, and then there is this circular peptide has a carbon tail, which typically is around 15 carbons. So this structure is extremely stable. And this is actually part of the problem for food safety. So you can actually sterilize it, nothing will happen to it, it's very stable. So what is known about this compound is that it causes membrane permeabilization, and that in turn causes nausea and vomiting. And some known concentrations needed to establish that have been established around 40 to 60 micromolar. Now, Bacillus licheniformis, a step back to food, it's a very common contaminant in food ingredients, and it's been quite well known that in milk powders, it can be the predominant species. So it really depends on the processing facilities. So in milk powders, you can get very dominant presence of very high heat-resistant spores like Geobacillus, but depending on the location and the facilities where milk powders are produced, it can also be Bacillus licheniformis, and there's quite a few studies on that. So coming from the background at NIZO on milk powders and dairy products, we have actually been transitioning to plant-based materials and being able to make plant-based dairy alternatives. So we were actually trying to make all these products, and lo and behold, we were encountering Bacillus licheniformis in a lot of the products we were trying to make. So we were actually wondering, okay, what about this contaminant in all these plant-based sources? So we started to do a survey on a lot of different compounds like pea and fava bean and chickpea, almond, and what we actually found is that the majority of all these ingredients, they contain quite high levels, sometimes up 10 to the 3, 10 to the 4 spores per gram, and of spore formers, and nearly all of these samples contain Bacillus licheniformis. And maybe this is not very surprising considering its natural amnesia, so it's really associated with plants and perhaps also root systems. But this is definitely something to bear in mind when you're dealing with these type of new ingredients. What is worth mentioning, other than that it's common in foods, some of the spores are very difficult to kill. So usually licheniformis spores will be killed by UHT, but 10% of all the strains that we've been isolating contain genetic elements that are actually making them more heat resistant. And quite a few, so roughly around 10% of all these strains, you cannot inactivate them even by boiling them for half an hour. And this is actually the type of heat that, is really the max for what you can use in plant-based ingredients. So then, coming to the aim of the study, so knowing that this organism is present in a lot of these plant-based ingredients and the type of products we want to make with these, where you cannot apply very high heat loads because it actually destroys the properties of the products we would like to make, we wanted to assess what the risk was of lichenisin production in foods. We have actually done quite a bit of work on plant-based ingredients, but I'm not presenting that today, but I would like to present the work that we did on the initial work on LB medium and on skim milk. So what we did, we took 11 different food isolates of Bacillus licheniformis, and we used these to culture in LB and milk, and we assessed lichenisin production, and in addition we looked at the toxicity to intestinal cells to see, okay, you know, the levels that you may encounter in food, could that be relevant to what you can experience for human intestinal cells? Well, this is a very detailed overview. It's actually in the publication that we have. I'm not going to go through this in great detail, but what we did here, we took 11 real food isolates, so you can find them anywhere really, but these have been problematic in foods. And in addition, we took a type strain, DSM 13, and we established just its common, or its growth potential, looking at growth at different temperatures, presence of salt, ethanol, and also establishing whether or not it could grow under anaerobic conditions. And what we saw is that the growth of the organism is actually really robust. It grows at 37, but most of the strains also grow very well at 55 degrees. They grow well at 37 and 55, even in the presence of 1 molar of sodium chloride. The organism grows in the presence of 4% ethanol and under both aerobic and anaerobic conditions, in this case actually supplemented with some potassium nitrate. In addition, we looked at the genetic profiles of these strains, and we did find these lichenisin clusters in there. And in addition, we established whether they could produce the lichenisin, and we found that was positive for all the strains. But some were much better than others, and this is just displaying for the situation for 3 strains. So on the left here, you see this reference strain, the DSM 13 strain, and the 2 other strains are quite high producers. In the top, you see in the red plates are hemolysis caused by these organisms, and you see that these 2 strains on the left are actually lysing blood cells. And in the bottom, you see an oil displacement. So what you actually do is you drop some of the supernatant onto oil, and if it has this ability of like this detergent ability, it displaces the oil, and then you see the water underneath. And actually this was very apparent for some strains, especially for these very strong producers. So we carried on the rest of the work with these strains. And what you see here is actually the growth of these strains in LB and in skim milk. So LB on the left, skim milk on the right. And you see that it grew faster in LB than in skim milk. But in both cases, very high cell densities were reached, around 10 to the 9. But if you then look at the ability to produce lichenisin, this was quite substantially different in LB versus skim, so much higher concentrations in LB, and in addition, lower concentrations in skim milk. But what you also see is that, so the white strain is a relatively high producer, the black bar is likewise, but this reference strain, the DSM 13, was very poor producer. So this actually indicates that there is a very large difference between the strains. I'm not going to go in very great detail on all the things we did, and we also looked at different chain lengths, etc., and they may actually have effect on the toxicity of the compound, but that can all be found in the paper. But one other feature I would like to highlight is that the temperature at which the strains were grown was also quite determining in the levels of lichenisin produced. And this is shown here, and we saw actually that the strains, when they were grown at 55 on the right, they produced less lichenisin than at the temperature of 37 degrees. Just to get back to the previous slide, so the levels that we found were actually around 36, let me see, how much was it? Yeah, 36 microgram per milliliter. And this now relates to the levels that this was produced in a liquid medium. And we also assessed what the concentration of lichenisin was to reduce the cell viability, in this case of CACO2 cells, by 50%. And we found that this was in the range of, let's say, 17 microgram per milliliter. So what is really important, I think, as a take-home message is that lichenisin can be produced at levels that are physiologically relevant. Of course, you cannot make a direct relation between how much is present in a drink and how much is having an IC50 in the human gut. Of course, there is the ingestion and whatever happens in between, but the levels are definitely relevant. And that's obviously also clear from the outbreaks that have occurred in the past. One last thing that I would like to remark is that the levels that the moment you start to see lichenisin production is really in the late stages of growth. So it's the late exponential phase and the stationary phase. This is where you see lichenisin produced, and we never saw it under the level of 10 to the 5 CFU per mil. So if Bacillus licheniformis is present in food at very low levels, under 10 to the 5, let's say, it's very unlikely that this will pose a foodborne hazard related to lichenisin production. However, there's always this potential of the risk of foodborne intoxication by this organism, and that really depends on the strain that is present, the food composition, and the storage condition of the food. So, yeah, to keep a product safe, it's either one of the two. One is to inactivate all the spores, make sure they're not there. The other would be to prevent outgrowth. So with that, I would like to conclude my presentation. I would like to thank all the co-authors on this. So Claire Yeek, she's nearly at the stage of defending her thesis next month. Mansa, Guido, they were actually part of the study at NISO. Blanca and Jerry at the host microbe interactomics group in Wageningen, and Chaco Abe himself is also co-promoter of Claire. So thank you very much.
Thank you very much for this nice presentation. Are there any questions from the audience? Yes, please.
Hi.
Very nice talk, Marianne. Good to see you again. I had a question. I was just wondering, can you consider lichenisin a kind of a variant of surfactant?
Not quite. It has two very distinct differences in the amino acid composition of the ring.
Yes.
And that is pretty consistent. So.
Oh, okay.
But in a way, yes.
And is their cytotoxicity the same?
It's very similar. It's very close to each other. Yeah, we did test the surfactant as well. I left it out of this presentation.
There was, I don't know, maybe you've seen it, there was a few publications on anti-COVID activities of surfactant and these, because, of the lipid outlayer and so they're anti-COVID, I don't know.
I'm sure it will be anti a lot of things.
Yes, it would be, but I'm sure any detergent will be probably,
yes.
But in these organisms, a lot of these organisms, like you have Velanesensis, amyloliquefactions, they produce a lot of these kind of thiopeptides, including fengisin and also bacteriocins like amylocyclicin. I was just wondering, in the repertoire that's in licheniformis, what do they produce? Do they produce fengisin,
Licheniformis not so much. Subtilis has a bigger repertoire than Licheniformis. But what is very striking is that all strains contain, as far as I've looked, all strains of Licheniformis contain this non-ribosomally synthesized, this cluster, which is huge. So it's a very big genetic burden on the strain. So it must have a very big advantage for the strains to be able to produce it. Okay, so I think we're, I don't know if anyone else has questions, please come and see me. I think we're ready for coffee. So, or if it's there, or the end of the session.
think we can begin the new session. So welcome to the hybrid and mixed session B and C. My name is Hélène Valentine. I work in Rennes at INRAE in France, and my co-chair is Christian Botta, and he will introduce himself.
Okay, thanks, Hélène, for kindly introducing me. So I'm going to read the long title of my presentation, Towards New Strategies for Extending the Ground Beef Shell Life, the Cases of Electrolyzed Water, Bioprotective Cultures, and Metataxonomy-Based Surveillance. First of all, I want to say thanks to the committee that gave me this possibility to present here. I'm really happy to be again in presence, face-to-face, especially here in Athens. So let's move to the case of study. If you may understood, we are going to talk about ground beef. Why ground beef? Because it's, let's say, the worst case scenario in terms of microbial contamination and perishability of the final product. So from the early stages of the slaughtering processing, from the carcasses are contaminated from the hides, especially if they are themselves contaminated from fecal residues. Along the processing line, this initial contamination grow step by steps, collecting spoilage bacteria from operators, the hands of the operators, saliva, but also cross-contamination between batches that is carried out by water and surfaces, and also gears, knives. So at the end, we reach the point of trimming, the mincing, the grinding passage, in which this contaminating, this spoilage microbiota is spread in all the products. And so we have a product in which the only hurdles to increase the shell life are the low temperature and the type of packaging. And this shell life is, let's say, proportional to the amount of bacteria or spoilage bacteria that have been collected during the process. So practically, the aims of this study, there are more than one study, is overall to find the most proper way to extend the ground beef shell life, applying different approaches. First, we tried with decontamination of the surface meat trimmings with electrolyzed water. We checked during the shell life the output of this treatment, performing plate counts, metataxonomic analysis, and combining these results with the volatilome, and also checking at the end of the shell life the potential presence of biogenic amines. In another study, we tried to exploit the let's say, self-protection capability of the ground beef by isolating or trying to isolate bacteria able to produce bacteriocins from the early stages of shell life, and then use it as a starters in this product that is not a fermented product against the contaminant bacteria, against the spoilage microbiota. So introducing them again in the final product. Going to the results, the first case of study, we treated trimmings for 90 seconds by immersion in electrolyzed water for at 100 ppm of free chloride. We decide this time and concentration in relation to previous laboratory experiments. We performed all this in collaboration with a company, 46 samples, 2 different production in 2 different periods were followed. So with treated and untreated thesis. Unfortunately, we cannot see the taxa. We have a problem with the taxa, but about the results, we observed an effective decontamination on the surface of the trimmings, but this was not anymore observable in the ground product, in the ground beef. On the other hand, we could observe a significant separation between the microbiological dynamics of production A and production B. So higher presence of lactic acid bacteria and total viable counts for the production A that showing overall, let's say, a faster spoilage tendency that was associated in terms of metataxonomic analysis with a higher biodiversity in the first 6 days. And also the presence, you cannot see, but I'm going to tell you by memory, higher presence of lactic acid bacteria like Streptococcus in the first stages, the yellow one. In both production, we identified common indicators of shelf life end in the Photobacterium phosphorum and Lactococcus species that were strongly associated with the production of biogenic amines, tryptamine and tyramine, and also negatively correlated to the linked amino acids. So we were able to discriminate between production in different periods. We were wondering at this point if we could discriminate between production, in the same production between batches of different origin. So different carcasses, but processed in the same environment, in the same condition, and with the same materials. From the microbiological point of view, from the counts, we could not discriminate the two batches. We could also, from the volatilomic profiles, is not visible, is not significant a segregation between the two batches, but is significant, especially for the, in this case, we analyzed also the microbiota, the fungal community. This differentiation between the two batches produced in the same day is possible with was possible with the metataxonomic analysis. Going more in deep, we can see that we have an initial transient microbiota that is common between the two batches, and mainly harboring Cocuria, Staphylococcus, and other bacteria collected probably from the skin of the operators and animals. But then the two, sorry, I have a problem with the pointer. The two microbiota are splitting, so we have a higher presence of Canobacterium divergens and higher production as a consequence of acetoin and hexanal, so hop flower related, volatile compounds related to hop flower. And so we can see more clearly, I hope, here with this network analysis, in which we analyzed the co-occurrence between all the taxa, and we observed that we have a purple group of highly co-occurring taxa, so ASV that are going hand by hand in the data set, and so they are present all together. This purple group here is here represented with this purple triangle, represent the initial microbiota. Then we have a turnover, but it's different if we talk about bacteria between the batch A and B. In the A, we have the development of Leuconostoid gelidum. In the batch B, we have higher presence of Canobacterium divergensis, as I said, and also Lactococcus species. That, as we said before, is an indicator of shelf life end. So this is the signature of the original carcass, the signature of the original trimmings, while the purple module, these species, are the shared community that came with the grinding passage. At this point, since we state that we observed this difference clear difference between the beginning and the end of the shell life, we try to exploit the potential protective, self-protective effect of this product, isolating, trying to isolate from the early stages, from the day of production, bacteria able to inhibit what they are the spoilage taxa, so the consortia of bacteria that are present at the end, so in the spoilage product. We perform this by challenge tests with the agar-well diffusion assay and other spot-on-low, searching for, also here is missing the color, searching for halos of inhibition. As indicator, we use as the first step the spoilage consortia collected from the same product. If we go on, we made for more than 700 challenge tests. We find out that 41 isolates from the time zero were able to inhibit this spoilage consortia. We tested all these 41 against several pathogens, and we found out that like 2 of them were able to inhibit Listeria monocytogenes. That is quite a big problem in the ground beef. So we proceed with these 2 strains, and for one of them, we confirmed the capability to produce an antimicrobial peptide, or more than one antimicrobial peptides, probably for sure nisin, and we're going to see later, but also something else because was able to resist in the agar-well diffusion assay to the treatment with proteinase K. So this good guy is Lactococcus lactis, a species Ornie. So from the same genus, we have an endpoint indicator for the shell life, Lactococcus species, but at the same time, we can find also potential protective strains. We characterize this strain in terms of phenotype. It's able to grow low temperature, in presence of salt, no so many resistance against antibiotics, and mainly is able to inhibit in liquid media, BHI, broad heart infusion, the growth of Listeria monocytogenes. At the same time, looking inside the genomes, we observed the full operon, the whole operon for nisin, but also other gene codifying. We are trying to understand the mechanism behind, but this is another story that I hope to tell you in the future. Right now, we tried to apply what is this promising strain in several replicates in the same product, so in ground beef, but unfortunately, in this case, we couldn't observe the same inhibitory capability as we saw in the synthetic media. This is probably because here we are in a solid phase, not liquid. There is no direct contact, or it's difficult, the direct contact between Listeria and these bacteriocin-producing strains. Going at this point to the conclusion, making a, let's say, a connection between these that are different studies, we can say that the treatment with electrolyzed water is unlikely to be effective strategy to ameliorate, to improve the quality if the product is already in a good condition. Of course, this doesn't mean that electrolyzed water is not working against high concentration of pathogens, but this was not the case of our study. Ground beef shell life depends, we understood, on the pre-grinding microbiota composition. We observed also signatures of perishability, signature of shell life, and the presence, for example, of Lactococcus species. This is quite interesting because it means that we can understand probably the future of a batch of ground beef in relation to the initial microbiota composition of the trimmings or the carcasses. About the bioprotection, we found there is this possibility. We isolated in the early stages bacteriocin-produced strains, able to grow in this at low temperature, but it often has happened in this kind of study. In vitro results are not always confirmed, as in this case, by the application in vivo. So the best, if we want to have a takeaway message, the best possibility could be to create a benchmark database of what is a good and bad metataxonomic profile. And in relation to this, and together with the initial counts, bacterial counts, define a batch-specific shell life. This could be also a way to finally move the metataxonomics, the more user-friendly techniques of the omics family, from the academic research to the laboratory of companies. I want to say thanks to all the co-authors, and also thanks especially to the students involved in the study, to the several companies, and all the others collaborators. And of course, thanks to all of you for your attention. If you have any question, please raise your hand.
Thank you, Christian. We have time for one question.
It's a bit long.
Nobody? Yes, here. Please.
Yeah, thank you very much for your presentation. I have a question on the culture that you tried to use in beef, and it didn't work. Were you sure that the nisin, or the compounds that was produced in vitro, was produced also in ground beef?
Thank you. That's a good question, and this is why we are trying to figure out right now. When I say I hope to tell you also another story, I mean how this Lactococcus lactis ornea is producing. If it's releasing of its, we are thinking in the last experiment we observed that maybe it's something bound to the membrane of the wall, or the wall of the bacteria. So this is probably a reason why when we are in a liquid phase, it can reduce it can limit the growth, not limit the growth of Listeria, but when we are in a solid matrix like the ground beef, is not effective. We have to understand first the mechanism, and then try if we can figure out another application. Maybe on not on ground beef, or we can use on stick on the surface. This could it will help to have a contact between pathogen and the bioprotective strain.
So I have just a question for you. Did you check that there is no problem with the volatile compounds produced by Lactococcus?
We didn't check with the GCMS, so chemical. We perform anyway organoleptic. I didn't have here this information. We perform in parallel with the, of course, not in the sample inoculated with Listeria, only the one inoculated with the Lactococcus and the one control. We perform a sensory analysis, and a panel of more than 100 people couldn't recognize by odor, in this case, a difference. Of course, if the idea is to implement this in a real product, we have to pay really the attention on what is produced after the 10th day, for example, in terms of volatile compounds.
Okay, thank you very much. So thank you for this clear presentation and for answer. We can pass to the next presentation. So I may inform you that the second presentation, initially dealing with E. coli, has been postponed to tomorrow morning. So now we are waiting for Maria Eleni Dimitra Kopoulou. She will speak about traditional Greek products, but I can't name it.
Αυγοτάραχο μεσολογίου. That's it?
Yes, hello, good evening. My presentation is about DNA-based tools for authentication of Greek BDO Αυγοτάραχο μεσολογίου. A few things to start with. Food authentication is a process that validates label information about the food origin, geographical or genetic, and information about production procedures. In fact, it is also an essential requirement for food quality. Why do we choose DNA-based tools? At first, DNA is an extremely stable and long-lived biological molecule that can be recovered from a biological material that has been under stress conditions. And then DNA can provide us more information than proteins due to the degeneracy of the genetic code and the presence of large non-coded stretches. So, DNA-based methods could contribute against food frauds because of their advantages such as simplicity, accuracy, and robustness. The aim of our study was to explore whether unique biological barcodes or markers of a high-valuable Greek PDO product could be discovered. What is Αυγοτάραχο Μεσολογγίου? The eggs from fishes, Mugilcephalus, caught in the Mesologi lagoon are known with the trade name Αυγοτάραχο Μεσολογγίου. The raw material of the product is derived from flathead mullet caught in Mesologi. Production, process, and preparation of this Greek PDO product are completed at the same region, and this product has a great commercial value, over 100 euros per kilogram, and constitutes one of the oldest in the category of fresh fish. Our steps of experimental procedure. At first, we started with a sample collection, followed by DNA extraction, selection of gene target, PCR amplification, and finally, we choose 3 molecular techniques, PCR-APD, PCR-FLP, and PCR-DGG. So, this is the map. In red highlight, we can see the geographical regions where the samples were collected, Greek PDO from Mesologi, samples from Mauritania and from Australia. Protocol for PCR-APD analysis. At first, we performed Enterobacteriaceae analysis with microbial cultures in VRBG medium. We extracted DNA from cultures and we proceeded with PCR amplification using random 10-mer primers, M13, 1245, 1290, OPA10, and OPA15. For PCR-FLP method, we extracted genomic DNA of samples. We amplify our DNA targeted to COI gene. Our ablicons were 655 base pairs, and we used primers FIS F1 and FIS R1. Then we sequenced these ablicons from COI gene, and finally we selected appropriate restriction enzymes. Protocol for PCR-DGG was started by bacterial DNA isolation as well. We amplify DNA targeted to 16S rRNA gene. A more specific V3 variable region of bacterial 16S rRNA from raw was amplified using primers GC338F and 518R. PCR products were analyzed by agarose gel electrophoresis and then were analyzed by gradient gel polyacrylamide. Recently, emulsion PCR-DGG has been proposed as an alternative tool for microbial analysis. Through emulsion PCR-DGG, we can obviate and face up limitations regarding conventional PCR. Emulsion PCR is implemented in third-generation PCR system known as Droplet Digital PCR. By means of emulsion PCR, we can produce droplets of DNA molecules in thermostable water in oil emulsion. And finally, genetic material is divided in droplets which contain more than one gene and is amplified in millions of independent reactions simultaneously. So, results of PCR-APD analysis in this figure, we can see cluster analysis of 16 sRNA binding profiles of bacterial communities extracted from fish eggs from different geographical regions. At 52%, we achieve total discrimination among samples. Figure 2 shows multiple alignment of the sequence of COI gene obtained from fish from different geographical origins. In green highlight, we can see genetic variations between samples. And finally, in table 1, we can see predicted restriction sites of 4 enzymes, HIN, FALU, PAEL, and IQ3, and the generated pattern from PCR-RFLP analysis. As we can see, by using IQ3 enzyme, PCR product from Mesologi PDO was not digested. So we have also here total discrimination among samples from different geographical origins. Results from PCR-DGG analysis, we have two different electrophoresis conditions. In figure A, we ran electrophoresis gel 16 hours at 65 volt, while in figure B, we ran electrophoresis 5.5 hours at 190 volt. With red highlight is Greek PDO Votaro Mesologiou, with yellow samples from Mauritania, and with blue samples from Australia. Results of emulsion PCR-DGG. In this figure, we can see a comparison between emulsion PCR-DGG and conventional PCR-DGG analysis. By emulsion PCR, we achieved to obtain 3 additional bands, so we can assume that emulsion PCR is more accurate and sensitive regarding bacterial communities, analysis of bacterial communities. So, in general, DNA-based techniques can provide a biological marker for any food type. They can also be effective by analyzing fungi or yeast or other genes of a foodstuff. And finally, combination of these methodologies could be a unique biological barcode and which will be accurate and valid. We performed 3 different molecular DNA-based techniques in order to explore whether a unique biological barcode could be discovered based on either genomic DNA or DNA from bacterial communities. Band profile resulted in these molecular techniques could be used as a barcode to certify the origin and authenticity of Greek PDO of Votaro Mesologiou. And it is worth mentioning that it is the first study that is successfully applied DNA-based methods for authentication and traceability of Greek PDO of Votaro Mesologiou. Finally, considering the results of the study, adoption of DNA-based tools seems to be very promising for determination of food geographical origin and authentication among different samples. In terms of global food trade, implementation of these techniques in food industry could provide a great impact regarding quality SIMS, food labeling, food safety, and food fraud incidents. And in conclusion, further DNA analysis and application to a variety of foodstuff in a large scale may be the proper solution to manage adulterations and to strengthen consumer confidence as well. Thank you very much. Thank you for your attention.
Thank you. So we have time for 2 questions.
Okay.
I have one question. Are you planning or thinking to move also other than DGE on other metataxonomic, for example, analysis metagenomics to characterize?
We are planning to
relation to the
study
viability.
at first other genes, and then we are planning to go metagenomics.
And how can we eat this kind of Greek products?
So, I'm sorry, I don't
How
hear.
can we eat this kind of Greek product?
Of how, what
It's not a scientific question, it's how to eat.
A culinary question.
I think raw with spaghetti.
With spaghetti.
Okay.
Thank
you very much.
much.
Okay. I'm gonna announce you the next speaker, Samantha Dutoit, with a presentation entitled Whole Genome Sequencing and PCR RFP Characterization of Listeria monocytogenes, Isolates from Ready-to-Eat Hummus, Fresh Produce, and the Food Processing Environment in South Africa. The floor is yours. You have 13 minutes.
Thanks. As announced, my name is Samantha Du Toit. I am a MSc Food Science student at the University of Stellenbosch in South Africa, and it's such a privilege to share part of my research project with you this evening. In short, the surveillance study contributes to the knowledge and understanding of the genetic diversity of Listeria monocytogenes in ready-to-eat hummus, fresh produce, and the food processing environment in the Western Cape, South Africa for the years 2018 to 2021. For ease of speech, Listeria monocytogenes will be referred to as L. mono here onwards. The main objectives of this study were to lineage type 60 L. mono isolates using polymerase chain reaction restriction fragment length polymorphism, and to identify the sequence type and serotype of a subset of isolates using whole genome sequencing and bioinformatic analysis. As you may know, L. mono is ubiquitous in agricultural environments and persistent in food processing facilities. Consequently, a variety of foods are contaminated and become serious health risks to immunocompromised consumers. The listeriosis outbreak experienced in South Africa a few years ago was evidence of this. The pathogen causes flu-like symptoms in healthy individuals and life-threatening invasive disease in vulnerable groups. Although the incidence of listeriosis is usually low compared to other foodborne diseases, most cases require hospitalization, and the disease outcome is often more serious. Infection is almost always caused by the consumption of contaminated foods, including ready-to-eat hummus and fresh produce. Both categories have been linked to listeriosis outbreaks and sporadic cases in the past. These foods are particularly concerning as they are typically stored at refrigeration temperatures for long periods of time and are consumed with minimal processing or no heat at home step. Collecting surveillance data for foods less studied supports a precautionary approach to food safety. Lack of awareness could have severe consequences to public health. L. mono can be classified below the species level into 4 lineage groups and 13 common serotypes. These serotypes can further be divided into sequence types. Only the sequence types of relevance in this study are presented on this slide. Different subtypes of the bacteria are associated with varying degrees of virulence and different clinical outcomes. Lineage 1 and serotypes half A, half B, and 4B are most commonly linked to human listeriosis and are therefore considered most pathogenic. Recorded trends allow the lineage types, serotypes, and sequence types of isolates to predict their potential to cause disease. Subtyping of L. mono from foods provides some insight into the potential risk these products pose to immunocompromised consumers. All samples included in this study were received from an accredited food laboratory called Microchem. A sample set of 60 isolates was selected from many received over the years. Samples were grouped into 3 categories. Ready-to-eat hummus samples ranged from an unspecified or original variety to jalapeno, za'atar, peri-peri, orange, red pepper, and coriander chili. Fresh produce samples included potato, cucumber, coriander, cling peach, leeks, spinach, and unspecified vegetables pre-wash. Food processing environment isolates were obtained from a variety of different facilities. A few environmental origins included floors, drains, food contact surfaces, equipment, and cleaning tools. No duplicates were added to the sample set from any category. All isolates were received on rapid almonochromogenic agar. To ensure isolate viability, presumptive positive almono was restreaked onto rapid almono plates. Colonies exhibiting characteristic almono growth were then streaked onto 2% blood plates for enrichment. DNA extraction was performed using a quick DNA fungal bacterial mini-prep kit. PCR amplification of the HLY gene genotypically confirmed isolates to be almono. Lineage typing was performed using the RFLP method developed by Rip and Ghoes. Isolates were essentially classified into one of 3 lineage groups based on shared single nucleotide polymorphisms. The HLY amplicons were subjected to restriction digestion using 3 enzymes. Different enzymes cut at certain positions specific to different lineage groups, producing fragments of varying lengths. A subset of 20 isolates was selected for whole genome sequencing. The subset comprised of 10 lineage 1 isolates and 10 lineage 2 isolates from a range of years. The purity of all DNA extracts was measured before being sent for whole genome sequencing. Whole genome sequencing was performed by 2 service providers, namely Cosmos ID and SAF, using an Illumina platform and an Ion Torrent platform, respectively. In silico sequence typing involved submitting the raw reads for the 20 isolates to the MIST 2.0 tool from the Center for Genomic Epidemiology in FASTQ format. Serotypes were then inferred from this data. Binomial and chi-square tests for equal proportions and chi-square and Fisher exact tests for independence were performed to identify significant associations between categories and isolate typing results. A p-value of less than 0.05 was considered a statistically significant difference. PCR results confirmed all presumptive positive isolates to be almono. Of the 60 isolates lineage typed, 57% were found to be lineage 1 and 43% to be lineage 2. There is cause for concern considering the pathogenic potential of lineage group 1 and its overrepresentation in the South African food industry. Other South African studies have come to similar conclusions. The 20 isolates sequenced were found to be of 7 sequence types belonging to 3 serotypes. The most common sequence types were ST5, ST121, and ST204. No novel sequence types were recognized from this research. Serotypes half A, half B, and 4B are collectively responsible for 95% of all described human listeriosis cases. All fresh produce-related outbreaks listed previously were caused by one of these 3 serotypes. The serotype 4B ST6 strain, responsible for the 2017 to 2018 listeriosis outbreak in South Africa, was not identified in this study. However, clinical isolates analyzed during the time of the outbreak were represented by 9 sequence types, including ST1, ST2, ST5, and ST101. A South African study by Mafuna et al. Suggested ST1, ST2, and ST204 to be particularly virulent due to the presence of many main virulence factors. At an alpha level of 5%, samples for the food processing environment and ready-to-eat hummus did not provide sufficient evidence to conclude that the proportions of lineage types were significantly different. However, lineage 1 was significantly associated with fresh produce. For this category, all isolates but 1 were found to be lineage 1. Similar South African studies have also reported a prevalence of lineage 1 in fresh produce. Kyoda and Okey recently characterized all 90 Almano isolates analyzed from fruits and vegetables in the Eastern Cape province as lineage 1. Both this finding and the results obtained in our study should not be overlooked, especially considering the ready-to-eat nature of many of these foods. Both serotypes half A and half B were recovered from all 3 categories, while serotype 4B was only found in fresh produce. No single sequenced serotype was significantly overrepresented in any of the categories. Serotype 4B, isolated from potato and leeks in different facilities, is considered the most pathogenic serotype. This serotype has previously been recovered from various fruits and vegetables in many different countries and has caused listeriosis outbreaks associated with ready-to-eat salads, caramel apples, stone fruit, bean sprouts, and leafy greens in the last 10 years. No found studies have identified the sequence type or serotype of isolates from ready-to-eat hummus. However, one publication deduced a strong association between infection, particularly by serotypes half A and 4B, and the consumption of hummus prepared in a commercial establishment in America. ST204 was most prevalent in the food processing environment, ST5 in fresh produce, and ST5, ST101, and ST121 in ready-to-eat hummus. Again, no significant associations between sequence types and categories were identified. Mafuna et al. Identified various stress tolerance genes, benzalkonium chloride resistance genes, and biofilm formation genes in ST204, and reported this sequence type as well as others to be well adapted to the food processing environment. Another study by Naditz et al. Found ST5 and ST121 to contain plasmids contributing to food processing environment associated stress survival. ST3 was only recovered from the food processing environment. ST1 and ST2, both serotype 4B, were only found in fresh produce, and ST101 was only sampled from ready-to-eat hummus in this study. Factory origin information of the isolates was requested from the supplier after analysis for discussion purposes. The contamination of ready-to-eat hummus either originates from raw ingredients or more commonly from cross-contamination within the food processing environment. It was noted that the same ST5 serotype half B strain was recovered from a hummus blender and a ready-to-eat and hummus blender and 2 hummus samples, original and coriander chili varieties, and coriander in the same facility, this bottom ready-to-eat and deli food factory, in 2019. This finding suggests possible cross-contamination within this facility and emphasizes the importance of good manufacturing practices, appropriate factory design and production flow, correct storage of raw ingredients and ready-to-eat products, and effective cleaning and sanitation. It was noted that 3 of the 4 factory origins supported the survival of more than 1 Almano strain. It was also evident that ST5, ST121, and ST204 were able to proliferate in very different processing environments. Relatedness analysis revealed distinct associations between lineage types, serotypes, and sequence types. Lineage groups and serotypes within lineage groups clustered separately. Isolates of the same sequence type clustered with 100% similarity. The 2 serotype 4B isolates clustered closely and were more strongly related to each other than to the other isolates. This sort of genetic profiling is very limited for Almano in South Africa, especially for the food groups of interest. Serotype and sequence type results for hummus constitute the first of their kind. Only one very recently published South African study has serotyped Almano from fresh produce in the Eastern Cape using an agglutination technique, and no found studies have identified the sequence type of isolates in this category in our setting. In summary, lineage 1 seems to persist in the South African food industry for reasons that still need to be explored in future studies. The continued recovery of lineage 1 from fresh produce suggests this lineage type to be well adapted to proliferate in these foods and potentially their cultivation environments. the significant association between lineage 1 and fresh produce, and the recovery of serotype 4b isolates from potato and leeks present this category as a higher risk. The recovery of serotypes commonly linked to human listeriosis from ready-to-eat hummus also suggests this product to be a potential health risk to pregnant women and immunocompromised consumers, especially considering the ready-to-eat nature of this food. Serotypes half A, half B, and 4B are evidently able to overcome the stresses implicated in the South African food industry and exhibit a competitive advantage over other serotypes. Sequence types detected in this study are reportedly prevalent in other parts of the world and have been responsible for listeriosis outbreaks, suggesting them to be a potential threat to the public health of South Africa. I would like to acknowledge the following people and organizations and their contributions to this project. Thank you for listening.
Thanks, Samantha, for your presentation. We have time for one question from the audience. If no, thanks again.
Thanks.
I'm glad to announce you the next speaker. That is my co-chairman, Dr. Hélène Valentin from HIRE, with a presentation entitled Metatranscriptomics and Metabolic Modeling to Identify Bacterial Metabolic Interaction During the Manufacture of a Modern Processed Cheese. The stage is yours.
Hello everybody. So today I propose to explore the potential of metatranscriptomics and metabolic modeling to identify bacterial metabolic interactions during the manufacture of a model pressed cheese. So you know that the activity of cheese microbiota is responsible for most of organoleptic qualities of cheese. The cheese are characterized, as many other fermented foods, by successive growth of microbes, specific and complementary microbial metabolisms. So we could ask the question, what are the metabolic interactions between these microbes and what are their role in the functioning of the global ecosystem? So today our scientific questions will be, which species contribute to the acidification? Which species contributes to the aroma compound production, which metabolic pathways are activated and when, and finally, which bacterial metabolic interaction contributes to the functioning of these microbial ecosystems. So our strategy was to sample cheese throughout a making and a ripening. First, we did numeration to assess culturable bacteria, and we also did a targeted metabolomic to quantify sugar, acid, and aroma compounds. Finally, we performed a metatranscriptomic analysis by sequencing RNA from bacterial cheese to give and to obtain a snapshot of metabolism of each of the species identified. So the cheeses were semi-hard cheeses, and we performed the manufacture in our laboratory in a pilot plant. The milk first was inoculated with 3 strains, Lactococcus lactis biovare diacetylactis, Lactiplantibacillus plantarum, and Propionibacterium fraudenreichi. The making was traditional. At the end, the cheeses were vacuum packaged to prevent yeast and fungi. And there was a 2-month ripening period. So we sampled the cheese throughout the making, and at all sampling steps, microbiology and biochemistry were performed. We also did molecular biology and metatranscriptomic only at 4 steps, at molding, demolding, and 2 of the ripening steps. Microbiology enables us to monitor growth of each species. Biochemistry and in particular chromatography gave quantification of sugar, acid and aroma compounds. We also did complete DNA extraction to get a complete genome of each of the 3 species, and RNA extraction. So the DNA was sequenced by both parent Illumina and PacBio, and RNA was only sequenced by Illumina. The computational methods, so we did a genome assembly using Unicycler, an automatic annotation of the genome using microscope platform, and a reconstruction of metabolic pathways using the pathway tools. The metatranscriptomics was done by mapping all the reads obtained by Illumina on a genome sequence using Bowtie, and the reads were confirmed on each genes of the genomes. A normalization step was performed, and all the results I will show you today is a set of differential genes identified using EdgeR, and all the results will be expressed as a logarithm of fold change. So we checked the repeatability of semi-hard cheeses before doing molecular biology, and the repeatability was very good in terms of humidity, fat, dry matter, and chloride, but also in terms of bacterial growth. Most of the examples I will show you today will be taken during these steps at acidification steps between molding and demolding. So the genomes obtained were complete with chromosomes and plasmids thanks to Unicycler assembly. And we did the mapping of RNA reads on these genomes, so we can observe all induced and repressed pathways throughout the making and the ripening, but I will choose for you some example of carbon metabolism involved in bacterial interactions. So first, the milk contains 2 sources of carbon. One is a citrate and the other one is a lactose. So first, the citrate was, the metabolism of citrate was observed, and the citrate was rapidly utilized between molding and demolding, according to the previously described pathway. In lactis, the induction was a twofold. The same pathway was also induced in L. plantarum, and in this case, we can say that the metabolic interaction is a competition for the citrate. And we have a functional redundancy using the same pathway induced exactly at the same time in L. lactis and in L. plantarum. Secondly, the lactose was also utilized by L. plantarum, and for the majority between molding and demolding. So the lactose was split into glucose and galactose, which was then metabolized for lower pathways. All genes were induced 5 to 7-fold in L. plantarum and 15-fold in P. freudenreichi, and surprisingly, at these steps, it was stably expressed in L. lactis. However, in L. lactis, we observe another pathway which was scarcely described in literature and which was induced at the end of the ripening. It is the tagatose pathway involving a permease and a phosphorylation of lactose. All these genes were placed on arbor on plasmid. My last example will be the production of acetylene, a typical cheesy aroma compound, And we noticed production of acetoin just after the molding. The previously described pathways was induced 2 to fold change for L-lactis and L-plantarum for the first steps in red, and was highly induced in P4.9C with 14-fold. In this case, for acetoin, we can imagine a division of labor if we think that the acetolactate can pass through the membrane and through the cell wall of lactic acid bacteria to fulfill the P4.9C. This interaction is characterized by a functional redundancy with specific induction of genes. So to resume just one slide, we get the successive carbon metabolism and the interaction. So first, the lactic acid bacteria produce lactate from lactose and citrate, and also acetoin. The lactate and the lactose was utilized in P4.9C to produce propionate and acetate. And at the end of the ripening, an acetoin production still arrived. So the bacteria seems to have a good team spirit for degradation and production, but one question remains: do they exchange additional metabolites, more than lactate and acetolactate? So we performed metabolic modeling to identify metabolic exchanges. So metabolic modeling was previously presented by a colleague from Israel, I think, today. So we converted the annotated genomes into genome-scale metabolic model, first at individual species for individual strains, and secondly for ecosystem, by mixing the three models. And under biomass constraints, we checked that flux were in agreement with metatranscriptomics and targeted metabolomics previously described. So we can show here a flux of lactose and also a production of lactate and acetolactate pathway. This metabolic model at ecosystem level enables us to detect a potential exchange. The exchange of lactate between lactic acid bacteria and propionic acid bacteria has been previously published, but we also identified an exchange of ribose, succinate, glycerol, and at the expense of both lactic acid bacteria, and also probably an exchange of serine and phenylalanine to the benefit of P4.9C. So we have to confirm, going back to wet lab. The take-home message today was metatranscriptomics enable us to identify induced pathway in each species, and it gives also a snapshot of successive metabolism during the cheese making and ripening. We also identified microbial competitions like citrate and lactose utilization, and division of labor for production of acetoin. The metabolic modeling gives us clues to metabolic exchange between the species. So the perspective, the strategy applied here can be applied to any fermented foods to decipher metabolisms and interaction. And since most identified pathways are species and strain dependent, this kind of knowledge could be very important to select and to combine strains to develop new fermented food products. So I will thank all my colleagues from INRA and INRIA, and in particular our engineer Wen-Fan Cao and my PhD student Maxime Lecomte. And thank you for you and for your attention. Thank you.
Thank you, Hélène, for your really nice and detailed presentation. we have time for a couple of questions from the audience. If no question from the audience, I have one curiosity. No, not from the audience. Okay. I saw in one graph production of butanoate, if I catch it. Do you have any, idea if it can be produced from one of these three, or about the pathway that can be involved? If it's
Which metabolite?
butyric acid, butanoate.
Butanoate. We don't know exactly the pathway for this kind of product, probably because we have Propionibacterium fraudenreichi, and we know that this species is hardly involved in lipolysis in cheese, in pressed cheese. The butyrate probably comes from lipolysis of milk fat, but we are not sure.
Okay. Any question? No more? Okay, thanks again.
Okay, thank you.
Ms. Moine, thanks again. Sorry, but I don't have the agenda. Okay, so let's move on with the next presentation. Paulina Sredrinka with the presentation entitled Optimization, it's online, okay. Optimization of the in vitro model of gut microbiota culture and application to toxicomicrobiomics studies. Paulina, the stage is yours. Can you hear us? Yes. We cannot hear you. Still no.
Is your microphone open?
Yeah, I can hear something, but it's really low.
Yes, it's better.
Better not that.
Good afternoon. My name is Paulina Średnicka.
Okay.
I'm PhD student in Institute of Agricultural and Food Biotechnology in Warsaw, Poland. My today's presentation is entitled Optimization of the in vitro model of gut microbiota culture.
Just a moment, sorry, Paulina, because we cannot see here from here the slides. So, okay, we wait for technical help.
Did you share your presentation?
Yes.
Yes, it's okay now.
Now it's okay, we can hear you, we can see the presentation. Okay.
Okay, so to begin with, I will provide a short introduction about gut microbiota and its interactions with xenobiotics. So, microorganisms gastrointestinal tract constantly interact with each other and with the host, playing an important role in maintaining.
Sorry, we can't hear you again.
We still have problems, but I think it's not your fault.
Can you hear me?
Yes.
Yes, now, yeah. Let's try like this.
Okay. Microorganisms inhabiting gastrointestinal tract can interact with each other and also with the host, playing an important role in maintaining human health. So this symbiotic balance can be easily disturbed by numerous factors leading to dysbiosis, which may have adverse health effects. So currently there is a great need to optimize methods of in vitro culturing of human gut microbiota to investigate and understand its link with pathogenesis of various diseases, but also in the comprehensive evaluation of environmental pollutants toxicity. So a branch of toxicology that explores such xenobiotic microbiome interactions is called...
Sorry, I have to interrupt you again. I'm really sorry. Can you just move a little bit down the camera so that we can see also your face? Perfect. Okay, let's start again with the time, and sorry.
So can you hear me and see me?
Yes. Okay.
Yes? Okay. So the branch of toxicology that explores xenobiotic microbiome interactions is called toxicomicrobiomics. I want to emphasize that one of the important roles of human exposure to xenobiotics is its migration into food from food contact materials. So in my research, I study mainly bisphenols, which are an example of xenobiotic migrating into food from food packaging. Xenobiotics ingested by the gastrointestinal tract have a significant impact on the intestinal microbiota. These compounds can be directly metabolized by microorganisms, which in turn may lead to the activation or biological inactivation of these compounds, as well as the production of toxic metabolites. Xenobiotics also indirectly influence the taxonomic structure and metabolic activity of intestinal microbiota. They can disturb the production of many signal metabolites, which are important for the host health. So the aim of my research was to optimize the methods of growing the gut microbiota so that the culturing process would allow us to maintain the high biodiversity and metabolic activity of the microbiota, and that it could be used in further studies evaluating impact of various xenobiotics. So gut microbiota samples were collected from 15 healthy donors, pulled together and frozen in a cryopreservation buffer. The anaerobic cultures were carried out using 4 different media selected from the literature. The incubation was for 24 hours in anaerobic workstation. The samples were analyzed for changes in taxonomic profiles using Illumina MySeq platform. Short-chain fatty acid content was analyzed using gas chromatography, and analysis of the untargeted metabolomic profiles was conducted using liquid chromatography. The tested media were inoculated with pulled stool samples marked as mix and incubated in vitro batch model for 24 hours. Then post-culture samples were treated with PMA viability dye to remove non-viable bacteria DNA from sequencing and to reduce viability bias. obtained outcomes were compared to the non-cultured mix inoculum, representing the initial structure of gut microbiota community. Here on the screen you see taxonomic composition of gut microbiota,
Sorry.
which is presented as mean of 6 replicates.
Sorry, we can't hear you again.
Can you hear me now?
Stop.
Really low volume. I don't know if it's a technical problem here or the microphone from your side.
No.
Here it seems okay, so try to...
Can you hear me now?
No, it's not enough.
Not well. Audience cannot hear you, I think.
No, it's impossible.
I don't know if it can...
Is it better?
Yes, it's,
okay, now.
it's better, yes. Sorry. What's that?
I'm sorry.
So you explain the mix of different medium.
Yes, here on the screen you can see taxonomic composition of gut microbiota presented as a mean of 6 replicates at phylum level. Only the relative abundance of Bacteroidetes and cyanobacteria was significantly different between media groups and non-cultured inoculum at phylum level. Next are 5 alpha diversity metrics were calculated. The non-cultured mixed inoculum was characterized by a high species richness and evenness by applied diversity measures, and after 24 hours of culture, no significant differences in the SM observed in terms of CHOW1 index, observed features, pillow, evenness, and fate PD index. However, in general, in vitro cultivation of gut microbiota was associated with depletion of the microbial community. In all of studied media groups, we noticed a significantly lower Shannon index, which is a quantitative metric of the species richness and the relative abundance. Next, we use PyCross analysis based on 16S rDNA gene data, which predicted significant differences in the functional potential of the gut microbiota, non-cultured mix inoculum, and 4 media groups. The results of PyCross prediction was an increase in relative abundance of genes associated with xenobiotic biodegradation and metabolism pathways, and it was observed in all media groups. Next step was evaluation of production of short-chain fatty acids by gut microbiota in 4 different media. Short-chain fatty acids are the end products of anaerobic microbial fiber fermentation, and they have been found to play important roles in various physiological processes, especially energy supplement, gut barrier integrity, and gut-brain axis. The highest total short-chain fatty acids production after culture was observed in SM, whereas the lowest production was observed in carbohydrate-free basal medium and fermentation medium groups. Next step was the evaluation of an untargeted metabolomic profiles. A total of 37,000 compounds were detected in fecal samples after fermentation in 4 different media, whereas almost 4,500 compounds were significantly different between tested media. However, almost 80% of these metabolites were unidentified. So differences between gut microbiota metabolomes are shown on the left on the PCA plot. We see similarities between SM and FM groups, whereas
sorry, Paulina, but the connection is very bad, and we can't hear you very well. So I think
maybe if you want to get to the conclusion, give the final message. If we can still hear you, because we are running out of time, and the connection is really going and coming back. And okay, let's try. I'm sorry.
So our results showed that the effects of different cultivation media on global human gut microbiota community composition and in metabolomic activity. In our study, we observed that the use of low carbon media like fermentation medium and led significant depletion of gut microbiota community and reduction of short-chain fatty acid production, whereas Schedler medium marked as SM allow us to preserve the highest biodiversity and metabolic activity of microbiota. Moreover, we observed significant enrichment of the biodegradation pathways of xenobiotics in this medium.
Thank you, Paulina. Despite all the problems, we can understand the big amount of work, and it's a really nice presentation. Since we are running out of time.
Yes, I think we have to keep the flow, and perhaps you can use the tools of chat you have on the app to connect people and to exchange with them. It will be easier, I think, by chatting.
Okay, thank you.
Okay, sorry about that. And thank you very much. So the next speaker is Meta. The next speaker is Meta Sterniza from a University of Slovenia. And she will speak about the Pseudomonas fragi biofilm.
Hello, thank you for the introduction. So my name is Meta Sterniza. I'm from the Biotechnical Faculty, University of Ljubljana, and today I will present topic about Pseudomonas fragi biofilm and how it improves Campylobacter jejuni stress response in aerobic conditions. So first about Campylobacter. This is still the leading cause of human gastroenteritis reported by different organizations, and it's a zoonotic infection. Most of cases are sporadic, and they are connected to consumption of undercooked poultry meat or because of cross-contamination. And outbreaks are mostly connected with unpasteurized cow's milk. But we can find Campylobacter also in water, and it was even reported in shellfish. What's characteristic of this bacterium that's on one side maybe making problems in the laboratory, but on the other side makes really big challenge for these bacteria in the environment is their microaerophilic and thermotolerant demands for growth. That's why these bacteria must overcome several stresses during their foodborne transmission. Now on the other side we have these really good adaptable bacteria which cause spoilage of food, and Pseudomonas fragi is one of the most commonly reported spoilage of Pseudomonas species, and it's psychotrophic. In recent years, there's been an increase in reporting that these bacteria can provide advantageous conditions for other bacteria. We were interested in the combination of these two bacteria that can be found in poultry meat, and we were interested at their growth at low temperatures. So our two first combinations were to test how these bacteria grow if they are co-cultivated, incubated at two different temperatures, 5 and 15 degrees, and we used two different growth media, so Miller-Hinton broth and chicken meat homogenate on stainless steel. And the second variant was to first prepare a preformed Pseudomonas fragi biofilm, which was incubated for 1, 3, or 7 days, followed by addition of Campylobacter jejuni for 24 hours. Everything was done with, lastly, with colony-forming units to evaluate the results. Now, as you can see from the graph, and that was reported, was done with our GLM analysis, you can see that numbers for Campylobacter jejuni alone could not be determined after 3 or 7 days, but we could determine these numbers when Campylobacter was in combination with Pseudomonas fragi. You can see that numbers are lower at 5 degrees compared to 15, and at 5 degrees there was also a difference between what we obtained in chicken meat homogenate, where values were lower compared to broth. This quadratic trend at 15 degrees, we were able to correlate it with how Pseudomonas fragi biofilm biomass was formed, but these results are not shown on the graph. So our speculation here is that Campylobacter jejuni, when it's alone, it cannot adapt to the stress that's happening in aerobic conditions, and it transits into viable but non-cultivable state. But when in combination with Pseudomonas fragi, it gets this support from Pseudomonas, and it can retain its cultivable state. Now, interestingly, when we use this second scenario, where preformed Pseudomonas fragi biofilm was used, there was basically no difference between 5 and 15 degrees, and also Campylobacter would increase in number only when Pseudomonas biofilm would also increase in biomass. So we showed that Pseudomonas fragi improved cultivability of Campylobacter jejuni at low temperatures, so at 5 and 15 degrees. And it's also known that Pseudomonas can reduce oxygen tension. This was reported before in the literature. We also could measure this reduction by 38 to up to 59%. So our belief here was that this oxygen gradient is done in Pseudomonas biofilm that somehow improves stress response of Campylobacter in aerobic conditions. But as I said in the beginning, Campylobacter can be really moody when working with it in laboratories, so we couldn't perform these new additional experiments at low temperatures. So we had to do optimization step before proteomic analysis, and we chose 30 degrees in aerobic conditions for this analysis. Just short points. So we prepared plates for incubation. Proteins were isolated and purified. We did database searching, and at the end, quantitative proteomics with statistical analysis. We, all in all, I determined 149 proteins. 9 of them were downregulated and 19 up. From downregulated proteins, there were some for oxidative stress, acid stress, translation, and also in upregulated proteins, there was a bit more diversity, again, with oxidative stress, translation, phosphorylation, general stress response, transport of proteins. But as said in the beginning, we were more interested primarily in this oxidative stress response, which is quite unique regulatory mechanism in Campylobacter jejuni, which is considered from 5 regulators, and then antioxidant enzymes that on one side present superoxide defense, and on the other side peroxide. And also we have proteins for purification and protection of damage in DNA. And from our results, we could see that superoxide dismutase B was the one that was upregulated, meaning that this first line of defense in this line was improved, and also BCP and TPX, which are the ones important for aerotolerance in Campylobacter, were upregulated. Additionally, the protein responsible for detoxification of oxidized methionine proteins was upregulated. But of course, these are not the only, we cannot look only at this oxidative stress response. There are also other proteins which were up and down regulated. I will not go through all the maybe chaos in this figure, but what's really stressing is that all this information lead to this, that in these conditions with Pseudomonas fragi, Campylobacter jejuni can maybe improve its stress tolerance. It can also lead to increased virulence and increased antibiotic resistance. But there's still some work for us to do here to really confirm this information. So we show that Pseudomonas fragi reduces oxygen tension and improves Campylobacter jejuni cultivability at low temperatures. This upregulation in superoxide defense is this first line of fight against toxic byproducts from oxygen reduction cycle, and it's probably the one that's better to prevent additional steps in this reduction. This adaptation, as I said, can lead to increase of virulence and antibiotic resistance. So all this information indicate for us that this joint growth could increase the possibility of Campylobacter jejuni transmission to humans. And we believe that these results can also be applicable to other Pseudomonas that can be found in chicken meat or cow's milk or maybe even water, and also to other bacteria which can adapt well to these environments where we can find Campylobacter and provide it with protection. And there is, of course, really big question about what's happening with horizontal gene transfer, because Campylobacter is naturally competent bacteria. So my question, which I ask all of you is if Pseudomonas fragi or other spoilage Pseudomonas are really just this quality problem, and if we talk enough about these bacteria, which on one side, of course, cause really big economic issues, but on the other side, we think that they have really big influence on pathogenic bacteria. So please see also more of our group's work on posters. And I would like to thank the organizers for this opportunity to talk about our work. Our group, Food Micro at Biotechnical Faculty, Dr. Marjorie Fournier, who helped us with proteomics, and of course our Slovenian Research Agency for financing. And thank you.
Thank you, Meta, for this clear explanation. Do you have any questions? Yes, here.
Some Pseudomonas fragi strains are known to produce siderophores that can help iron transport. And sometimes the siderophores, it's not only for Pseudomonas, they can give their siderophores. So do you think the mechanism you saw can be a consequence of this?
Yeah, we saw that it influences also iron metabolism, but we have to do some additional bioinformatics to see about it. Yes.
Is there any questions?
I have one question. Are you planning to investigate, maybe you said but I didn't catch, other mixed biofilm with Campylobacter and others like Listeria or...
You mean other bacteria?
Yeah, to study interaction with Campylobacter, this kind of biofilm pairwise interaction with Campylobacter with some others.
We know that similar happens with pathogens like E. coli and Listeria. When you put them together with Campylobacter, we know that they, on electronic pictures from electronic microscope, you can actually see that Campylobacter is in the middle and the other bacteria go around, or maybe Campylobacter is the one that goes inside. And also with Bacillus, also from our group research, we know that it can support Campylobacter. So I think that, as I said, bacteria that can adapt well to such environments that we can find Campylobacter in, that Campylobacter take advantage of this and kind of hides in these biofilms. With also chemotaxis was activated, so yeah.
Okay, thank you.
Okay, thank you for all. So we can acknowledge all the speakers of the session and also all the participants for their questions. So I think it's a last talk and we can... Pascal has something to...
If by chance somebody found a cellular form, it may be mine, so just you can put it at the registration desk, or if it's in here, you can give it to me. So, thank you. Sorry for this.
So have a nice evening and see you tomorrow morning. Bye.